Good afternoon, everyone. I think probably there is no one is going to chair this conference, and its time is up. So I think I'd better start. I'm the first speaker. I'm from EM Polytechnic, Singapore. So the title of my talk today is Zero Waste Upcycling of Food Byproduct through Fermentation. So today I'm going to go through this fermentation, what is fermentation, what is food byproduct, and I'm going to cover upcycling of food byproducts through industrial biotechnology and food biotechnology. So fermented food, fermentation is nothing new. It is used to produce the fermented food, such as yogurt, cheese, fermented fish and sausage, kimchi, beverage, fermented milk, miso, natto, and beer and wine, right? It's not something new. So what is fermentation? In this talk, fermentation is bioconversion in the presence of live microorganisms. So food byproduct refers to, when we process food, food byproduct is the secondary product obtained as a result of manufacturing of the main product, often with a market value. So this includes the fruit pomace, vegetable peels, husks, bran, and germ of cereals, etc. The vast majority of these byproducts are lignocellulose. They contain cellulose, hemicellulose, and lignin, and also vitamin, protein, minerals, and nutrients, etc. They are rich in polysaccharides.
Hello, sorry to disturb you, ma'am. I'm Dr. Neha from Amity Institute of Food Technology. Ma'am, we will be actually our inaugural session is still going on. So this is a separate link, and all the participants, including our rest of the speakers and our head of the institution and the convener and the whole team will be joining this link soon, within 5 to 10 minutes. So it is requested that, ma'am, please hold for the session. We will be starting the session soon, because at present our the chief guest for the conference, they're in a deliberation with the question answer round is still going on. So it's a request, ma'am. We will be starting it soon. So the chair for the session chair will also be joining soon. So it's a request that you can give us 5 to 10 minutes, ma'am.
Oh, okay. Sorry. I think there is no chairman for this session, so I'll just start.
No, ma'am, we have a session chair. Dr. Jayprakash sir is there with us. He has joined. So just since the inaugural session was in a separate link, so just give us 5 minutes. We have 5 to 10 minutes. Yeah, ma'am. The founder president of the university is also us, and he's speaking, so we can't interrupt in between. So I'm so sorry for the inconvenience to all of you, sir. Just give us 10 minutes, 10 more minutes, please.
All right, so
thank you.
So, yes, please, I will wait. Let me know when should start.
Thank you. Yeah, definitely, ma'am.
Okay, ma'am. Then now we can start. Yes. Okay, yes. Sure, ma'am. Okay, a very good afternoon, everyone present here. I'm Mandeep Kaur on behalf of Amity Institute of Food Technology, Amity University. Warmly welcome all the dignitaries, delegates, and participants from industry, academia, research institutes, and the students to this technical session 1 of International Conference Food Sustainability Challenges and Opportunities for the Future. The theme for technical session 1 is climate change and sustainable food production. Now, I would like to request Dr. Neha Sharma, Associate Professor, Amity Institute of Food Technology, and the coordinator for this session, to introduce the theme of this session.
A very warm welcome to our honorable guests, Dr. Jayprakash, Dr. Enli Gang, and Dr. Deepak Mudgal, and my other panelists. I also welcome Dr. V.K. Modi, our head of institution, and my faculty coordinators. Along with me, Dr. Monica Thakur and Dr. Renu. So for today, the theme is climate change and sustainable food production for the present technical session 1. As we all know, in the current scenario, there due to the due to the activity of the human beings, there has been various impact on the environment as well as the climate, and there has been drastic change. So in our today's technical session, where our motto will be to emphasize what will be the effect of climate change on the overall food production and what other steps can be taken to obtain a sustainable food production in the coming years. As predicted by one of the agencies, by the year 2050, we have to feed a population of more than 9 to 10 billion globally, and at the same time we have to reduce the environmental impact on the food production. And these environmental impact includes like greenhouse gas emissions, biodiversity loss, land use change, and loss of ecosystem services. And we need all these sustainable food production practices so as to ensure food security for all the people. As per the United Nations definition, food security it exists when all the people at all times, they have physical and economic access to sufficient, safe, and nutritious food to meet their dietary needs as well as food preferences to lead an active and healthy life. Ensuring food security along with reducing the environmental impact of food production is today's demand. Now, just tinkering with the existing food production practices that we are practicing globally will not help, and we need to take now out of the box thinking has to be done to meet the sustainable food production in coming future. All these things in like reducing greenhouse gas emissions, biodiversity loss, all of them in themselves, each of the problem they give it's a huge challenge. Okay, and we need to have out of the box thinking. We need to develop those food practices where we can sustain our food demand and supply, as well as we can maintain our ecosystem so that our future generations are not affected and they are being benefited. So by 2050, feeding a population of around 9 to 10 billion people is already an enormous challenge. So in our today's technical session, we are going to discuss different aspects of climate change and food sustainable food production. And we will listen to the ideas of and the views of our honorable speakers. So with this, I invite Dr. Jay Prakash, our session chair for today. Dr. Jay Prakash, I welcome you, sir. So
okay. Thank you very much, ma'am.
Yeah. Just a brief introduction for Dr. Jay Prakash Verma. He is working with Banaras Hindu University, one of the most prestigious university in India, for more than a decade. He has been a visiting fellow at HIE, Western Sydney University, Australia, and for a period of 1.5 years. He has expertise of more than 11 years on microbial consortium, PGPR, phytomicrobiome, sustainable agriculture, plant soil microbiome interaction, etc. To his credit, he has already published 82 research and review articles, 26 book chapters. He has been editor of 2 books. He has published 56 abstracts, 2 articles, and 40 Hindi news coverage of fertilizer popularization among 4,000 farmers of Eastern UP. He has submitted more than 300 bacteria strains in NCBI. At present, his citation is more than 5,500 as per Google citation, and he has a cumulative impact factor of more than 350. He has been associated with more than 12 research projects related to plant microbe interaction. He has visited many countries like Spain, Paris, Shanghai, and Australia. He is, along with all the research work he has been carried out, to his credit, he has also been involved in administrative duties like being a placement coordinator. He has been a member of IQAC of the institute. He has been also the in charge of Environment and Sustainable Development Institute, BHU. He is also a task force member of Patent and IPR Committee, DHU. So it's my pleasure to invite you, sir, to be the session chair for today's session. So welcome, I welcome you, sir. Over to you, sir, now.
Okay, thank you I am audible?
Yes, sir.
Okay, thank you very much, Dr. Neha, and for a nice introduction to me. And thank you for the invitation as the session, the chairs. So I just invite, want to first lectures of these sessions, Professor Anil Jain. So I just give some biography. Dr. Jain is here. Yes, Anil Jain is here. And Dr. Anil short biography with detail. Dr. Jain Anil is currently the president of the Bioenergy Society of Singapore, and she is the senior lecturer at the School of Life Science and Chemical Technology of Nagi and Polytechnics. She is a co-founder and director of Mycosortium Private Limited and Sunvisia Biotech Limited. Prior to join Nagi and Polytechnics, she worked at the Institute of Environment Science and Energy as the research scientist and group leader. Dr. Jain obtained her PhD in the Chemical and Environmental Engineering from the National University of the Singapore under a government scholarship. She received her MSc, MTech Engineering and BTech Engineering degree in Biochemical Engineering from Tianjin University, PR, China. Dr. Jain has more than 20-year R&D research and development experience working extensively on the environmental biotechnology, green energy technology, and industrial biotechnology. Having expertise in the microbial strain development and fermentation, her current research focus at Nagi and Polytechnic is the development of novel biocatalysts and bioprocesses for bioconversion of lignocellulose to value-added products such as novel nutraceutical ingredient and novel foods. With these short biography, I invite to Anil Jain for your delivering the lecture. Over to you, Dr. Anil Jain.
Okay, thank you, Chairman, for your kind introduction. Okay, I'm going to share my screen.
Okay, yeah.
Okay, good afternoon, everyone. It's my pleasure to present my talk, Zero Waste Upcycling of Food Byproduct through Fermentation. For the sustainable production of food, it's very important to fully utilize what has been produced. So in my talk, I will talk upcycling of the byproduct through fermentation. So I'm going to talk about what is fermentation, what are the food byproducts, and I will cover the upcycling of food byproducts through industrial biotechnology and food biotechnology. So fermentation is nothing new because it is the technology that convert to produce our fermented food, such as the yogurt, cheese, sausage, kimchi, beverage, wine and beer, milk, natto, miso. Okay, so we actually are very familiar with fermented food. So what is fermentation in this talk? In this talk, fermentation refers to the bioconversion of in the presence of live organisms, okay, live microorganisms. So sometimes bioconversion can also be done by enzymes only, but in this talk, it is in the presence of live microorganisms. So what are food byproducts? Food byproducts is a secondary product that's obtained as a result of manufacturing of the main product. So they are often with a market value. So these include, fruit pomace, vegetable peels, husks, bran, and germ of cereals, etc. The vast majority of such food byproducts are lignocellulose. They contain cellulose, hemicellulose, lignin, and vitamin, protein, minerals, and nutrients, etc. They therefore still have quite a substantial value. Okay. So they are rich in polysaccharides, sugars, protein, enzymes, dietary fiber, fatty acids, flavor, aromas, and bioactive compounds. So utilization of such food byproducts for food application is very important. So, so basically for agriculture and food and industrial residues, the food byproducts are produced from food industries. And the agriculture is actually from the agriculture industry. Okay, so take a look at the composition of the byproduct. So they are so-called lignocellulose. So lignocellulose contains cellulose, hemicellulose, and lignin. These are the three main components of the food byproduct. Besides that, they also contain some protein, vitamin, minerals, so on and so forth. Now I'm going to talk about upcycling of food byproduct. Okay, there are two aspects. One way is to produce, to upcycle the food byproducts through industrial biotechnology. The other is through food biotechnology. In industrial biotechnology, we use the food byproduct to produce enzymes, chemicals, fuels, biopolymer, pesticides, biolubricant, biosurfactants, etc. In food biotechnology, the food byproduct can be utilized to produce single-cell protein, microbial lipids, pigments, bioactive compound, flavoring compound, prebiotics, probiotics, and soluble fiber. This is the industrial biotechnology. The food byproducts such as the vegetable fruit peels, okara, spent grain, spent barley grain, beet from brewers, will be processed by the microorganisms. In this case, microorganisms are considered as a cell factory, microbial cell factory. The microorganism will convert the food byproduct to fuels, chemicals, and materials. This is so-called industrial biotechnology. So in this table, you can see, so these are a series of mushrooms, actually. Mushrooms use the food byproduct to produce peroxidases, enzymes, okay, lactase, xanilase, protease, okay, and and also other enzymes, okay. So a lot of enzymes. So the food product can be used by mushroom. Using mushroom produce enzymes, okay. Then this is one of my early research. So I actually in Singapore we don't have agriculture. We use a horticulture waste to produce lactase and the solid state fermentation. Also, the food byproduct can also be utilized to produce cellulolytic enzymes, which are celluloses. Okay, so include cellulase, cellulose, and beta-glucidase. So you can see these are all the food or agriculture byproducts are being used. In this case, a lot of cases is used filamentous fungi such as trichoderma, penicillium, aspergillus, so on and so forth. They can also be used to produce a hemicellulosic enzyme, for example, xanilase. Okay, so these are food byproduct. Okay, so this is another example in my research. We use a horticulture waste to produce the cellulase and the hemicellulase. So the food byproduct can also be used to produce other enzymes, for example, pectinase, lipase, mannase, protease, and tannase, inulinase, so on and so forth. Okay, so these are why those food byproduct can be produced to produce so much of the enzymes, because they contain the fiber. The fiber can be released, digested to produce sugar. That's why the microorganisms can utilize such food byproduct to produce enzymes. And also the food byproduct can be also used to produce fuels and chemicals, for example, succinic acid, lactic acid, and ethanol. And the other chemicals, for example, biosurfactant, the biopesticide, and biolubricant. So now let's take a look at the structure of lignocellulose. So these are bundles of the cellulose, and these are the hemicellulose, and these are they are connected by lignin. So hemicellulose, cellulose, they are the bundles of this fiber polysaccharides are actually connected by lignin. And the cellulose is mainly confirmed the polymer of glucose, but the hemicellulose is a polymer of a number of C5 and C6 sugars. Lignin is the polymer of aromatic compound, but it always have a complex structure. That's why to break down this to sugar, it takes a lot of energy. Need very special enzymes to work on it. Besides that, cellulose, hemicellulose, and lignin, they also have extractives. So the research of the industrial biotechnology to convert the lignocellulose biomass to chemical fuels involves pretreatment, because the structure of the biomass is very recalcitrant for digestion. That's why they must be pretreated, such like using chemical or thermal method to break down the structure, to separate cellulose, hemicellulose, so that those polymer are accessible by the enzymes. After the pretreatment is hydrolysis. In this case, the enzymes such as cellulase and hemicellulase is applied to digest the cellulose and hemicellulose to monomer sugars. So this includes the C6 sugars, glucose, galactose, mannose, and the C5 sugar, xylose and arabinose. After the monomer sugars are obtained, then yeast or bacteria are added into the hydrolyzate, so to convert them to ethanol. So this is so-called industrial biotechnology, biofuel, bioethanol production. This is one of my research focus in the past 1 decade. Okay, so this is another example of we use the oil palm empty fruit bunch as the raw material, and we produce the enzymes on site using Trichoderma ricci to using cellulose as the substrate and wheat brown to produce the cellulase on site. And this oil palm empty fruit bunch will be treated by alkali and temperature, right? At the elevated temperature, then the pretreated oil palm empty fruit bunch will be digested by adding this crude enzyme prepared on site, and that hydrolysis will be conducted at 50 degrees C and for 96 hours for 4 days. Then the OPFB hydrolyzate was obtained. After the hydrolyzate was obtained, then a hybrid yeast SCF2 was added to fermentate to ethanol. So that's the process. Another examples, we use the oil palm empty fruit bunch, follow almost the similar method. The only difference is we use a bacteria. In this case, it produces a polymer PHB. Okay, so in this case, you can see this is C6 sugar, this is C5 sugar, and this bacteria can use both C6, C5 sugar and produce this polymer PHB. So one more example is we use this oil palm empty fruit bunch. Same method, the oil palm empty fruit bunch is pre-treated and then enzymatically digested to sugar. Then we use recombinant yeast to produce ethanol and the muconic acid. Other than the lignocellulosic biomass, the citrus peel is also a very special food byproduct. Okay, so its composition is different from lignocellulose because its main component or monomer sugar is actually galacturonic acid, not glucose. Of course, glucose is also one of them. So utilization of citrus peel to produce a fuel and chemical have some challenges because this galacturonic acid cannot be utilized by the yeast. The advantage of using citrus peel is it has very low lignin. Okay, so pre-treatment don't need to be so harsh. Okay, so you can see there's a citrus peel different from different fruits. It can contain this galacturonic acid at different composition. After that, they also contain glucose is still the major monomer sugar. Okay, and the xylose, arabinose, raminos, galactose, fructose, sucrose, so on and so forth, right? So you can see a lot of inline monomer sugar. So the process of using citrus peel to produce chemical is very similar to the lignocellulose. It's also to go through a pre-treatment method, then followed by enzymatic digestion to produce the monomer sugar, then followed by fermentation. If native yeast is used, they produce ethanol. If recombinant yeast is used, they can, in this case, the galacturonic acid can be utilized. Otherwise, native yeast cannot use this acid. And then the recombinant yeast can produce ethanol and other chemicals. Now I'm going to talk about the upcycling the food byproduct by food biotechnology. Similar, okay, so still we are going to use a microbes as the cell factory to convert such a food byproduct to food ingredient. For example, microbial protein, microbial lipids, and the microbial pigments. And also bioactive compound, the flavoring compound, soluble fiber, amino acid, vitamin. So through this bioconversion, the byproduct fiber is more digestibility is enhanced, and it also improves the nutritional value. So now we are talking about the vegetable and fruits byproduct upcycling. So fruit and vegetable byproduct has very high dietary fiber, rich in polysaccharides, sugars, proteins, enzymes, dietary fiber, fatty acids, flavors, aromas, and bioactive compounds. They normally contain very high amount of water. They have diverse compositions, and they are distributed in different locations. So biovalorization or the bioconversion of this such kind of byproduct can increase the bioactive compound content, enhance the antioxidant activity, generating new functional materials, produce microbial lipids, pigments, and improve nutritional value. So these are some examples. For example, so they use a different phyllum fungi, and the first one is a mushroom. Okay, so this fruit byproduct can be produced, used to produce phenolic compound, higher lipid recovery, and other phenolic compound, gluconic acid, bioactive compound, cinnamic acid, flavonoid, so on and so forth, and fruit flavor. So you can see, so the fruit byproduct can actually be used to produce some bioactive compound as a food ingredient. So one example is that the vegetable residues are fermented by LAB. LAB is lactic acid bacteria. So through such fermentation can produce LAB probiotics, because lactic acid bacteria are probiotics bacteria. And at the same time, it produces lactic acid, other organic acid, enhance the antioxidant activity, reduce pH, and lengthen the shelf life of the vegetable residue. And such a fermented product, this LAB can be used as indigenous LAB probiotic bacteria. And the produced lactic acid may be reintegrated in the food chain as an ingredient, and enhance protein digestibility and the sensory properties, the lactic acid, and could also be used as a food ingredient. So one more example is the citrus peel, citrus pomace, fermented by lactic acid bacteria, generating new functional materials So the lactic acid bacteria used is the Lactobacillus plantarum. Okay, it reduces the bitterness and umami taste and increase acidity. Fermented products contain both flavonoid and probiotics, and can serve as a new functional food ingredients. So you can see the power of fermentation. It can convert the vegetable or fruit byproduct to a food ingredient. One more example is to produce lipid. Okay, so there are two fungus were used, and this fungus present a higher ability to produce GLA-rich lipids. So it's lipids is produced, and the amount of beta carotene and the lutein increased. Another fungus, A. elegans, okay, increased significantly the total phenolic compound and flavonoid contents, and enhance the antioxidant activity measured by DPPH scavenging activity. Okay, so the food byproduct is the grape pomace. So that's why the food product can also be used to produce lipid at the same time enhance its biological activity. Now I'm going to talk about the biovalorization or upcycling of okara as through fermentation. So okara basically is a byproduct when you produce the soy milk and tofu. It's a byproduct. It contains about 50% fiber and 25% in protein and 10% in fat. It contains a lot of nutrients such as vitamin, amino acid, and trace element, and also phenolic compound. Okay, so for each kilogram of soybean used to produce the tofu and soy milk, about 1.1 to 1.2 kg of okara are formed. So okara consists of about 84%. It's very wet. It contains 80% of water, about 3 to 4% protein, and most of the insoluble soybean components. So this is an insoluble soybean. Yeah, so are in the okara. So the okara protein isolates contain all the essential amino acids. Actually, the quality of the okara protein is better than the protein in soy milk and tofu. Okay, that's why it has a lot of nutritional value. So upcycling of the okara can increase its value to food. So through fermentation of okara, it produces this bioactive compound and improve antioxidant activity, produce food for direct consumption. For example, can produce okara tempeh. It can be directly consumed, and degrade okara dietary fiber. Okay, so when you ferment, the dietary fiber amount is decreased, and fermentation also increased the free amino acids significantly because it's also digest this fiber, also hydrolyze the protein, and so you can use the okara to produce tempeh. You can also use it to produce a miso. This is one example that the okara is fermented with this Bacillus subtilis and another bacteria. The fermentation can degrade the anti-nutrition factors and enhance the nutritional value. And this fermented okara is used as animal feed.
Dr. Anil Jain, excuse me, you have 2 minutes left.
Okay.
Okay. Yeah.
So yes. So one more example is that through fermentation, the okara antioxidant activity is greatly enhanced. And so in Singapore, Prof. Liu Shaojian from National University and his student produced the okara beverage, okay, through fermentation. Okay, so this is a summary of upcycling of agriculture or food byproduct for food application. So through fermentation, the fermentation increased the phenolic compound content, enhanced digestibility, increased soluble fiber and protein content, increased functional properties such as antioxidant, anti-inflammatory, and immunomodulatory effects, enhance water holding capacity, making them more suitable in food formulation and potential in generating novel ingredients and novel food. And basically, during the fermentation, the microbes will degrade macromolecules to small molecules and degrade other chemicals too, so that the anti-nutrient factors are decreased and synthesize new proteins, synthesize primary and secondary metabolite, and the grow cell biomass such as single cell protein, lipid, and mycoprotein. So the challenges of using such byproducts such as okara, because they are scattered in different locations, transport is a problem, and vegetable, fruit, pomace, okara, brewers, spent grains, they are very contain very high amount of water, unstable, can be easily decomposed, inconsistent quality from batch, very wet. If you want to dry them, it is very energy consuming. It has, they all have very complicated composition and the reconcentrate structure because of the fiber. The microorganisms used to do fermentation is yeast, bacteria, and koji is a fermentous fungi. They are generally regarded as if they are food grade. So for the circular economy concept, during the food processing, about 30% of byproduct is produced. So through fermentation, it is possible to bring this byproduct back to the food chain. And so in the future, the byproduct can be digested using a microbial consortium concept to produce microbial protein, lipid, and pigments, or together with other nutrients. It is possible to use such kind of byproduct to produce alternative meat in the future. Okay, thank you. That's all for my presentation. Thank you very much for your attention.
Okay, thank you very much, Dr. Anil Jain. You have given a nice presentation regarding how to use the fermentation technology to convert the value-added products for the food byproducts, and how food byproduct waste can convert in the bioethanols productions and different beneficial enzyme, and use up lot of probiotics microbes, lactic acid bacteria and some other, and as a fermenting microbe as a yeast Saccharomyces cerevisiae to convert the lot of the byproduct of beneficial products. In these words, I just invite to some any questions, query regarding any question from the audience. So please ask the question one by one, I think. If I have any questions, please let me know. I think there is no questions. Dr. Neha. No, that's fine. My query is,
Yes, sir. We have mentioned if from also all the participants, they may not be able to unmute their mic due to the technical setup. So that's why we have posted that if there are any questions, then they can send us in the chat box, and then we can take it up, sir.
Okay, no worry. I think that is thank you very much, Dr. Anil Gar. And maybe just to proceed for next session.
Yes, sir, for the next three. Yes.
I think that next lecture is for mines.
Yes, sir.
Yes. Okay. Okay. You have already given my detailed biography. No need to. In this word, I just share to my slide and start presentations. I think this is visible.
Yes, sir. This is visible, sir. You can, if possible, sir. Yes, sir. Now it's on full screen mode.
This is the full screen, okay?
Yes, sir.
Okay. So just my topic is the how phytomicrobiomes helpful for enhancing nutritional quality and sustainable food productivity under the changing climate. This is the nowadays very big issue, climate change, other is nutritional deficiency and quality of food. Due to the use of lot of pesticides and green revolution and heavy use of chemical fertilizers and changing soil quality and fertility and health itself. So now this era new terminology come phytomicrobiome as a microbial consortia as a two approach in the culturable and non-culturable approach. In these words, I just move ahead further my presentation. There is behind the background regarding this presentation. First is global demand for agriculture productions in a sustainable way. This is the main things for that. Other is the major problem, green revolution and changing climate. So green revolution, everybody know how the when 1960 India has independent and start the population increasing, and then and in the globe as a use of green revolution to food productivity enhance, but ultimately we have added lot of chemical input in the soil, then we don't bother about and farmer don't bother about the microbial population. In another things, changing climate. Nowadays climate is changing, maybe weather is shifting, maybe weather and winter season and summer season and rainy season totally shifting, and we can't imagine in the sometime maybe rain again comes and moisture may be different. In these drought condition it's going sometime heavy rain in very short period, but those rain in the previously in the long time may be short and maybe 2-3 months slowly. So that is the maintain the moisture and quality of soil productivity. Other is environmental problem, pollutions due to others xenobiotic compounds used in the natural system from the anthropogenic activity. And then food and energy security is bigger problem, and nutritional deficiency. Nowadays nutritional deficiency disease increasing day by day. So this is the major problems. And farmers and others I have seen that conducted 4,000 farmer training and have visited in the villages and find that many people have the suffering from many nutritional deficiency disease like arthritis and anemia and so many nutritional deficits, and blood RBC formation is very less and multivitamin deficiency disease is increased. Why this problem comes? Because we are just taking chemical farming foods and nutritional deficiency, and new hybrid variety come, but those variety are not very short time they produce, but nutritional deficiency have recorded in that. So during this lot of problems, we just focus on the organic farming practices like biofertilizer, PGP, plant growth promoting microbes and plant growth promoting fungi. And new term comes in nowadays, phytomicrobiome. So this technology has huge and recently in worldwide very emerging technology, and I think this is new things we can minimize some problem of the soil microbial activity to enhance further. Other is biopesticide, farmyard manure, integrated nutrient management, crop nutrition, and these technology we can enhance the sustainable productivity out of food. So global pesticide consumptions, how you can see that China, India, Malaysia, Pakistan, Thailand, and Vietnam, every country has top 10 country. The huge China is most populated, India is second, and others Malaysia, Pakistan. How they increase the pesticide contamination as per FAO state data. So I think this is the great problem for our quality of food. Food demand is increasing. 1915 maybe 1.5 billion population, but today according to most recent estimated 7.3 billion, but in the future 9.7 billion by 2050. So this, if population is increasing, land becomes shrinking, how we can give the food? And in the sustainable food, nutritional deficiency is the major problem. This is major challenging nowadays. So this is the big problem, dietary problem and eating protein deficiency nowadays. And these things we expected to increase food productivity by 2050, 59% or 98%. This is very challenging task for that. Other is climate change scenario, how they impact on our system for that agriculture system. We can say that more drought and wildfires comes in that, The last year, in Australia has very big fire, and so many other country, USA have the big fire to lot of biodiversity problems for that. Warmer oceans, maybe in future temperature is rising, they create a lot of problem to city have go down in there. Habitat loss and shifting the range in the migrations and earth absorptions more energy. So this is the greenhouse gas trapped in environment and increase the temperature, and behind that moisture level is goes down and soil degradation is the problem. So this is the climate scenario, how impact on our agriculture system directly, indirectly. The other problem we can see that here, India, rice, wheat, maize, sorghum, there is a major problem and the negative impact of the climate change. By 2030, rice and wheat likely to see about 6.10% decrease in yield under the current changing climate scenario. Yes, ultimately, few crops and maybe some mix maybe positive impact on the gram, soybean, onion, but ultimately 1.5% loss in Indian GDP through agriculture. This is the very great loss in the climate change. So we need to overcome by the use of sustainable practices. For soil degradation is also one is a major problem to find out that the human activity have the hamper the soil structures and soil quality, use of the lot of the xenobiotic compounds for this. How you can see that word, I think not more clear. Water erosion is a major problem in the worldwide. In India itself also, lot of southern and northern part have the problem in that for that. Vegetation affected for that. Sustainable solutions. So this is the lot of problem, but nowadays sustainable solution we can use the beneficial plant microbes. They help the plant growth promotion, enhance the nutritional quality, and those microbes extracted from maybe culturable approach. If you just choose culturable and non-culturable, two type microbes. Till that maybe less than 1% is culturable, and only 1% that drastic change in biotechnology and food technology is there. Maybe future, if you increase the research is going on, increase the culturable microbe, they will be more beneficial for our society and helpful for that. How microbe is changing in the whole plant metabolism. All functional property of plant is governed by the beneficial microbes. So you can say that the harmful effect and maybe sometimes beneficial. If harmful effect, there is no microbe, or maybe non-beneficial microbe, they affect the plant and they degrade the plant health for that. So that maybe you can say the rhizosphere, endosphere, rhizoplane and rhizosphere, lot of microbial activity, maximum microbial activity take place here, and that those microbe goes colonize in the through root, in the stem and the leaf and other parts for the flower and food itself. Food is just, you can say the seed bank. Seed is the just a bank of the microbe and nutrient things. We have to generation to generations. If you grow one plant to particular climate, harsh climate, and those climate they have different microbial genera, they stored in the plant. If you go for another climate, maybe those plants activate the particular microbe to sustain plant under certain conditions. This is the impact of phytomicrobiome further. Others, this is the how harnessing phytomicrobiome as the cultivable and non-cultivated approach. Yes, this is the cultivated approach. We can just culture in the petri place and agar media, and we can just modify their genome study. For that, those microbe have different functional property. Other is for the plant assisted microbial breedings. If you just breed two microbe, one microbe have beneficial, maybe core group microbiome, another is those deficiency of the particular microbe. If traditionally made the traditionally transgenic plant, maybe breeding, those maybe exchange of microbe itself also, those support the beneficial. Other is selective screening. If you just screen microbe for the very healthy plant to convert the develop consortia for their seed and soil and plant foliar application to beneficial microbe. Other is non-cultivable approach, in situ microbiome. How in situ microbiome approach to change transgenic plant. If you just change in any agrobacterium mediated gene transformation, drought tolerant, insect tolerant, MAB, biofortified crop of mutant enriched. You at least we need to see that how our microbe changes. We study, we can enhance the by transgenic approach to beneficial microbe. Signolome approach, this is also if you lot up if any microbial anti insect resistant plants if you develop, if any pathogenic microbes affect the plant, they develop a volatile flavonoid compound and they give the signal. So we need to study also, that is the we can protect our plants. Other is hub microbiome. This is very important. These hub microbiome or core microbiome group, those are functional microbiome, functional group of microbiome which interlink to each other to govern the main property of when plant under stress, particular group of microbe is activated and they enhance the and support the plant health further. If microbe under the stress condition, salinity condition, drought tolerant, so some group of microbe is dominated, that is the and they affect the plant. Other is synthetic biology and the GMO as the microRNA technology. Now how to convert the microRNA technology to change the GMO crop and their functional property can enhance. So this paper has been published in Australian journals. So this other is the phytomicrobiome, all seed microbiome, shoot, leaf, and flower microbiome and rhizosphere microbiome. How these microbiomes study and isolated microbe we can use different the culturable approach, PGPR property, functional characterization, plant hormone production, phosphorylation, ammonia production, siderophore, SCN. Then we can develop synergistic antagonistic property among the microbe have the beneficial or not. Then we develop consortium. Nowadays consortium very not new, is older, but consortium is very worse if 4 microbe have different property mixed together. They have the give the cumulative property and enhance the plant productivity for that. So this is consortia, pesticide degrading, biostimulator, biofertilizer, biopesticide. They enhance the seed treatment, soil treatment, and foliar application in nowadays tea plant, others plants. This is the enhance the plant productivity. Other is if you go for metagenomic microbial identification for 16 years. Other way, how we just go for metagenomic analysis, far we can find how much microbial dominancy is taken place in the plant if certain condition. If you provide the stress condition, which type group of dominated, and study how much percent of microbial we go for whole DNA extractions from the plants and the parts and the for 16 years ITS, NGS sequences, time software, and develop OTU and the phylogenetic tree, and we found that how microbial analysis is found. So this is the plant, how you can just see the root microbiome, proteovector, echinobacter, ferment. This is a group of microbial texts are dominated, and seed microbiome very few, 4 genera. Either you can say the flower microbiome, some specific other group. The shoot microbiome is different micro, and the leaf. So how you can say the different part of the plant having different group of microbiome. Some common group is fine, but those plant, those microbes govern their functional property of the microbe. So this is our research. This is unpublished under a major revision in the Science of the Total Environment Journal. We have just studied the chickpea microbiome analysis. We have just developed some 2 bacteria, endophytic of Enterobacter harmoisi, and other is Brevandiomonas niaginensis BHU strain, other plus Mesorhizobium strain. So we have added in the T4, T10, T3 different combination. We have found that how microbial genera, if you inoculated 2 microbes, is a 3 microbe, 4 or 5 microbes in mixing together, we have find that they have enhanced the group of. In the soil, you can say in the control there is no activity such by that, where is only proteovector is dominated. But when we just inoculated with particular T4 introvectors with QPSP15 and 2 microbes, 3 microbes, we find the how enhancement. Other, so you can see our purpose of showing this graph the microbial exchange in the soil is more when inoculated, and they transmitted very few in the root, you can say, then they transmitted in the seed part. Soil is how can say the more diversity, and the seed is very specific for that. Then shoot is very limited. That means shoot is not residing. The maximum you can say the seed is the bank of microbes in that. This is a group of dominated microbes. Other how the functional properties. Suppose we just analysis for the chlorophyll and plant protein, seed protein, plant length, dry weight, and L. How you can see the different functional groups. Actinobacteria here you can say the significant for NPK analysis in the plant and the soil itself. How you can say the dominated proteobacteria here, plant length, chlorophyll, and plant carbohydrate. So this is the way to how seed microbiome. Just either we have just added the seed microbiome, and you can see the seed bacteria and say different genera is more dominating the different functional property and NPK for that. And this plant microbiome and human microbiome, they interchange each other, because when human take the things from environment, nutrient uptake and resistant pathogen, regular host immunity, we just take, you can see that. Others, sexual health, reproductive successes, and biotic test and pathogen. So each other, human are living in environment, and plant, we eat the plant and together. This is the functional similarity, you can say the human gut flora. If gut flora is healthy, similarly plant healthy, the plant gut flora, you can say the phytomicrobiome. So if you are taking the healthy food, you take every day banana and apples, and say the microbial flora and fauna or the gut is healthy, and human being is healthy. This is the similar gut flora and human plant microbiome is the beneficial for that. So some other study how just phytomicrobiome is the culturable approach to enhance the term of consortia. You can, if you isolate and develop the PGPR as the stain for the development, the use. How you can see the colonization of PGPR as the in the, you can say the Arabiasis thaliana, one plant, after 2 hours you can see the colonization much more. After 5 hours, how microbial dominated in the just adhering for that. You can say the make the biofilm part. Just if you use for the microbes, this is the very much effective for that. Other is culturable approach. This is the chickpea seed endophyte. They are isolated by my students and they develop. You can see this is the experiment for how they enhance the shoot, and this the root number of root biomass is increasing if increase the microbial different seed endophytes for that. This is the beneficial impact we can see there. Other is how fusarium inoculated. This is the biocontrolling agent. If you have the beneficial microbe, you can see the fusarium as the affected, not growing. If you add the fusarium plus biocontrolling agent, they also enhance the PGPR for that. This is the shoot length, branch length, and this is the research work done by my students. Another how nutritional content, because we have just mentioned nutritional quality, quantity is very important. If you add the microbial inoculant of the chickpea plant, you can see that protein in plant, if the part and fill, they have the T1, T0, T2. T2 and T3 have the inoculation. T3 is the consortia of the 2 microbe. T1 is the 1 microbial inoculant. How they increase, you can see the, so that means nutritional content in enhanced if we the increase the microbial consortia and inoculated for that. So that means the nutrient enhancements. Other is micronutrient in the seed chickpea, how calcium, potassium, zinc, magnesium, iron is also enhanced with the use of the biostimulant consortia for that. So this is the how these micronutrient, nutritional and plant growth activity enhanced due to the phytomicrobiome, culturable microbe, their property of direct, indirect. Direct, you can see the biological nitrogen fixation, nutrient solubilization, iron sequestrations, and plant hormone productions. So this is the so many, you can say that this is the microbes have the many variety. I think time is very less. We have only 2 minutes, then we'll finish. You can say the different functional property or the phosphorus logging, you can say the laboratory step, and then you see now This is the other microbe. This is the PGPR activity, how just find that they enhance the property of the plant growth promoting microbes. They enhance the IA production phosphohistology. They screen on the basis of that we have published this paper. They enhance nutritional content in chickpea farm. Other siderophore production, microbe have the ability to produce siderophore. They have iron chelating to provide the iron deficiency in that. Other is indirect mode of actions for antibiotic production, chitinase productions, and induce systematic resistance and SCN. They directly, indirectly help to product the plant protection by the different activity for that. You can see here antagonistic effect of Rhizoctonia sololi and different bacteria have the, how just you can say that they have the biocontrolling agent, they inhibit the growth of that. So the other we have published these papers and you can find that Pseudomonas aeruginosa inhibit the growth of Fusarium for that. This other trichoderma is one potential biocontrolling agent used for that. You can see how if a biocontrolling agent they degrading the mycelium of the pathogenic microbes for Rhizoctonia sololi. This is the other Beauveria bassiana. They have the insecticide fungi. They great potential biocontrolling agents for the used for the insecticide fungi. Other different types of biofertilizer, how they carrier based liquid and solid. And constants are there may be biofertilizer used like up standard packings and maybe market novelty, no microbes lack of quality for that. This is very problem, and maybe sometime microbial count is less and maybe constant of strain is not more efficient. So this is the you need to indigenous strain is the more effective, and like of standard packing some maybe time if increase the efficiency maybe. Other is the yes field level. This is very important. Native microbial population is more better if you are using the inoculation technique. Inoculation practices should be better for enhancing their production. In all laboratory we have used for our students for that. Yes, treatment may be seed treatment, seedling, fruit, foliar, and flower treatment. So multiple type application used, then maybe more effective of the phytomicro and PGPR property. This is some publication I am not going. In our laboratory we have developed some major Rhizobium Pseudomonas fluorescens. This treatment combination give the approximate T24. This combination give the 40% chickpea production in the our field trial we have just got by that. This is the potential consortia for that. You can say that 29%. And how you can say the modulation for the research and development needs to set up appropriate guideline for quality aspects, market structures, enhance the shelf life and certification, and futures perspectives and changes of organic farming. Yeah, maybe phytomicrobiome for need biofortified rich organic fertilizers, maybe huge and develop indigenous strain that is more suitable for different crop and agroclimatic conditions. Other may be characterization of biochemical and molecular signal. That's a very good effect for that. This how organic farming of phytomicrobiome is the alternative to save the environments, and they affect our society, promote the biofertilizer and microbial consortia, use the mixed cultures for the enhancing the our society as the healthy for that and fertility. I think thank you very much for sorry to take the more times. Okay, Dr. Neha, I think we need to start from next lectures. Yeah, any questions? Audience, please, you have any question? Please put in the chat box, and maybe you can email me direct so I can reply. I think yes, Neha.
Yes, yes, sir. We can move on to the third session. Third lecture for today's session.
Okay.
Dr. Deepak Mudgal has joined, sir. You may please invite him, sir.
Okay, okay, yes. Dr. Deepak Mudgal is here.
Yes, sir. Good afternoon, sir.
Okay, 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 sir. I just read your short biography. Sir, Dr. Deepak Mudgal, sir, is presently working as a senior assistant professor in Department of Dairy and Food Technology Mansin Bhai Institute of Dairy and Food Technology, Mehsan, Gujarat. He earned his master degree and PhD degree in food technology and Department of Food Technology, Guru Jameswar University of Science Technology, Hisar, Haryana. He has 13 years of teaching and research experience. He has published around 60 papers in national and international journals, and he has published 8 books and 13 book chapters and filed 2 patents. And he has completed 20 research projects funded by external funding agency and in-house research projects. He has also developed several food products for commercial interest and value-added products. He has been ranked amongst top 2% scientists of the world in 2020, and he has received several awards at the national and international level. Sir, is this short biography? I must congratulations to you, have the 2% as a scientist in the list. Great,
Thank you so much, sir.
congratulations, sir. And instead, I invite to you for your presentations. Over to you, sir.
Thank you so much, sir. Let me share my presentation.
Yeah, sir. Yeah.
Is it visible?
Yes, sir. Fine.
Okay, firstly, I would like to thank the organizers for inviting me for the talk on partially hydrolyzed guar gum as a sustainable source of dietary fiber in International Conference on Food Sustainability, Challenges and Opportunity for the Future, 5th AMIFOST, organized by MIT Institute of Food Technology, MIT University, Noida. So thank you very much. I'm starting my presentation. So basically, I am talking about the partially hydrolyzed guar gum, which is a very novel and a rich source of dietary fiber. And dietary fiber in today's diet is very important for us. So before going to partially hydrolyzed guar gum, so we should know about the guar gum, the base material from which the partially hydrolyzed guar gum can be prepared. So first, guar. Basically, guar is a cluster bean, and its botanical name is Amopsis tetragonolobus, and it is mainly cultivated in India, Pakistan, Brazil, Australia, South Africa, and USA. And India is the world leader in the production of guar gum. And India and Pakistan both collaboratively produced around 90% of the world production of guar. And within India, the major producer states are Rajasthan, Gujarat, Haryana, and Punjab. Now the guar plant, as I told you, its botanical name is Amopsis tetragonolobus, and its seed, like the other seeds, is composed of 3 layers: husk, germ, and endosperm. And the main portion of interest is the endosperm, which constitute about 35 to 40% of the seed portion, kernel portion. And this is in the right side, the pictorial diagram of this seed components: germ, endosperm, and hull. So basically, the guar gum, the galactomannan, it is extracted from the endosperm portion of the guar seed. So now, what is guar gum? As we know, it is extracted from the endosperm portion. It is composed of galactose and mannose. That is why we are calling it galactomannan. And mannose and galactose are present in 2:1 ratio. The main backbone chain is of mannose unit. The mannose units are composed, are linked with beta-1,4 linkages. And on this backbone chain, galactose units are attached as side chain with the alpha-1,6 linkages. And this galactomannan is a very high molecular weight polysaccharide, and it is not digested by our intestinal secretions. That way, it can be used as a dietary fiber source. Now, properties of guar gum: it is off-white in color, then it is somewhat odorless, and it has a bland taste, not a specific type of taste. And the good property is its solubility and viscosity. It is water soluble in nature, and it gives a very high viscosity solution, very high viscous solution when dissolved in water at a very low concentration. And apart from that, its solubility, it is not only soluble in hot water, it is also soluble in cold water. That's why it opens the various scope for its application in food products where the temperature is not required. So it is cold water soluble, and it makes it a unique source of dietary fiber. And it forms very strong hydrogen bonding with water. That's why it increases the viscosity of the aqueous phase system, and it can be used as a thickening agent, emulsifying agent, and stabilizing agent. And it also has a film forming property, which can be used for the edible coating purposes of fruits and vegetables to extend the shelf life of fruits and vegetables. And we know that every food product has a main ingredient, we can say, main component as water. And this guargum controls the rheology of that product by aqueous phase management. And the viscosity of this guargum solution is influenced by temperature and pH. Now, how we can prepare guargum on the industrial scale is the process starts with the seed, and the guar seed is split into two halves, and with having husk, and after that it goes for the thermal dehusking operation, and the guar split is produced. Meanwhile, during the thermal dehusking, the germ portion is also separated from the endosperm portion, and now we have clean guar splits, and these splits are milled into the guargum powder. So basically, on industrial scale, it is available as a guargum powder in powder form, solid state. This is the manufacturing process flow sheet for the guargum preparation. So the operation starts with the sieving of the guar splits. Then the blower is there for expelling out the impurities from that, and then it goes for the to the mixture for the hydration of these guar splits, and these hydrated guar splits goes to the dryer to maintain the moisture content, so that it can go to the flaker mill where the flakes are formed, and then it goes to the ultrafine grinder for the ultrafine grinding of the guargum. And the powder fines are collected in the cyclone, and then the rest of the powder goes to the sifting section where the sieving is done, and on the basis of particle size, it is mixed or blended into certain proportion to prepare a specific or desired final product, guargum powder. So this is the pictorial picture of the guargum powder making, where we can see this the raw material, the intermediate products, and the final product. So first one is the cluster bean, having the seeds of the guar. Then these are the guar seeds. Then this is how the guar splits look like. And finally, this is the powder. Now the industrial applications of the guargum are food industries, textile industry, pharmaceutical industry, cosmetics industry, paper industry, explosive industry, and oil well drilling. So guargum has a vast scope in industrial and as well as food applications because of its providing viscosity to the aqueous phase water system. And the main application is its stabilizing property, emulsifying properties, and water thickening property. So in food industry, it is used in ice cream, ketchup, sauce, noodles, pasta, bakery industry, bakery products, dairy products, beverages, fruits and vegetables. So in every product, apart from providing the stabilizing action, thickening action, or emulsifying action, it also provides certain specific properties. Like in ice cream, it provides it prevents the formation of ice crystals. In ketchup and sauce, it prevents the phase separation from the aqueous and the serum phase separation. Likewise, in bakery products, noodles and pasta, it also provides machinability to the dough for the easy processing into the products. Dairy products, it can be used for increasing the dietary fiber content and also for its stabilizing action. And in beverages, it can be used, and in fruits and vegetables, like I said earlier, it can be used for enhancing the shelf life of fruits and vegetables, and it can be applied in the form of edible coating with other polymers like proteins, etc. So this was all about the guargum, which is the base material for the preparation of partially hydrolyzed guargum. So now partially hydrolyzed guargum, in this presentation, I will also call it PHGG. So why PHG is required, or why we are preparing partially hydrolyzed guar gum from the guar gum? Because guar gum is a good source of dietary fiber, and it is a soluble type of dietary fiber because of its water solubility. But it is restricted for its use in food products beyond a certain concentration because of its high viscosity, because it provides around 1% solution of the guar gum provides around 6,000 to 7,000 centipoises viscosity, which is very high, highly viscous solution. So we cannot use this product for partially dietary fiber fortification in food products as such. So that is why we have to go for the hydrolysis, or we can say the partial hydrolysis of this guar gum to prepare a product which can be suitable for fortification purposes in food products. So this high viscosity of guar gum also decreases the protein efficacy and the lipid utilization, and it interferes the digestion and absorption of the nutrients, which is a negative part. So these are the reasons why we have to prepare partially hydrolyzed guar gum from guar gum. So now, what is dietary fiber? As we know, these are the plant materials mainly derived from the plant cell wall portion, and they are resistant to the digestion by human gastrointestinal secretion or enzymes, we can say. So there are two types of dietary fiber: soluble dietary fiber and insoluble dietary fiber. In soluble dietary fiber, the main components are pectin, gum, and mucilages. Insoluble dietary fiber, lignin, cellulose, and hemicellulose are the main components. And if we talk about the sources of these fiber, the sources of insoluble fibers are wheat bran, whole grains, and vegetables, we can say. And sources of soluble fiber are oat bran, barley, beans, psyllium, and some fruits also. So now this table is showing the comparison between the soluble and insoluble fiber on the basis of mechanical and metabolic effect. So we can see here the soluble fiber are more important as compared to the insoluble fiber, because they are providing more metabolic effects as compared to insoluble fiber, like delay in gastric emptying, increased fecal bulk, regulate colonic transit time. Means we can say it is improving the digestive health of the person who is consuming it. And it is also beneficial in diabetes, like slow glucose absorption from the small intestine and reduce postprandial blood glucose level during the clinical test. And it is also beneficial in heart-related problems because it lowers the serum total cholesterol and low density lipoprotein cholesterol. So now, how we can prepare partially hydrolyzed guar gum from the guar gum? So there are several methods by which we can prepare this partially hydrolyzed guar gum, like enzymatic hydrolysis, acid hydrolysis, irradiation, microwave, ultrasonic techniques. So, but enzymatic hydrolysis is the preferred one in the food industry, because it is not using any chemicals, or it is not having any side effects related to the final product. So first, enzymatic hydrolysis, it is the preferred one. And in this, many enzymes like mannase, pectinase, cellulase, and pullulinase, it can be used for the enzymatic hydrolysis of the native guar gum to produce partially hydrolyzed guar gum. So how this partial hydrolysis occurs? These enzymes act on the linkages between the mannose backbone chain, and it shortens the backbone chain and reduces the molecular weight of the polymer, so that this lower molecular weight leads to the lower viscosity of the product. And this low viscosity product we can use as such for dietary fiber fortification in the products. This guar galactomannan hydrolysis via mannase enzyme is the most popular one among all the enzymes. And mannase treated guar galactomannan has reduced molecular weight and viscosity. And another thing is, the main functional structure of this native guar does not change during this process, enzymatic hydrolysis. Means the galactose to mannose ratio, which is around 1.8 to 1 or 2:1, which remains same in the hydrolyzed product as well as in the native product. So basic change which occurs during this enzymatic hydrolysis is change in the molecular weight and the viscosity. So, but there are certain parameters, process parameters we have to optimize or we have to control during this enzymatic hydrolysis, like temperature, pH, reaction time, and the enzyme concentration. Based on that, it will decide the properties of the final or resultant hydrolyzed product. And during enzymatic hydrolysis, guargum is not fully hydrolyzed. That is why it is known as partially hydrolyzed guargum. Another method is acid hydrolysis. So controlled acid hydrolysis can also be used. And the main acid used here is HCl, and it can be used as room temperature as well as in elevated temperature like 70 degrees Celsius. And it also reduces the molecular weight of the resultant product. And one more thing where it is necessary to neutralize the acid used in the process, so that it can be used for the further food application, because most of the product applications are at food applications are at neutral pH. So we have to neutralize it. Then the thermal hydrolysis. Hydrothermal process can also be used like at 200 degrees Celsius temperature or at a reaction time of 7 minutes. We can produce oligosaccharide or PAGG, we can say, having a degree of polymerization of 20. So here pH is also very important in this hydrothermal process. It will affect the viscosity of the final product. The next process is irradiation. Irradiation hydrolysis. Irradiation dose can also be used for the hydrolysis of guargum, and the mechanism of hydrolysis here is the random fracture of the polymer chain. So with increasing dose level, intrinsic viscosity of the product decreases, means the viscosity of the product decreases with the increase in the dose level, and it is beneficial, and we can prepare this partially hydrolyzed guargum. And it also changes the rheological property of the product, final product, from initial native guargum, which is non-Newtonian in nature, means it is having a shear thinning like behavior, to the Newtonian fluid. Partially hydrolyzed guargum behaves like a Newtonian fluid, means its viscosity does not change with the shear rate. Then irradiation can be done in the powder form or in the solution form. So we have both the options using cobalt-60 gamma irradiator. And one study declared that the initial moisture content of the guargum affects the final hydrolysis of this product during the irradiation process. So initial moisture content control should be there. And in this process also, there is no considerable chemical functional group modification, which is confirmed by the FTIR spectroscopy study. So there is no functional group modification. The GM ratio also same in this process. And irradiation of guar leads to reduction in its apparent viscosity and consistency index, like in other processes. Next method is microwave hydrolysis. So microwave hydrolysis can also be used, and in this catalyst potassium persulfate is used, and it is more effective in the solid substrate as compared to the aqueous solution. So here we don't have to increase the moisture content of the product, or we don't have to prepare the solution of guargum. So guargum powder is mixed with the water containing potassium persulfate at a very low concentration of the water, and kneaded well in the form of dough consistency, and then it go for the grinding, microwave treatment, washing and drying, and thus the final product can be prepared. And this resultant prepared product, we can refer it as a PAGG because of its low molecular weight as compared to the native guargum. And similarly, it has also less intrinsic viscosity, and microwave hydrolysis is more effective as compared to the thermal hydrolysis treatment, like hydrothermal treatment. The next hydrolysis method is ultrasound hydrolysis. Ultrasonic waves can also be used, and using this high intensity probe type ultrasonic processor, it can be used alone or in combination with other technologies like enzyme hydrolysis of the microwave hydrolysis. And in combination with the in microwave hydrolysis, it is more effective for the production of PAGG, and it also shows synergy with the enzymatic method, like using cellulase, we can produce PAGG with a superior quality. Now, after this production preparation methods of partially hydrolyzed guar gum, we should know about the properties, physicochemical properties of the partially hydrolyzed guar gum. So the main properties are related to the viscosity, different type of viscosity like intrinsic viscosity, apparent viscosity, viscosity stability, shear viscosity, molecular weight, and then flow behavior index and consistency index, crystallinity index, solubility, and tensile strength of the films prepared from the PHEG. So now the intrinsic viscosity, it is the characteristic of an isolated individual macromolecule, and it depends on the shape and the specific volume of the individual molecule of the polymer, we can say. So intrinsic viscosity can be measured by the capillary viscometry, and the range is around 2.7 to 0.28. And in the preparation of PHEG, all the viscosity like apparent viscosity, which is generally considered as a resistant to flow of the liquid, and it is measured at a single speed using a viscometer. So it is very low, 4 CPS for 1% aqueous solution of partially hydrolyzed guar gum, which is very much low as compared to the native guar gum solution, where the 1% solution of native guar gum produces around 6,000 centipoises viscosity solution. So viscosity stability is also increased as compared to the native guar gum here. So effect of temperature is not so much. It can be stable at a very high temperature of 130 degrees Celsius, and the viscosity stability is there for a wider range of pH like 3 to 7, means it covers the acidic range of the pH. Then salt concentration, it is also stable at a very high salt concentration up to 10%, and digestive enzymes like amylases cannot digest this product, and the viscosity of the product also not changes. So that is why it can be used as a source of dietary fiber for the fortification of dietary fiber in the products, food products. So shear viscosity, as I told you, the enzymatic hydrolysis or the another type of hydrolysis for the production of partially hydrolyzed guar gum, it changes the rheological behavior of the product from non-Newtonian to Newtonian, means from shear thinning behavior to the Newtonian behavior, where the viscosity do not changes with the shear. Now the molecular weight of native guar gum and partially hydrolyzed guar gum. Native guar gum molecular weight is around 9 lakh Dalton, and of partially hydrolyzed guar gum it is around 8,000 Dalton. And the reported studies shows that from it can have a molecular weight of around 5,000 to 20,000 Daltons, which can be effectively used as a dietary fiber source for clinical trials. The flow behavior index and the consistency index. So flowability of the product increases in case of partially hydrolyzed guar gum. It is high as compared to the native guar gum. Consistency decreases with the partial hydrolysis in case of PHEG. The guar gum, native guar gum is providing more consistency to the product. And crystallinity index is also reported, which shows the crystallinity of the product, means the molecular alignment in the particle structure of the substance. So using X-ray diffraction study, XRD studies, so the crystallinity index also reported. Partially hydrolyzed guar gum has a significantly high crystallinity as compared to native guar gum, 13.2%. Now the solubility, as I told you earlier, PHEG is water soluble, and it is insoluble in organic solvent, and it is soluble in both cold and hot water like native guar gum. And pH has no significant effect on the solubility of the PHEG. So good solubility in water and very low viscosity means the viscosity of 1 to 2% of partially hydrolyzed guar gum solution is equivalent to just like the simple water, we can say. We can compare the viscosity. So same viscosity without increasing, we can say the zero viscosity also here. So it can be used as a unique dietary fiber source for the beverages like liquid products, where the viscosity is a very much important parameter. Tensile strength of the film is also important, and it improves the tensile strength of the film for fruits and vegetable application for edible coating purposes. Then the food applications of partially hydrolyzed guar. I think we have less time, so it can be
Yes,
used
yes.
for application
You
of
can, you 2-3 minutes, no worry.
yes. For food application, it can be used for bakery products, dairy products, beverages, and noodles and pasta type products. So we can see here, partially hydrolyzed guar can be used for the dietary fiber fortification of bread, and it also improves, not only improve the dietary fiber content of the product, but also improves the functional characteristics of the product, like firmness and low volume in bread, and hardness decreases in cookies. So softer cookies can be prepared using this, but a little bit overall acceptability of the product reduces. Then in yogurt, it can be used, and it improves the physicochemical as well as the textural property of the yogurt. So we can improve the texture of the yogurt using this, and dietary fiber also, which is a active ingredient. So in beverages, where the viscosity is very much important, the consumer do not want a viscous beverage. So that is why PAGG can be used, because it has a very low viscosity and up to 4 to 6% level in the beverages. And in certain beverages, it can be used up to 10%, like lassi-like beverages. So in buttermilk, it enhances the viscosity at acceptable level, and it also reduces the whey separation, which is a defect in the product. So it prevents the whey separation in the buttermilk, so which is also important. Then in noodles, it can also be used and improves the textural characteristics of the noodles. Then there are several health benefits. As we all know, the dietary fiber have several health benefits, like in cardiovascular diseases, diabetes, irritable bowel syndrome related to digestive system. Then it is also having prebiotic effects. Prebiotics are the food for the bacteria, and it improves the health of that bacteria. So it has also prebiotic action because of its low molecular weight. And the laxation effect is also there, appetite control and weight management. So this is a some sort of studies have been published related to this appetite control and weight management, but more work can be done in this aspect for obesity control and weight management using this partially hydrolyzed guar. So these are the health benefits. So I think I can conclude here. Partially hydrolyzed guar is a water soluble dietary fiber, and it is prepared from the native guar, in which the India is at #1 in production. And we can also increase at government level. We can also recommend or suggest that its production can be increased for this preparation of partially hydrolyzed guar. And it has low molecular weight and viscosity, and it is a very sustainable source of dietary fiber. And in Asian countries, including India, there is an environment which is friendly for the cultivation of this guar plant. So we can recommend it, and we can produce more guar, and that guar can be converted into partially hydrolyzed guar. And it contains, the native guar contains 80% of the galactomannan. Means no other food product or cereals can produce or can give that much high content of the dietary fiber. Means a single product can give 80% of the dietary fiber, soluble type of dietary fiber. So, and having low viscosity, water soluble nature, and tasteless. It is tasteless. It do not provide any taste to the product. So it do not interfere with the taste of the product, and it is colorless, odorless behavior is very important. Then it increases the dietary fiber content, and it also provides all the health benefits which are associated with the dietary fiber, like diabetes and cardiovascular disease prevention, prebiotic effects, satiety control, and digestive problems. So thank you very much. That was all from my side. And if any queries are there.
Yes, if any query, audience, please raise hand or maybe write the questions. So I think no questions. Okay, thank you very much, Dr. Deepak, for nice presentation and detail. I think you have done a lot of research regarding on this topic, and given the how partially hydrolyzed guar gum has a beneficial impact for the multiple aspects. You can say the multi-dimensional aspect for food, textile, pharmaceuticals, and health benefits, and what is the how we can produce at the industrial level. This is a great lecture. Give the detail
Thank you,
about
sir.
guar gum production. Just my small query have how we can produce, what is the cost benefits of that? How suppose we can generate 1 kg or something, from how much cost may involve for production from grain or seed?
At industrial scale, we can produce. Hello,
Yeah.
am I
Yeah,
audible,
yeah.
sir?
Yes, yes, sir. You are audible. Yeah, you are audible. Just my query is that how much cost may be involved for productions of that, suppose 1 kg, 2 kg.
Per kg of the product, partially hydrolyzed guar gum can be produced at the rate of 200 rupees, sir. 200 rupees per kg.
Oh, that is very cheap, I think.
Yes, it is a very cheap and a very rich source of dietary fibers.
Yeah, yeah, that is very beneficial for that, and I think there is a multiple application in food and medical and medicines, and this is a great finding, I think. Okay, thank you very
Yes.
much for your nice lecture.
Thank you so much, sir.
Yeah, welcome.
Yeah. So, Dr. Jayaprakash,
Yeah.
since there are no more questions in the chat box, so sir,
Yes,
can we proceed with the second part of the session, that is the 4 oral presentations that we have?
yes. Okay,
So,
yeah.
so I would like to thank all our 3 eminent speakers for their talks and highlighting the different, byproduct, like starting with Dr. Enli Geng. She highlighted on how food bioproducts through fermentation and with the use of industrial and food biotechnology, it can be used for the upcycling of agro-industrial products for further use in food applications. She threw light on bio-valorization also. Then Professor Jayaprakash, he told us about the role of phytomicrobes, both the cultivable and non-cultivable approaches, and their role in in sustainable food production practices. Then I would also like to say that we before this we haven't thought of guar gum, that is the
Yeah.
hydrolyzed guar gum, can be used in such a broad manner that that modification, just to partially hydrolyzing guar gum, can make it as a such a better as a better food additive, and can be used for as a sustainable source of not only as a dietary fiber, but from other aspects of food additives also. So I thank all the speakers. I once again request the participants, if meanwhile we are having the oral presentation, if they have any queries, then they can post us in the chat box till we are carrying out the oral presentation. So now I would like to call upon our first oral presentation. It is by Mr. M.I. Hasni from Amity University, Rajasthan. Is Mr. M.I. Hasni with us?
Yes, ma'am, I'm here.
Here. And
Yeah.
it's a request to all the presenters who are there in the oral presentation that we are already lacking behind in time. So I request you to please wind up your presentation in 4 to 5 minutes, because from 3 p.m. onwards in the same meeting, the next technical session has to start. And I once again regret the inconvenience caused to all the participants and our eminent speakers. As our first inaugural session, it was a bit late. It ran behind a bit late. Yeah. So I request my husband to please give his presentation.
Yes, please start. Yeah.
Good afternoon, everyone. I am Muhammad Imam Hassani. I'm currently doing Masters of Food Technology at Amit University, Jaipur. Today, my topic of plant-based protein, opportunities and challenges. So to about the topic, plant-based protein is basically a protein that are derived from plant sources.
I think network issue, yeah.
Am I audible?
Yes, yes, you are audible.
So nowadays, consumers are shifting from traditional animal-based protein toward plant-based protein for a number of reasons. We'll see them in upcoming slides. So the main sources of plant-based proteins are soy, pea, wheat, potato, and rice. Now, the driving forces are nothing but the reason why consumers are shifting from traditional animal-based protein to plant-based protein. We have global warming. We all know how much the meat processing industry are causing harm to our environment. And for that reason, most of consumers are shifting toward plant-based protein. Animal welfare, the wellness and suffering of animal cannot be ignored. We have human health concerns. Here, it has been proved that processed and red meat is associated with several chronic diseases like cancer, type 2 diabetes, and so on. With religious concerns, so in a country like our India, where majority of people are Hindu and vegetarian, it's a good opportunity for the population to consume plant-based protein. So, and also, as I said, the vegetarian diet is having lack of protein. So it is also an immense opportunity for the vegetarian population, products like plant-based proteins. Now this is the market of plant-based product by ingredient pea, soy, wheat, and so on. Am I audible?
Yes, yes, you are audible.
Yeah. We have a number of types like beef, chicken, pork, fish, and lamb. And we can find several kind of products in the market like burgers, strips, meatballs, and many others. Now, processing of plant-based protein. There are several plant-based protein products, but today our main concern is meat, plant-based meat. So the common technique used for plant-based meat is thermal extrusion. As we can see on the slide here, this is the processing principle of thermal extrusion. The extruder has different zones from a feeding zone to fiber stabilization. And at first point, the extruder is fed with a mixture of powders. It can be either a mixture of soy or wheat or rice. And then the process goes on. Water is added. The process goes on till through the process, the temperature is increased because the product will undergo some molecular changes, and for that we need higher temperature. At the end of the process, we cool the mixture using the cooling diet, and as a result, we have muscle-like chunks. And further processes are added, like the addition of vegetable oils, antioxidants, and some other additives imitate the meat-like texture and colorant. So now a question arises, which meat is healthier? There is no straightforward answer for this because as we can see on the slide, in every side we have pros and cons, but one thing is sure, plant-based meat has a low amount of cholesterol and fats, and that is a good thing for human health. Now, what are the opportunities of plant-based meat, plant-based products? Efficiency of food production. We all know that plant-based sources do not need food in order to produce, and we have affordable nutrition in comparison to animal-based meat. 1 kg of plant-based meat is less costly than the previous one. Improved health benefits, as I already stated, lower risk of contamination. Well, plant-based protein products have low moisture content, and for that reason, it decreases the risk of contamination. Reduce deforestation. We all know that in comparison to animal-based meat production, plant-based meat use less land, and for that, there is less deforestation. Now, moving to the challenges, the biggest challenge of plant-based meat texture and flavor, because for that, manufacturers use several additives such as preservatives, stabilizer, and colorants. So here, one question arises, are these products really healthy? Because consumers nowadays prefer food less processed with less the use of all these additives. It becomes a hard challenge for the plant-based protein industry.
Mr. Husni, you have another 1 minute to wind up. You have already exceeded your time. You are all the
Okay,
presentations are to be supposed in 5 minutes. It's already 8 minutes.
Okay. We
Yeah,
have
so-
3 more challenges, such as presence of anti-nutritional factors and regulatory challenges. Now, to conclude my presentation, plant-based protein products are gaining interest because of several driving forces I've already mentioned, and additional research is needed in order to address the challenges. And also, finally, some innovation in crop processing technologies will help to create a sustainable global plant protein supply. So that was all from my side. Thank you.
Any questions from the panelists? Jayaprakash sir, Mudgal sir? Any
Yeah,
questions
that's
from?
fine. I think Mr. Hassan, you have just mentioned the nice presentation, plant-based protein. Just my query is there, and suppose plant-based protein, how much alternative of plant-based protein from the you may be consider in plant-based protein in the algae itself, maybe fungi itself also, yeah, maybe some other plant.
Sir, could you repeat your question?
Just in the plant-based protein, you have just mentioned only plant-based, yeah, maybe fungi itself also consider in that. Fungi, you know the.
Yes, sir. Yes.
Yeah, because fungi is the major protein source, rich single cell proteins, you know.
Yes, sir. There are some other options like cell-based proteins. They are also available in the market.
Yeah. So this is the how much difference of that maybe cost difference, yeah, maybe because the plant-based protein maybe take the time and the lot of processing for their purification. Yeah, I think so. Then as compared to maybe single cell protein.
Yes, sir. But for the comparison, I don't know much about cell-based protein, but yeah, what I know is that they are available in the market.
Yeah. Okay. No worry. Yeah.
So thank you, Mr. Hasni. So now I would like to call upon Mr. M. Bhalerao from RTM Nagpur University.
Yeah, yes, ma'am.
Yeah. So, Madhura, you will be having the next 4 to 5 minutes to sum up your
Yeah, sure.
presentation instead of 8 to 10. Kindly stick to the time.
Yes, definitely, ma'am. I hope my screen is visible and I'm audible.
Yes, yes, please.
Yeah, thank you. So my complete presentation is based on the review work on the modification and isolation of non-traditional oils and why we are focusing more on non-traditional oil. So moving very first to the background that we have day-to-day usable commodities, and these products are available with the raw materials which also have the petroleum crude fractions into it, whether it will be a derivative or a direct hydrocarbon may be used. So with time we move towards the other alternative, that was the vegetable oil. But with time vegetable oil also has increased in the consumption, that is the human consumption, because we are completely dependent on such edible oils which we use with the fractions in food industries and other non-food industries as well, such as soybean, sunflower, groundnut oil, and so on. Non-edible oil are also present, that is the castor oil. But yes, more majorly this vegetable oil has to this meet the demand of our consumption, human consumption, and also the industrial need as well. So the other alternative to this is the non-traditional oil seeds. So these traditional oil seeds are basically the one, these are selected basically for their unique fatty acid profile. This fatty acid profile can be modified and then used into the food products as required, because on modification they'll give different type of functionalities. Further, the modification processes may vary on the end product use, and the extraction and isolation of active compounds and the bioactive compounds present in these types of non-traditional oils. So this is the novelty which we can get from the non-traditional oil seeds. This is the current status of our import of the oil from other countries in India. Even though we are producing our own production, still we are importing for the edible and non-edible purposes and industrial purposes as well. This is the chart which gives basically the area of the land which we have and the production which we are achieving. This is shared because the collection of non-traditional oil seeds is naturally available in India, but going towards the forestry department having the permissions always is not possible. So we still have the area of production with us, and agricultural new farming technology can be involved into it. This will create employment and as well increase the production of non-traditional oilseed oils as well. Being having the area of production as well and the import, we still have the export data as well with us, because till 2017 we were not exporting any kind of crude oil, that is basically the edible oil. But after 2017, India government has given the permission for the local producers as well. So this is the reason very first that we should have a very indigenous technology with us, so that we should refine, modify, and technology which will give the direct final product into our market, because we are exporting our crude oil and we are importing the finished product. So this is a very vast difference in the costing, that export of a crude oil is very cheap, and import of the final products, that is non-traditional oil final products, whether it is a refined oil or a final product of a food or any cosmetic industry or any non-food industry. So the other non-traditional oils which here will be considered as the kusum.
Ms.
Pisa.
Madhura,
Yeah.
Sorry to disturb. You have 1 more minute to wind up.
Yeah. Okay. Thank you. So these are all the mentioned non-traditional oils. So here the modifications can be done as the hydrogenations, fortification, blending. These are the main modifications which could be done. The literature till now is that these oils are already used into the cosmetics industry and food industry as in present, but this is affecting the shelf life and the quality of the product due to presence of other ingredients in the oil seeds. So this is the potential pathway which can be achieved to have the specified modification and the final product. These are the variables that is raw materials and market study will be done. For the formulation of end product, the process pathways will be differing on the basis of raw materials as well and the modification process. So the methodology basically will include the isolation of bioactive, modification of fatty acids, and fatty acids with the emulsification power, that is the HLB ratios, will be achieved. So this will be the deliverables at the end of this research, if it is being done, that this particular bioactive compounds could be used in the direct final products into food industries, and also the blending and fortification processes will help the cardiovascular and diabetic patient mainly present in India. Also, the specialty chemical products could be modified product development from these non-traditional oils. So these are all the references. Thank you.
Sir, any quick question you would like to ask, or can we move to next presentation?
Yeah, that's fine. We can move next part.
Okay. So next, I would like to call upon Ms. Shalini Kumari and Jyoti Kumar. Ms. Shalini Kumari, are they here?
Yes, ma'am, available.
Yes, kindly share your screen. Yeah, please start with your presentation. We have 4 minutes with us for your presentation, so kindly stick to the timeline, please.
Okay, ma'am.
Yeah, instead of it would be better if you can give us the crux of the presentation, because the timings have been reduced to half almost.
Okay, ma'am.
So
Ma'am,
yeah.
it's a part of my research work. The topic
Okay.
is the mineral composition of the some neglected wild edibles plants mainly consumed by the Munda tribe of Jharkhand, India. It's wild edibles are those that are neither cultivated nor domesticated, but they are naturally available in their natural habitat, and they are used as a food source, but they are just neglected. They are not utilized. But research has showed that they contain huge amount of minerals and other nutrients, and they are compared with the these are the summary of some plants which I collected. These are the pictures of the plants. They are dried in the shed dried, and their sample collection procedure is like they are just sorted out and dried in the shed, and then crushed and powdered form are made. And they are their mineral composition is analyzed with the help of atomic absorption spectroscopy by using the method of AOAC 19th edition. And their mineral composition is calculated according to their standard by comparing their standards. These are the results. This result showed these wild edibles are more nutritious, contain more amount of macro and micro minerals as compared to the commonly cultivated spinach and amaranthus. These are the results of macro minerals like nitrogen, potassium, calcium, sodium, magnesium, phosphorus, and chloride. And the plant like Commelina bengalensis and showed high amount of, comparable to the 4 plants, show high amount of nutrients. And likewise, micronutrient contents, I showed iron, magnesium, zinc, and copper. Same, they are compared with the commonly cultivated plants like spinach and amaranthus. The results showed that they are more or less same. Some plants contain huge amount of minerals as compared to the spinach and amaranthus. And these are the some comparison with other wild edibles of other state or other countries like Nigeria, Pakistan, and Thailand. People work there, and their other tribes, people working there, other tribes, their other wild edibles comparisons are there. So more or less, these plants are highly nutritious. So these are the micro minerals content comparisons of Assam, Nigeria, and other states. So my conclusion about this, due to shortage of fertile land, rise in food prices, high rate of population growth, and huge difference between demand and supply of food, which leads to hunger and malnutrition, these wild edibles can erase the gap and favorably be used for the most of the commonly used vegetables. So commercialization of these wild edibles may create income for the poor peoples, as they are the rich source of protein minerals. So more systematic studies required for this so that we can explore it industrially. That's my research work for now. And these are some pictures for my work. These are some references. Thank you. Thank you.
Thank you, Ms. Shalini. Sir, any questions from your side?
That's fine. Salini, just my small query have you have presented research. Thank you very much for your nice. You have presented your research. Just my query: have you studied about the some secondary metabolite, a bioactive compound?
Yes, sir. Yes, sir. It's a part of my research works. Next, it's about the secondary metabolites, phytochemicals,
Yeah.
and about subproteins or proximate composition. I have covered all parts.
Yeah, so I think so if you check for antimicrobial or biopesticide property of the secondary metabolite of these plants, so I think this is a better application for future. You can go for patent itself also, because so many wild plants have till date not much more
Yes,
study
these
about
plants
the
are not
metabolite.
much explored, sir. In
Yeah,
it's
yeah.
my fourth plant, that is alternantheria. It's first time I'm exploring this plant, sir. Nobody has
Okay.
worked earlier about.
So thank you for your
sir.
presentation.
Thank you, Shalini.
Okay.
Now, can we have our last oral presentation for today? Ms. Usha Sharma and Salim Siddiqui from Sharda University.
Yes, ma'am.
Yep. Ms. Usha, will you please share your slides? And please stick to the timeline of 4 minutes.
A very good afternoon to all. I am Usha Sharma, PhD scholar at Sharda University, Greater Noida. So this is my presentation topic, seaweed, a sustainable food source in the food industry.
Just
So
make your
basically seaweed,
presentation is the full mode.
yes sir.
Just make your presentation presentation mode, full mode. That means
full screen mode,
full,
please, Usha.
full screen mode. Yeah.
yes ma'am, this is in a full screen mode, ma'am.
On our screens, actually, what sir is saying, we are able, it's not on full screen mode.
Ma'am,
Only
but
you have-
it is, but ma'am, it is.
Okay, fine.
Okay, no
You
worry.
no, don't
Shall I
worry,
start
please.
now, ma'am?
Yeah, and please come to the
Shall I
main
start now?
crux of your topic.
Yeah, yes. You go ahead.
Yeah, that.
Okay, sure, ma'am. Thank you. So basically seaweeds, as we know that nowadays seaweeds gaining a momentum place in food processing industry because of the increasing population, shrinking fertile land, and more demand of the nutritious and vegetable like plant-based nutrition product. So as we know that there are worldwide 221 species are there which are explored out, and out of which 145 species are as used for the food consumption, and 110 species for a phycocolite productions. And they are very high rich in the protein, mineral, fatty acids, dietary fiber, and all bioactive
Oh, something problem.
Usha, are you there, still there? Your screen has gone.
Yes. Some network issue, yeah, maybe somebody is there.
Yes, sir. Maybe some network issue.
Yeah, I think so. Yeah.
Yeah. And the large scale cultivation of seaweed is practiced in Tamil Nadu, and after that Gujarat, Maharashtra, Andhra Pradesh, Kerala, Karnataka, and Odisha. And but in India, the lack of information and lack of some advanced technology for the production of extraction of the seaweed, and poor training facilities and underdeveloped marketing facilities, and this growth
Usha,
of
your presentation is not visible. Your screen is not visible to us.
now it is visible, ma'am.
Yes, now it's
Yes.
visible.
Okay, ma'am. So because of the information and other parameters, because of that, and the sector of the seaweed cultivation is hindered in India. And if we see the growth of the seaweed in market, so as you see here the growth chart, and it is increasing widely from 2017, and it is estimated that it will grow further 24.992 billion in 2028. As you see here, the seaweeds delivered as a fat-free, gluten-free, mineral-rich, low sugar and low calorie as a healthier substitute. Now, it also possesses some biological properties photoprotective, neuroprotective, anti-carcinogenic, antiviral, anti-obesity, prevention from the osteoporosis, and anti-inflammatory activity. Like as we know that there is Alzheimer is a neurological disease. That one is a common, which is protected by the seaweed. There is a lot of evidence is there, and on the chronological disease, like there is a lot of disease like HIV and other cancer and Alzheimer, so they can be prevented from the seaweed extract. And if we see, the seaweed are classified into majorly three groups like chlorophyta, rhodophyta and phaeophyta. And chlorophyta is green in color, and rhodophyta red, and phaeophyta is brown in color. So it contain sulfated polysaccharides like alvan, carrageenan, agar and fucoidan. They are possess a very biological and functional properties in the food industry, like carrageenan, which possess like antioxidant, antimicrobial, anti-inflammatory activities, and also as a lipid barrier properties. It is very functional, and it is shows a functional property in food processing industry, like it give a gelling, binding, thickening, emulsion and protein stabilizing, and also used in like making in the beverage industry also. And like the agar, and it is considered as a vegetarian gelatin, and it is basically excellent gelling properties and thickening, and they are also like provide the laxative properties to the food product. And fucoidan, which possess a very good
Usha,
role
you have
in the
only one more minute
pharmaceutical.
to go on. You have
Okay,
only
ma'am.
one more minute. So please
Okay.
conclude accordingly.
Okay, ma'am, sure. So these are the methods which are generally used for the cultivation of seaweeds: long line method, net bed culture, bamboo raft, and there are the few industries which which cultivate the seaweed in India, like marine chemical coaching and S&P natural and alginate products, and these all are the like industries which produce seaweeds in India. So there are the few of products are there which are has been developed from the seaweed, like there is a we can laminaria, and laminaria are used in 2.2% to make a soft cheese, and other one is a sagrism, mangitarium, which is a Indian brown seaweed, which is used to like make a pasta. So these all are the like pictures are here, and which is like used to develop the seaweed product. And there are the there are the some products which are like show in the supermarket. So seaweeds, and other is the application, like it is like basically used as a biopolymer and a phycocolloids, and biopolymers like basically phycoidan and carrageenan and alvan, which are widely used in the as a formation of a biocomposite, nano food packaging film, and phycocolloids like thickening and gelling agent, and also very widely used in the production of jellies and fruit candies. And also they act as a gas barrier or a fresh cut fruit packaging. And other use like laminaria, that laminaria species are used in the increasing the shelf life of the product, like
Your time is up, Usha. Your time is up.
okay, ma'am.
Yeah.
Thank you, ma'am. This
Thank
is
you.
only the future scope.
Oh, thank you, Usha. Sir, would you like to ask any questions from her?
Yeah, that's fine. That's fine. Okay.
Okay.
The time is less.
So, okay. Thank you, sir. So, thank you, Usha. Sir, after this, can we conclude the session?
Yeah. Yeah, sure.
Ms. Bandi, over to you. Ms. Bandi, Dr. Bandip, you are on mute.
Yeah, sorry. So now on behalf of MIT Institute of Food Technology, I would like to extend vote of thanks for this technical session 1. First of all, I thank Professor Jayaprakash Verma for chairing this session. Thank you so much, sir. I thank our distinguished invited speakers, Professor Enli Gange and Dr. Deepak Modgil, for sharing their valuable thoughts. And also I would like to thank all the participants and the audience for taking time to be here. Thank you so much.
Okay,
So-
welcome. Thank you very much.
Thank you, sir. And with this, we request the team for technical session 2 to take over. From Amity
Okay,
Institute
thank
of
you.
Food Technology, Dr. Loveleen Sharma and Dr. Alok Saxena, please.
Thank you.
Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Good afternoon, everyone. I am Dr. Lovely. Welcome you all in AMIFOST 2022. We will now proceed with technical session 2, and I would like to invite Dr. Alok Saxena, who is a co-chairperson of this session, to give us a brief introduction of the theme. Over to you, sir.
Thank you, Dr. Levine. So in this technical session 2, the theme of the session is technologies for food safety and quality analysis. So I invite our chairman of the session, Dr. Miguel Angelo Prito Lage from University of Vigo. Just to introduce him, he has done twice master's degree in the food science and technology, also in biosystem engineering from the University of Vigo and University College Dublin, respectively. He's doctorate in food science technology at the Institute of Marine Research from IIM CSIC Vigo, Spain. And currently he is a Raman Y. Casal researcher at the Spanish Council for Scientific Research. And in his credit, he has published more than 145 peer-reviewed articles, all in SCI journals, with more than 4,000 citations, and he is having an H index of 37. So I invite him to take over the session. Dr. Miguel.
Thank you, Dr. Alok. Thank you for the nice introduction. Please, first of all, I want to thank you, the organizer committee, for the invitation to chair this session, for participating in the international meeting. As Dr. Alok said, I will be chairing the session 2, Technologies for the Food Safety and Quality Analysis. In this session, we have 3 invited speakers, Professor Xingyu Lin, myself, Miguel Angel Prieto Lage, and Professor Dr. Brendan from United States. I think, and afterwards we will have 4 oral presenters. If you agree, Dr. Alok, I will introduce the first presenter, which will be Professor Xingyu Lin.
Yes, sir, go ahead.
Hopefully, Professor Xingyu Lin, I'm pronouncing your name right. Professor Xingyu Lin.
Right. Yeah.
Thank you.
Thank you. Thank you.
From the Biosystem Engineering and Food Science Zhejiang University in China. He has received a bachelor's degree in chemistry in Xiamen University, and he got a PhD degree in the Department of Chemistry in Zhejiang University, China. He has working in Caltech, California, as a postdoc researcher from 2016 to 2019. He's currently a professor in biosystem engineering in Zhejiang University. He has published more than 20 SI papers, including biomaterials, green chemistry, biosystems, analytical chemistry, and other more. And his group centers the development of innovative nanotechnology and nanofluidic chips from rapid detection of harder compounds and foodborne pathogens in food. I think I leave the word to Professor Xingyu Lin, and he can introduce much better his work. Thank you.
Okay, thank you, Professor. Yes. Maybe I need to share my screen. Okay. Okay, is that okay?
Yeah, perfect.
Yeah, okay. Thank you for inviting. My name is Xingyu Lin from Zhejiang University in China, and today I will make a presentation about the rapid detection in the food. Yeah. First, I will introduce myself first because I'm new here, and sorry, I need to. I'm Xingyu Lin, and I come from Zhejiang University, and at 2016 I graduated from Caltech, and now I go back to the Zhejiang University to study the. My major is in the analytical chemistry, so I have some new analytical chemistry method in to detect the hazard material in the food. So now I will start my presentations. Okay, what's this? Yeah. My presentation may be divided into two parts. The first one is the chemical detector of the chemical compound, and the second is the detector of the foodborne pathogens. As all know, the foodborne disease can be caused by the foodborne many hazard material in the food can cause the illness, and many people dies with food diseases. And there are many toxins, and there are many hazard material in the food, like the bacteria, virus, toxins, heavy ions, pesticides, and antibiotics, and so on. So it's very important for us to develop a rapid detection method that can, like the sensors that we can use to detect the hazard material in the food directly without any preparations, sample preparations. But however, the food sample is very complex. They contain many substances like the lipid, proteins, and any other things that will interference or inhibit the sensors. So our goal is to develop a sensor that can be used to detect the target molecules in the food directly without using any sample pre-treatment. Like we use a sensor immersed in the milk, and we can detect the pesticides and antibiotics in the milk. So we learn from nature. In the cell membrane, we know that there are many biological nanochannels in the cell membrane, and these nanochannels have very high selectivity and high throughput, and also they have excellent anti-fouling and anti-interference properties. That means they can function very well in the complex matrices like the cell matrices, and they will not their selectivities and their functions will not be destroyed in the complex samples. So if we can put these nanochannels in our sensors, maybe it will help us to detect the substance in the milk matrices. However, these nanobiological channels, although powerful, they are very unstable in the outside. So we thought if we can develop the artificial nanochannels, which can help us to develop powerful sensors. The preparation of the artificial nanochannel is also very simple. Just put the substrate like the electrode in the solution for self-assemble. The CTAB surfactants will make micelles and absorb onto the substrate, and the silicas will deposit in between these micelles, and then we can got fumes. After we dissolve the surfactant with the ethanols, we can go to a nanochannel membranes, and the membrane can be peeled off from substrate. And then finally, we can get a membranes, a film with nanochannels on it. So this is the top view, TM top view, and the pore size is just 2 nanometers. And this is a cross-sectional view, and we found the channel is actually not a nanopore, it's actually a channels throughout the membranes. So using this method, we can get a similar mimic artificial nanochannels. And the channel have very high selectivities, like toward the molecular with different size and with different charge. And also we found the molecular with different hydrophobicities can, for example, the hydrophobic molecular can transport while the hydrophilic molecular cannot. So at last, we also found that the molecular transport in the nanochannel is very fast and very fast, which is beyond our expectations. So next, we want to see how we use it in the food science, and we put these nanochannels on the surface of the electrode sensors, and these sensors can put directly in the complex real samples like milk, juices, carrot juices, and soils. And if we immerse this sensor into the sample directly, and the target signals like the OPS, pesticides, or the antibiotics or TNTs can be detected by our sensors without any sample pre-treatment. This is because the large molecules like proteins and lipid will not transport through the nanochannel since it's very ultra small, just 2 nanometers in diameter. And only the small one and the small hydrophobic and no charge, the small one can transport through the nanochannels and to the electrode, and we can get the sensors. So the red line is the signal we got use our developed sensor, while the green one and the blue one we didn't see any obvious peaks here. So that means our sensor can be work very well in the complex real samples. Okay. And also we also use this modified the nanochannels to detect the caffeine in the coffees, and also the heavy ions in the fruits. Okay, the next due the time limit, so I will introduce quickly. And my next is the detection of the foodborne pathogens in the food using our nano sensors. There are many targets we need to detect, like the virus, bacteria, and the parasites. They all will contribute to a very foodborne disease. We use a digital PCR method, which was very powerful for quantifications. The method is very simple, like just first to partition the sample into thousands of droplets, and each droplet may contain one target pathogens or nucleic acid. If the droplets contain one target molecules, it will amplify and got a fluorescence dot. And if it was didn't contain the target molecules, it will be a negative reaction. So we will not find it's a fluorescence. So finally, by counting how many positive droplets in the sample, we can know how many like bacteria, how many viruses in our samples. So it was an absolute quantification method, and no standard curve needed to be done in the rapid detection. So here we developed a hydrogel-based digital lamp method, which can amplify the DNA in situ, and the amplified DNA can form a very a fluorescence dot. And finally, we can count how many dots in the hydrogel. We can know how many bacteria, how many viruses in our samples. So it's something like the culture, but the time is much shorter than the bacterial culture. It only takes less than 10 minutes. We can get the result. And we investigate the details, the functions of the DNA amplification in the hydrogel, because it was under a nano confinement, different with that in the bulk solutions. We found when DNA was amplified in the nanoporous materials, the sensitivity was increased, and the reaction rate is accelerated, and also no false positive was found in the reaction, and the efficiency was also enhanced. Okay. And what's interesting me most is that our method can use for the real sample directly without any DNA extractions and also anything else. You can see that if we add milk into the DNA amplification, like the PCR systems, the PCR will be inhibited. But if we added it into our hydrogels, no inhibition was found. So we can use our sensors to detect the pathogen like the E. coli and the salmonella in the key milks. And also we detect them in the fruit, and we can got accurate numbers. With the more and more E. coli in the sample, we found more and more fluorescence dots in the hydrogels. And finally, now because of the COVID-19 virus, we also detect the COVID-19 virus in the food, because there are many transmission of the virus by the food cold chain food logistics, and the virus can be spread in the food from one place to another place. So we wanted to detect the COVID-19 in the food, like the fresh fruit, and with the method is also very simple and can detect the like this one dot correspond refer to one virus in the samples. Okay, this is another we will maybe if I can miss this, and we also developed a similar method that can be put directly. The whole experiment can be conducted on a piece of membrane, because the membrane has many nanopore or micropores, and the digital PCR or digital lamp can be performed on the membranes. Okay, with this is the positive fluorescence, positive reaction, and this is negative reactions. We can count how many positive pores on this membrane. We can know how many bacteria or virus in the samples. Oh, this is the membrane we prepared. Yeah. So using this membrane, we can do the filtration and the amplification and the purification together without any sample pre-treatment. Okay, last, and my group is focused on the nano confinement or nanofluidic that can be used to directly detect the hazard material in the food. And we focus on the nano confinement because it can give us a very interesting phenomenon. Like we didn't need to purify or purify the food samples, or need to do any other pre-treatment. So just put our sensor into the samples so we can get the results. So that's what I want to focus in the few years. And at last, I thank to the invitation from the universities and also this conference, and also thanks to the National Foundation of China. Okay, thank you, because the time limited. Thank you very much. Yeah.
Thank you, Professor Xingyu Lin. Just very nice presentation. So I open the questions to everyone, if anyone has any question. So I have a question, Professor. Like I see that your work is based in chemical detection and pathogens detection. Just this is a more general question than do you think that your work can be introduced at the industrial level? Could you think that we could see this in a few years? Yeah.
There are any other voice coming in? Yeah. Hello?
Yeah. So what do you think, Professor Xingyu Li? We will be able to see this technology introduced at industrial level in the future?
Yeah, yeah. You mean the outside of the lab, right?
Yeah.
Yes, it's we think it's more suitable for commercialized, because there are many companies who contact me, like the one in the Singapore and any other countries. Also, they are care about using this to detect the some hazard in the food. Those they think to the collaboration to, if I think it's a very method that didn't exist in the paper or just in our lab. I think it's ready to commercialize. So my next stage will to put it, to forward it to in the commercialization, that we can just we can finally get a sensor that can we can go outside and to detect the anything we want in the market or in the supermarket or in the farm in the farmers. Yes, that's all that I want. Yeah, thank you.
Thank you very much. Thank you. Nice presentation and the questions you have answered. The next presentation will be by myself, but I'm passing the word to my colleague, Maria Carpena, Dr. Maria Carpena, who is a postdoc in the University of Vigo. She holds more than 45 manuscripts published in the last 5 years, and she will be presenting the synergy and antagonisms, the events behind the antioxidant agents used in food industry.
Good morning. My name is Maria Carpena. I currently work in the University of Vigo with Professor Miguel Angel. Thank you very much for the introduction. Today I'll be presenting the work entitled Synergy and Antagonism, the events behind antioxidant agents used in food industry, which has been the collaboration of other institutions like the Polytechnic Institute of Braganza and other partners in Italy, China, and Argentina. First of all, I would like to introduce our research group, which is directed by the Professor Miguel Angel, and also with the collaboration of Professor Jesus Simal Gandara and Professor Jan Bosiao. We also work with people from Portugal, the Polytechnic Institute of Braganza and the Polytechnic Institute of Porto, and together with the Research Institute of Science and Technology of Materials in Argentina and the Department of Sustainable Food Processes in the Università Cattolica del Santo Cuore in Italy. Our research lines are main focus on the chromatographic separations and extraction technologies, the characterization of secondary metabolites and bioactive compounds in food and other biological samples. And we also focus in the study of biological properties of enriched extracts for the development of new natural food products enriched with bioactive compounds, the valorization of food byproducts, and we keep a focus also in the bioinformatics and chemical engineering, focus on the response of biological properties of bioactive compounds. I would like to introduce this work talking about the compounds diversity. In nature, we can find a very diverse agent. We can have antioxidant activity. We can separate them in pure compounds, but also we can find complex extracts of natural products. The problem with this situation is that for testing the antioxidant activity, we can apply different quantification methods, and results are commonly treated with different calculation procedures. So sometimes it's really difficult to use this information to establish comparisons between the analysis and get to solid conclusions. So some of the main obstacles that we can find is the intrinsic factors, which depend on the oxidation mechanism and the surrounding environment of the antioxidant reactions. But we also have accidental factors like the temperature, pH, the solvents, or in this case, which is our main focus, the appearance of synergistic or antagonistic effects. So nowadays, different food additives or chemicals are applied to food products because of their individual benefits. But what happens if we combine two or more of these agents? This is where we can find concepts like synergy and antagonism, which have been frequently used to describe these relationships. Basically, we can define synergy as the interaction of two or more substances that produce a combined effect greater than the sum of the separate effects. The antagonism, where the interaction is lower than the sum of their effects, and the additivity, which is focused on the interaction producing a combined effect that is exactly the sum of their separate effects. As you can imagine, what we would expect for the greater results is to find synergy between two or more compounds. However, perform all these assays would be time consuming, and sometimes it's not the desirable option, but we can apply simple combinations to test and expect and predict which are the expected results. So at last, I will only like to introduce in summary the antioxidant assays that we used to test the response in this study, which were the DPPH, the ABTS, beta-carotene, and protein, which are all of them colorimetric assays based on the chemical reactions that you can find displayed on the screen. So what we basically did in this study was to take 7 compounds that they are commonly used as antioxidant agents that you can find displayed on the left part of the screen in blue, which are butyl hydroxynisole, propyl galate, alpha-tocopherol, etoxyquine, ascorbic acid, third butyl hydroquinone, and trollops, and analyze the resulting interactions when they are applied with other antioxidants commonly used in the food industry, like trimethylquinoline, butyl hydroxytoluene, and the combination of these last two. In this case, what we did was to test the individual time dose effects of the individual compounds, just adding 50 microliters of the compound and 2050 and 50 microliters of the reagent of each specific antioxidant reaction. And to test the synergistic and antagonist effects of binary mixtures, we use half of the volume of each antioxidant agent and the other half of these ones placed in orange, or the combination of both of them. Then the absorbance was measured at every minute during 300 minutes with agitation at 660 cycles per minute, and samples extracts were analyzed kinetically for 8 different concentrations by serial dilution. So the assays were developed in microplates, where we combined 8 times 8 arrays of 2 antioxidant mixtures at equal increasing concentrations. Then the independent action and the concentration addition, and with these two, the quantification of the interactive effects, were modeled according to the equations that you can find depicted on the screen. And the 64 concentration combinations values were used for the identification and quantification of the interactive effects between the different antioxidant agents. Moreover, we use response surface methodology to estimate the coefficients of the selected parameters, such as it was the concentration of radical reduced, or the mass of the antioxidant, or the average molecules of the reduced radical between, depending on the antioxidant. So briefly, getting into the results that we obtained, in this slide, what you can see is the individual effects for each one of the 7 antioxidant agents, also for the 3 that we used to combine with, and for the 4 antioxidant assays. In each graph, each dot corresponds to the experimental point that we measure at the laboratory, and the line corresponds to the model that we propose for modeling and for the expected results. And you can see that, for instance, in this case, for the propyl galate or the etoxyquine, we find complete different responses between the both of them. Then what we did was to study the combination response to test the synergistic and antagonist effect of binary mixtures. So in this case, we select the DPPH just as an example, and we can find that the left column, which is highlighted in orange, corresponds to the joint response of the antioxidant, while the right column refers to the difference between the respective null interaction and the obtained response. So you can see that in some of them, the interaction is negative, corresponding to an antagonist effect, while in other cases is positive, corresponding to a synergistic, and it can vary depending on the doses that you are applying of each compound. At last, what we saw here is the matrix combination of responses between the antioxidant agents that they use for combined and the two of them. So for each one of the 4 antioxidant assays, we can find in the diagonal the results obtained for the control. In the top part, we can find the surface response for each pair of antioxidant combination. And in the bottom diagonal part, again, the difference between the respective null interaction and the response showing this null interaction or positive interaction corresponding to synergy. Just to end here, we have the test reproducibility of the assay, where we can see in the left part the raw responses of, in this case, BHA and ACE, corresponding to the combination of antioxidants. And on the right part, we can see how the values are distributed on the reproducibility and how accurate is the model. So to conclude, we could just say that synergy and antagonism are controversial characteristic behaviors of very diverse systems. The results that we find here and the models that we proposed were directly expandable to other types of assays. Responses for null synergy and antagonism effects were found when characterizing the interactions between several pairs of individual and complex mixtures of chemical antioxidant agents, and interactive effects between antioxidants were introduced into the model with auxiliary functions that describe the variations induced by each antioxidant in the parameters that define the effects of the other. So at last, we would like to thank all the institutions that were collaborating in this work, and also to our research group. And thank you all for being here today listening. And I will be pleased to answer any question that you have, and so will be Professor Miguel Ángel. Thank you.
Thank you, Dr. Maria Carpena. Any question from the audience?
Yes, Maria, I would like to ask if I noticed that you use both natural and synthetic antioxidants. Do you notice any difference between the behavior of both types? And I would like to know if there is any plans to a future application in the industry of this finding. Thank you.
अंदर के साइड में वो रेड टाइप में कल मैंने देखा वहाँ पे
Turn off the microphone. It's very difficult to hear. Yes, thank you. Dr. Aurora, please, can you repeat your question?
I noticed that in that study, you use both natural and synthetic antioxidants. Do you find any difference in the behavior of the both kinds of antioxidants? And I would like to know also if there is any plan to future applications of these findings in the industry. Thank you.
Thank you. Dr. Maria?
Oh, thank you, Aurora. In this case, we find different responses for synthetic and natural antioxidants because of this wide compounds diversity that I was talking at first, which is different between pure compounds and complex mixtures where these effects of synergy and antagonism are more pronounced. And for the application questions, the idea here is just a preliminary say, but it would be to better know the consequences of putting all different of food additives together in a food product, and just think that as much as you put in, it would be better, but in sometimes these effects are not like this. So it could be to a better comprehension of how the addition of food additives is directly corresponding to the effect of each individual one.
Thank you, Maria.
Thank you. Thank you, Dr. Aurora. Any other questions from the audience? Okay, I think we can move to the next part of the session, which will be the next invited speaker, Dr. Brendan Niemira. Are you here, Dr. Brenda?
Yes, I am.
Okay, Doctor, I just give just nice to meet you. Just a quick introduction about Dr. Brendan Niemira. Is the research leader of Food Safety and Intervention Technologies Research Unit at the U.S. Department of Agriculture in the Windamore, Pennsylvania. He leads a team of more than 20 scientists, engineers, technicians, postdocs, and students developing tools to kill human pathogens like E. coli, Salmonella, Listeria, Shigella, norovirus, etc., on produce of meats, poultry, eggs, and other foods. His personal research focuses on the development and validation of cold plasma and other non-thermal treatments. Dr. Niemira has published more than 150 peer-reviewed research articles, book chapters, and critical review. He serves on the board of directors for the Institute of Food Technologies and is a member of the editorial boards of Food, Applied and Environmental Microbiology and Journal of Food Protection. Please welcome all of us, Dr. Niemira.
Thank you very, very much, Dr. Laca. And I would like to thank the organizers for inviting me to present to you today on what is a very interesting technology that I hope you'll find useful in inactivating different kinds of pathogens on fruits and vegetables, as well as other kinds of foods. So what I'll be talking about is specifically viruses, using cold plasma as a way to inactivate viruses on different kinds of foods and food contact surfaces. So let me make sure that there we go. Okay, so cold plasma is a relatively new technology. I would say, a couple of years ago, I might have described it as an unfamiliar technology, but it is becoming more widely known in food processing circles. So it's adapted from existing applications. Cold plasma is used in materials processing and in textile manufacturing and in other kinds of non-food applications. But in recent years, in the last, say 5-10 years, there's been a great surgence in technology application for food and food contact surfaces, fruits and vegetables, meats, seafoods, powders, flowables, seeds, and other kinds of foods. Cold plasma is a very attractive and interesting technology because the inputs to the system are relatively limited. You only need electricity and some kind of a gas to turn into a plasma. And so those of you perhaps not familiar, plasma is essentially an ionized gas. And so you've got a complex mixture of ionization products, high energy species, and plasma chemistry that happens in the plasma plume that is very broadly effective against a wide range of target organisms. And the output from this ionization process is a highly reactive, self-quenching plasma. So in addition to all the regular reaction products, free electrons, monatomic oxygen, free protons, and so on, all of these things ultimately will then recombine with themselves if they don't hit a target organism. That's what's called self-quenching. These will resolve to making more familiar chemical products such as peroxides, nitric and nitrous acids and oxides and other kinds of products, ultimately resolving to very little chemical residues. So there's no chlorine in the system. There's not necessarily any water in the system. So there's a lot of advantage from the standpoint of sustainability and energy efficiency in how the process works. Plasma can be very effective. It's very flexible. There's different ways to make plasma depending on the design of the electrodes. And I'll go into that somewhat as I go forward and present the information about viral inactivation. But some of the things that are very important in how plasma will affect the food and how effective it's going to be in inactivating the organisms is how the plasma is created, how it's delivered, the shape of the target food that you're treating, as well as the container that it's on and the circumstances under which you're applying the plasma. Some real key factors here are the plasma flow rate, the contact time, as well as surface effects. If you look at the diagrams on the left showing that the plasma applied to blueberries in a jar, if that's a system where all of the plasma is captured and it's entirely delivered to your target organism, you get all of the benefits of all of the plasma, but you can also capture some thermal energy and you get some temperature rise. If, on the other hand, you're applying it in an open system as applied to, say, a conveyor belt or a food contact surface, you don't have that kind of rise in heat, but you can sometimes have a diminution or a dilution of the chemical species of the plasma. So that can make a difference. In the middle, you see the plasma, same plasma system applied to a melon or to an egg. So the kind of product that you're treating can have a real difference. The netted surface of the melon means that when that plasma plume is contacting the surface, those surface features cause a lot of microturbulence at the surface of the melon, and it causes a greater contact time of the plasma plume with the surface, so you get better inactivation. An egg, however, is smooth, so you can see kind of from the image, as the plume is hitting it, you get a laminar flow of the plume around the egg, so you get less contact time, and you need a greater overall treatment time to get the same kind of inactivation. Looking at the diagrams on the other side, you see if you're generating plasma inside a container to treat seeds, as you are in this example, then you need to adjust your plasma creation factors, voltage and pulse frequency and so on, so that you don't burn through the container and cause damage to the plastic. You maintain that packaging integrity, but you can still get full, complete treatment of everything that's inside that package. Looking at the almonds that are being treated in the same system, you can see that the plasma is wrapping around the almonds. You get complete coverage of each one of these individual food products. So whether you're treating a continuous flow type system of lettuce on a conveyor belt or individual almonds or things inside a package, depending on how you're designing the system can really influence what kind of ultimate inactivation levels you're going to get. The way that plasma inactivates viruses as well as bacteria and parasites is because of all of the different kinds of chemical reactivity that's created in a plasma. And what actually will do the killing for you is going to depend on how you design your system and how you have your food or your target organism, your target substrate inside the system. Where you have your product inside the plasma field, entirely surrounded by the densest possible form of plasma, as you see in A, the direct form, you get everything. You get all the electromagnetic field pulse density, you get electrons, you get heavy ions, you get radical species, you get UV radiation, you get some of the thermal effects, so you get the intense visible light. A lot of advantages there, but the downside is you got a very small space between those electrodes. So you're kind of limited in the kind of products that can go through that sort of a treatment system between, as you see here, a dielectric barrier discharge. Powders, small seeds, small fruits might be appropriate. Larger products such as apples, oranges, whole steaks, whole shrimp might not be so appropriate. If you widen the gap between those electrodes, you get more flexibility in the kinds of foods that you can put through the system, but you start to lose some of the most reactive kinds of plasma. So it's a bit of a trade-off in terms of the intensity of the plasma treatment versus its flexibility for how it's applied. So you start to lose some of the UV radiation and some of the electromagnetic field density, but again, you gain some of that, some of the flexibility. Going even farther down that road, if you start to go to an indirect treatment system, whereas instead of having the food between the electrodes where they're exposed to the fully dense plasma, if you create the plasma with the electrodes and then deliver it to the food, you're losing some of the most reactive products in the plasma plume and the plasma chemistry, but you're gaining a lot of flexibility in the types of foods and food contact surfaces that can be treated. And then ultimately, if you take that even one step further and introduce the plasma into a tank of water or put mist through the plasma system, so you're creating plasma activated water, plasma activated mist, you're using that water to capture the plasma reaction products and then deliver that newly created sanitizing solution to the food. You are losing most of the reactivity and most of the action that is happening in plasma chemistry, but you're gaining a lot of flexibility because now what you've created is a sanitizing solution just from air, water, and electricity. And sanitizing solutions, of course, are used very widely in the food industry, and so we're very familiar with how to use those. So that is very readily slotted into industrial processing. From a efficacy standpoint, as you can see, there's a very broad range of efficacy. The modes of action can be very complicated and very broadly applicable to lots of different kinds of pathogens, viruses, bacteria, parasites, spore formers, spores, insect pests, and so on. And here's just a couple of examples of the kinds of electrode systems that you can use to create plasma. These can be ring electrode, collinear electrodes, concentric, widely spaced dielectric barrier discharges, and other kinds of things. You'll see these as I go forward. I don't have time to go into all the details, the technical details of all these systems, but if you have any questions, I'm certainly happy to take them at the end of the talk. So talking specifically about viruses, the term virus, of course, it's a broadly applicable term. There's a lot of different kinds of viruses, many different sorts, and of course during the pandemic, we're all very widely interested in different kinds of airborne viruses or communicable viruses, some of which are foodborne, some of which are waterborne, some of which are airborne. These can be on food products and ingredients, and the substrate that the viruses are on can have a serious impact on how effective any kind of intervention might be, but the same could be true for cold plasma. As you'll see during this presentation, different types of cold plasma systems have shown efficacy against a lot of different kinds of viruses. And these can be medical viruses, veterinary viruses, phytopathology viruses, as well as along with the viruses that might influence humans, which we'd be concerned with primarily from a food safety or a food processing standpoint. So here's an example of a system that we worked with. This is some work coming out of my lab, where we were looking at plasma jet as a way to inactivate norovirus surrogates. And this is using blueberries as a stand-in or as a model system for any kind of small fragile fruit. So these were inoculated with a couple of different surrogates for norovirus, murine norovirus and two-lane virus. We looked at a lot of different things in this study, but I'll only be focusing on the viral inactivation here. We looked at how much we were able to inactivate the virus, these two different surrogates for norovirus, and also looked at what the heat was doing to the berries, so we could get a sense of what kind of engineering controls you would need to have this applicable in a real-world scenario. And what you can see for both two-lane virus and murine norovirus, to make a long story short, the longer you treated it with the plasma, the better the kill you got. So we were ultimately able to reduce these down multi-log inactivation until they were past the point of detectability. One thing that we saw in this system where the berries were inside the jars, we were capturing all of the plasma reactive species, and the temperature was going up. And it got to the point where we were raised this to the point where we started to get hot enough that this was beyond the point of thermal inactivation. So to answer the question of, well, is this just a thermal inactivation or is there actually some plasma chemistry going on? We altered the system and introduced some engineering controls to keep the temperature down below where you would see thermal inactivation. And when we did that, the virus was still inactivated. So you had a little bit of diminution in the efficacy of the plasma, but the virus was still going, was still going away, even though the temperature was not hot enough to make this a thermal process. So in this work, we demonstrated that, yeah, this is a non-thermal process. And so here, this opens the door to applicability to room temperature or refrigerated systems, or even sub-freezing applications of plasma as a way to inactivate these types of norovirus surrogates. Looking at a different study by Choi et al., this is an activation of actual norovirus on oyster meats. So we go from blueberries to oyster meats, very different system, and yet you still see that the virus is still being inactivated. Now, in this case, the plasma by itself was inactivating the norovirus. Was getting rid of it. In this study, it was very interesting. They introduced propydium monoazide, which is a dye to look at cell disruption. And what they found was this enhanced the kill of the plasma. And so what we're thinking in the study, what they said is that the way that the propydium monoazide was interacting with the DNA of the norovirus was actually preconditioning it to make the plasma more effective. And you'll see this as a look at some other things where plasma by itself can be quite good, quite be useful for inactivating your target pathogen. But there's a lot of opportunity to combine plasma with other types of chemical interventions or physical interventions to make both of them work better in an additive kind of a process. Here we have another study looking at a different kind of a system. The previous study was looking at dielectric barrier discharge. This is looking at a plasma jet inactivating norovirus on salmonella. So I don't believe that I'm muted. I'm getting a message.
No, it was 2 minutes, Dr. Brendan. It just was 2 seconds. Sorry.
Oh, okay. All right, great. Well, if my sound does cut out again, please do let me know.
Yeah, I will.
All right, thank you. So on this study where you're looking at where you're looking at the plasma applied to inactivate norovirus on salmon, the key part about this study is they were looking at multiple gases. So in many plasma systems, we just use air. And the reason for that is that air is really cheap. One of the advantages of cold plasma is the cost of inputs. If you have air and electricity, this could be a lot cheaper than if you're using organic acids, or you're using peracids, or you're using other kinds of systems that would need to be purchased from a chemical supplier. Air and electricity can be very flexible. It can be done in a lot of places in a processing plant or out in the field on processing equipment, because you're not having to carry a lot of things along. But the question is, what kind of gas to use? So if you just use air, then you've got a mixture, obviously mostly nitrogen, with 22% oxygen, and you get very effective reduction of the norovirus on the salmon. If you just use pure oxygen, then you still get the reduction. It's actually not as good as if you're just using regular air. And if you're using pure nitrogen, you still get a little bit of reduction, but it's not nearly as good as you see when you have predominantly oxygen that's available to be ionized. Part of the reason for that is because of the plasma chemistry. So oxygen is a double bond atom. Nitrogen is a triple bond atom. Nitrogen, that triple bond is very difficult to break. It's very hard to break entirely. Oftentimes you'll break one of those bonds or two of those bonds, leading to a partially ionized or partially broken molecule of nitrogen. That then will recombine with oxygen singlets out in the plasma plume to make nitrous or nitric oxides, which can have their own physiological effects. But from a straightforward interaction, a straightforward reduction of the viruses, having a preponderance of oxygen available to accept that voltage to create those oxygen singlets is really going to give you really, really good results in terms of the antimicrobial or antiviral activity that you see. So this is a study looking at aerosolized MS2 virus. MS2 is a bacteriophage of E. coli. It's used widely in lots of studies looking for viral inactivation. So this is a different kind of a system where instead of having the plasma created and then applied to the surface, you had the plasma generated inside an air reactor, essentially a bead reactor, and then the aerosolized virus was passed across that. So this is really kind of an air cleaning environmental type inactivation. And what you see from these from this system where you had the plasma being on, multi-log inactivation, at 15 minutes, at 30 minutes, or 45 minutes, as this virus is passing through the reactor system, you're getting serious inactivation, multi-log reduction of this. So this can be very recirculating. This can be very good for controlling of environmental contamination or airborne viral particles. In the same kind of a system where they looked at a different kind of a thing with cold plasma jet, again, this is with MS2, where you apply the either air or argon, an argon oxygen mixture or a helium argon mixture to this, you get this viral inactivation. So why might you be concerned with an argon or a helium inactivation? The reason for that is argon and helium take a lot less electricity to ionize. So they're a lot more expensive than air, but they take a lot less electricity. And because they use lower voltages to ionize, then your equipment can be smaller, your equipment can theoretically be cheaper. There are some applications where this type of a trade-off might make a lot of sense. For medical applications, dental applications, you might want to have a smaller, more portable, somewhat cheaper system to treat, say, dental caries or ocular problems or skin tumors or things like that, where you want those reactive oxygen species to inactivate viruses, say for herpes virus or other kinds of skin lesion type viruses. In the context of food processing, the more expensive you make the feed gas, the more problematic that is for larger scale industrial application. Another thing that was interesting about this was that if you take the virus and apply it to air, you get a 1.3 log reduction after just a second of exposure. It's very, very quick. If, on the other hand, this virus is waterborne and you're pushing the plasma into the water, it takes longer for that to for that to inactivate the viruses. Both of the noble gas systems, gas mixtures, argon-based or helium-based, because 98% argon, 98% helium with the balance being oxygen, both of these do yield a lot of reactive oxygen species. All right, so there is a lot of chemistry that goes on when you break apart the noble gas that then contributes an electron to an intact oxygen atom, oxygen molecule to break that up into monatomic oxygens. If you just use pure argon or pure helium, it's not really very effective. You don't really get very good results. You need to have a little bit of oxygen to create those oxygen singlets that really do a lot of the heavy lifting in terms of the antimicrobial or antiviral activity. So here we have a test looking at a Newcastle disease virus. This is an infectious virus of animal pests or of animals, of poultry and livestock. Causes lethargy and death. It's very serious. In this system, what they did is they took plasma, the plasma electrode, and they shot it into a solution, either just water or into a mild saline solution, a 0.9% sodium chloride solution, or a 0.3% hydrogen peroxide solution. Again, a fairly mild sanitizer. They got complete inactivation of the virus after 30 minutes in this system. But these graphs are very interesting because they really illustrate the kinds of chemistry that you see when you create plasma. So the graph in the upper left, the oxygen reduction potential, all three systems, all three solutions, just water, a very mild saltwater solution, a very mild peroxide solution. All three of these have a lot of increase in their oxygen reduction potential. So all three of them are suddenly becoming very effective sanitizers, just with this kind of plasma injection. We look at peroxide concentration in the graph in the upper right. Again, water, essentially no peroxides. All three of these systems suddenly go up to 1.7, 1.8%. So starting with just water, you use the plasma, you get a lot of peroxides. If you start with a slight peroxide solution, this is going from 0.3% up to 1.8%. So this suggests that even starting with water, as opposed to starting with a defined chemical solution, you're getting this kind of peroxide reduction. You look in the lower right, you go from water, which has a pH of about 7. All three of these systems, water, salt, or peroxide, they have a pH after the treatment of pH 3. So you get this acidification solution. So what's going on here? When you're ionizing water, the H2O will be broken up into H and O. So you've got hydrogen singlets and oxygen singlets. Well, that hydrogen singlet is just a naked proton. So that is a pure acid. Chemically, that's as pure an acid as you can get. So you get a rapid reduction of pH. So you can get this acid effect in an acid sanitizing solution. But that hydrogen singlet and the oxygen singlet can recombine to make hydrogen peroxide. So you're creating a peroxide solution when you're introducing the plasma system. Now, in the final graph in the lower left, the conductivity, water all by itself is not terribly conductive. But all three of the other systems, then after you introduce this plasma system, you're adding to the conductivity of the water. And not surprisingly, if you start with a slight salt solution, the sodium and the chloride ions, those contribute to the overall conductivity of the system. It's not quite like creating electrolyzed water, some of those other types of systems you might be familiar with, but it's kind of related to that. So these were all done in vitro, where you have the virus suspended in solution. How well does it work in vivo with actual eggs, with actual chicks? If you don't have any kind of control, if you just use water, then the eggs that have this Newcastle disease virus, they're all going to die. I mean, they're all going to die if they have the virus. However, any one of these three peroxide, whether it's peroxide or salt or starting with just plain water, any one of these plasma activated solutions will inactivate the virus completely. So when you inject this, you have complete recovery of all of these inoculated or affected eggs by these systems. All right, so this really suggests that plasma is very, very flexible. You can start with peroxides, you can start with other kinds of chemical systems if you want. But even if you're just starting with plain water, you get serious benefits in terms of sanitation, viral inactivation, and ultimately industrial application for plasma systems. So here's one where we're looking at inactivation of influenza A, obviously a human contaminating virus. So this is a system that uses a hydrogen peroxide mist that is shot across a small, dense electric arc. So that creates a plasma plume with ionized species resulting from the peroxide introduction, Where you have no spray, your viral particles will grow and persist on masks. This is a system that was done looking at inactivation of N95 respirators, looking at influenza A. There is other data out there looking at inactivation of COVID-19 and of SARS virus and other type of inactivation. This kind of mask is very difficult to sanitize because it's a mesh web. It's hard to make sure that you're sanitizing molecules. The reactive species are getting in to inactivate the viruses that might be adherent to the fibers. But here you have really, really good inactivation of these viruses. This suggests a greater application of plasma, this kind of combination of peroxides and plasma for different kinds of applications of different sorts of food contact surfaces. And so in conclusion, that there's different kinds of ways to make plasma, different ways to apply it. It is broadly effective, and there's ways to achieve these inactivation of the viruses and still have a good looking, good tasting, saleable product at the end of the day. And with that, I would be happy to answer any questions if we have time. And again, thank you very much to the organizers for who have invited me to be with you today.
Thank you, Professor Brendan Niemira. Thank you for the nice talk and presentation. I think we have time for one or two questions. Anyone in the audience have any question?
Yes.
Okay,
Yes.
go.
I would like to know if you noticed any changes in the organoleptic properties of the food treated with the plasma. Thank you.
Sure. Yeah, there are. Well, there can be changes. I think, like any industrial process, if you turn it up too high and run it too long, you'll start to see some impacts. One of the things that we found with the blueberries that I did looking at the norovirus surrogate studies was that if we ran it too long, then the temperature accumulation on the berries would start to see some softening or even melting of the wax coating on it. So they sort of go from being that kind of a hazy, cloudy surface to be kind of shiny. If we really ran it too long, we started to get some thermal damage. They started to turn to mush. And that's why we introduced the temperature controls to keep the temperature down. With that in place, that thermal or organoleptic properties reduced quite a bit, to the point where it's hardly noticeable. For things like seafood or for chicken breast or for other type things, things that have a lot of moisture in them, just the airflow itself of a plasma jet can lead to some drying of that, so you need to control for that. But I think it's like any kind of industrial process where, if you can get really, really great kill if you run it a long time. I'm on Zoom, so I'll do vertical instead. If you run it a long time, you get great kill, but you start to see some damage. If you keep it down to the point where you don't see any damage, you might not get the kind of kill that you want. So the goal here is to have an optimized level, right? Where you're getting the kill that you want, but you're not getting any kind of other sort of organoleptic impacts.
Thank you for your answering the question. If anyone else has any question. Okay, Professor Brendan, I just have a quick question. Do you think in the near future we will see this technology at the real industrial level in different processes?
The technology is developing rapidly from just from a straight technological standpoint, developing plasma systems that are cheap and easy to use and are effective and are flexible and can be slotted into different types of industrial applications for foods and food contact surfaces, targeting different sorts of organisms. One thing that needs to be in place for it to be used commercially, of course, is regulatory oversight. It's one thing for it to work, it's another thing for it to be legal to use. And one of the things that's happening right now is regulatory bodies in the US and in Europe and India and around the world are looking at what are the controlling legal rules on how you use it, where it can be used, how you verify that it's working properly. One thing that can happen with an emergent technology is somebody will create a device and say, oh yeah, it works great, this is terrific, but it doesn't really work the way it's promised. This is one way for process validation to take hold and to have proper controls on how the systems are being used, so that it is actually developing the kill or inactivation for viruses or bacteria or whatever, to make sure that it's actually doing what it's claimed to be doing, and that it's not doing things that we don't want it to do. So you don't have unwanted reduction of nutrition or vitamin inactivation or other kinds of unexpected or unwanted impacts.
Thank you very much, Professor Brenda, for the nice presentation.
Thank you all very much.
I think we can move to the next part of the session, which will be the oral presentations. We have 4 oral presenters. The first one, it will be the establishment of lipidomics applying in strawberry gray mold interaction, presented by Dr. Quang Ma. Quang Ma, are you here?
Yeah, thank you, Professor. Can you hear me?
Yeah, perfectly. I sorry if I didn't present your name really well. It's Quang.
It's okay. It's okay. You can call me Mark. You can call me Mark.
Okay, Ma. Yes, if you want to introduce yourself and start your presentation, go ahead. Thank you.
Okay. I wonder if my slide move.
So far, I cannot see your slide.
Can you see it now?
No. Perfect, Dr. Ma. Perfect. Now we can see it moves perfectly.
Yeah. Okay. Thank you. Good day and good night. It's my pleasure to present. I am Ma Quan from Zhejiang University in China, and my supervisor is Professor Luo Zhishen. And here I want to present part of our recent research, which is about the establishment of the lipidomic analysis of strawberry and its application in strawberry and gray mold interaction. And we're going to present according to these 3 sections: the background, main results, and the conclusions and acknowledgements. First, let's start with the introduction of the background. Strawberry is an important economic crop worldwide. However, in developing countries, the post-harvest loss of fruit products is about 13%, and we believe strawberry will be more. The post-harvest loss of strawberry were primarily caused by disease and quality loss. Among various diseases of strawberry, gray mold caused by botrytis cinerea is devastating to strawberry industry. However, up to now, there are no measures that can efficiently control the gray mold disease of strawberry. And these situations indicate that the discovery of the underlying molecular mechanisms are urgently demanded. And the lipid compounds are very important to immunity of plant. Lipids are energy storage compounds which will rapidly change while facing with pathogens. And besides, lipids, including glycerophospholipids, are the primary substances of membrane, which is responsible for the anchoring, transportation, and the fold of the resistance proteins. Lipidomics is an emerging approach for analyzing the change of lipids in biological samples and to elucidate the molecular patterns between gray mold disease in strawberry from the perspective of lipid. We established this lipidomic analysis method. And here we're going to perform the main results of this work. We developed the experiments based on the comparison between the mold group and the gray mold inoculated group. We first established the gray mold inoculation model, and the gray mold lesion significantly, we can see here, significantly increased at 2 days after inoculation with the rising ratio of BC tubuling to FA23S. And on the other side, mold group underwent a normal post-harvest ripening stage, indicating the reliability of this model. And the extraction of the lipidic compounds is the most important process of lipidomic analysis. Hence, we tried 5 extraction methods to optimize the input substances. We tried the FOCH 1 and 2 and 3 and the dichloromethan and the MTBE methods. These methods were performed for optimization. The total ion chromatogram figures are present here, and some peaks were found significantly higher than other methods while utilizing the dichloromethan method. And to further validate the optimized results, we used the MASTEL for identifying the detected compounds. As a result, method D, which is the dichloromethan method, obtained the most compounds among 5 methods. So we finally selected method D. And the UPLC coupled with Q-TOF secondary mass spectrum system was utilized. And the details of the LC and the mass sections were referred to our previous work and modified according to the related reports. After obtaining the data, PLSDA analysis performed that in inoculated group, all samples were separated well, and in mold group, the 1 and 2 days after inoculation cannot be separated very well. We also underwent BCA analysis, which performed similar results. And eventually we obtained 203 and 249 lipidic compounds in MOC and the inoculated group respectively. And for 114 lipids were commonly detected in 2 groups, and 121 were only detected in the inoculated group. Among 114 lipidic compounds which were commonly detected in MOC and the inoculated group, we selected lipidic compounds whose p-value of ANOVA and the t-test were lower than 0.05, and the VIP value was higher than 1. And finally, 35 differentially expressed lipids were selected according to the standard. And we drew a heat map to visualize their expression of the 35 DELs, the DLS DELs. And we clustered the lipidic compounds which performed in a similar expressive pattern. And for each cluster, we drew a variation trend which were in response to the heat map. And we selected the lipidic compounds that were clustered into different clusters in MOC and the inoculated group. We could see the expression trends of them were different between 2 groups. And we also did correlation analysis between DELs and the lesion diameter. We found HBMP 54 to 7 were highly correlated with the lesion diameter, which was not significantly in MOC group. And pathway analysis was also performed. We utilized both of the database of Arabidopsis, the model plant, and Fragaria vesca, the diploid strawberry. As expected, the glycerolipid and the glycerophospholipid metabolism were enriched the most significantly. And besides, biosynthesis of the secondary metabolism were also enriched into database, combined with the induction of the related genes. And these are the main results of our current work. Generally, in this work, we established a set of measures for optimally obtaining and processing lipidomic data of strawberry. Although we tried our best to complete the aims and the scopes of this work, it is still difficult to separate and recognize the lipidic compounds in strawberry or in gray mode, respectively. Thanks for spending time on listening to this presentation. This work is financially supported by NSFC, and we would like to thank all people who kindly give assistance on this work, especially Dr. Taran, for introducing and providing chances to present in this great conference. And thanks for all efforts contributed by Amity University and Zhejiang University. And that's all. Thanks again for listening. We welcome any discussion and cooperation. Thank you.
Thank you, Dr. Ma, for your nice presentation. And I think we are running a little bit of time, so we will raise the questions for the afterwards. All the oral presenters are finished, if that's okay. But thank you for your presentation. Very nice.
Okay. Thank you, Professor. Nice to meet you.
So next presentation will be adulteration of cadmium heavy metal in food, potential health risk for humans. The presenter, it is Harsh Sable. Harsh Sable, are you here? Dr. Harsh? I cannot hear you, Dr. Harsh.
Am I audible now?
Yes, now yes, we can hear you, not see you.
Okay, just a minute. Okay, thank you. Good evening, everybody present here. The topic of my presentation is contamination of cadmium heavy metal.
Dr. Harsh, just one thing. Can you put your presentation? Yes, exactly. Thank you. Introduce yourself, please.
Yeah, thank you. My name is Harsh Sable. I am a PhD research scholar from Sharda University in the Department of Forensic Science in the School of Allied Health Sciences, Sharda University, Greater Noida, India. The topic of my presentation is contamination of cadmium heavy metal in food, potential health risks for humans. So as we all know, food is one of the basic necessities and the vital element for every human being. So what is food? Food is any material made up of carbohydrate, water, and proteins that people and animal can consume or use for nourishment or for the vital components of life. what is food adulteration? And food adulteration is a thing where we add any poison substances or anything for the personal gain. So that affects the basic nature of the food. So food adulteration happens on the worldwide scale and variety of forms, affecting the nearly all food items. So what is heavy metal? Heavy metal poisoning yeah. So heavy metal poisoning is this thing where the accumulation of heavy metals in the toxic amount in the soft tissues of the body. The poisoning of cadmium in human occurs due to its high exposure via various different ways, such as food, water, breathing, air pollution, skin contacts, and workplace exposures. As you can see in this schematic diagram, the humans are exposed to the heavy metals through industrial activities, mining, the air that is polluted. Through that, we can get the toxic heavy metals. The seafood that we can consume contains the heavy metals, or the vegetables or the cereals or the tubers that we use contains the heavy metals that enters to the vegetables through the ground or the irrigating water that we give them. Heavy metal poisoning is the result of toxic accumulation in certain metals. Such heavy metals compete with and replace certain essential minerals, in the course of which any of the several body organs or system can be affected. What is the basic things that happens with the food adulteration is it's a kind of a food fraud that results in the financial gain for the manufacturers, but ultimately results in a loss for eventual consumers of the items. The food adulteration is a long-standing and a widespread problem that affects both low and middle income national countries as well as industrial countries. So how cadmium enters the environment? So as you can see in the diagram, the air pollution, the polluted airs that is removed, excreted out from the industries, the cadmium can be mixed in the air, and the water that is excreted out contains cadmium as well. So the groundwater is also groundwater get polluted, or the aquatic life or the water bodies that are present near to the industries get affected with it. And due to that, the irrigating water that we are providing to the crops or vegetables gets the cadmium from the water and from the soil as well. So the heavy metals that we that are gets accumulated in our body can have serious effects on our body. Can decrease the liver function. Can cause this yellowing of skin, intestinal diarrhea. It can cause can cause the muscular weaknesses, cardiovascular diseases, and the stomach infections as well, and the no urine production. And the basic toxicity that the cadmium causes, it affects the bone, it affects bones, it affects our renal system as well. So the cadmium level in food varies depending on the geographical regions, bioavailability from soil, agricultural genetics, agronomic studies, strategies utilized, and post-harvest procedures. The maximum allowable cadmium value is 0.2 1 gram per gram, are recommended by the WHO and the United Nations Food and Agriculture Organization. The 1 mg per milligram is fatal, considered fatal for the humans immediately. The cadmium, it affects the respiratory system. It causes pneumonitis. It causes destruction of mucous membrane. It affects the kidney. Causes proteinuria, kidney stones, glomerular and tubular damages. It affects the bones on the skeletal system. For example, like loss of bone density and mineralization and eye disease. And it also affects the reproductive system. For example, testicular necrosis, estrogen, like if affects the affection of steroid hormone synthesis as well. So the generation of free radicals is the main mechanism of its toxicity in human beings. It causes oxidative stress and damages biological molecules like enzyme, lipids, protein, and nucleic acids. So as this diagram shows, the cadmium that enters can enter our system through water, soil, and air. For example, in food crops, it enters through soil. So the food that is affected or the food that contains the cadmium in high percentage enters our body and cause biomagnification processes, and it affects it ultimately affects the different organs of our body, causing the mortality or like cancer or different diseases. So for the analysis purpose, we can use the AS, which stands for atomic absorption spectroscopy, or EDXRF, or ICP, inductive coupled plasma, or inductive coupled plasma atomic emission spectroscopy, are the techniques that can be utilized for measuring the trace levels of cadmium in the samples that we are going to analyze. Or even ICP AES can be used for the multi-elemental assessment of biological materials as well. Some of the suggestions from my side for reducing the cadmium in food are the adaptation of good agriculture practices to minimize the cadmium contamination in food crops, or to obtain the food supplies from reliable sources, such as like sources where we can doesn't use the mineral chemical fertilizers, or ensures the food imported or for sale are fit for human consumption and comply with the legal standards. Applying lime to soil can increase the pH and decrease the bioavailability. Increasing organic matter and microbial activity regulates the solubility of cadmium. And determining salinity and the chloride level of irrigating water can minimize the uptake of cadmium in crops. And for reducing the cadmium level in water, we can use the bioremediation techniques to remove cadmium from water, so that it can be fit for consumption. Okay, that's it. Thank you.
Thank you very much, Dr. Harsh, for your presentation. First of
Thank
all,
you.
I want to say my apologies to Dr. Ma, because previously I said there was no time for questions. We were running a little bit late, but I just been told that we have a little bit time for questions. So if anyone wants to ask a question to Dr. Harsh, please go ahead.
Hello.
Hello.
I was wondering if you consider that adding lime to soil will mobilize other heavy metals besides cadmium.
Actually, I haven't done studies about that, so I have no idea about that. But
Okay,
maybe it can. Yeah.
Thank you.
Yeah. Thank you.
Thank you. Thank you, Dr. Harsh. I think we can move to the next presentation. Nobody has any question. Anyone has any other question? So I think we move to the next presenter. Next presenter is about review of various food safety analytical techniques for the prevention of foodborne illness. It will be presented by Ritu Tangwar. Dr. Ritu Tangwar, are you here presented?
Yes, sir, I'm here.
Hello, nice to meet you, Dr. Ritu Tangwar. I hope I can pronounce your name right. Otherwise, you can teach us how to pronounce your name.
Sir, I'm Ritu Tanwar.
Perfect. Can you share with us your screen and also your camera so we can see you?
Yes, sir, sure. Sir, is my slide visible?
Yeah, your slide is visible. Please, if you can, use full mode presentation. Yes, perfect, Dr. Ritu Tangwar.
I am Ritu Tanwar, student of MSc Microbiology from Calcutta University, Greater Noida, India. The title of my presentation is Food Safety Analytical Techniques for the Prevention of Foodborne Illness. I would like to thank the organizer for giving me this wonderful opportunity. This is my abstract on the topic review on various food safety analytical techniques for the prevention of foodborne illness. First of all, what is foodborne illness? Foodborne illness is any illness resulting from the spoilage of contaminated food by pathogenic bacteria, viruses, or parasites that contaminate food, Symptoms vary depending on the cause, but often include vomiting, fever, aches, and may also include diarrhea. Foodborne illness arises from improper handling, preparation, or food storage. Food safety, the action of monitoring food to ensure that it will not cause foodborne illness is known as food safety. It refers to the handling, preparing, and storing food in a way to best reduce the risk of individuals becoming sick from foodborne illness. Food safety is a global concern, a variety of different areas of everyday life. The principle of food safety aim to prevent food from becoming contaminated and consuming food poisoning. This is achieved through a variety of avenues such as properly cleaning, sanitizing equipment and utensils, maintaining a high level of personal hygiene, especially hand washing, storing, chilling, and heating food correctly with regards to the temperature, environment, and equipment. Food safety analytical techniques for the prevention of foodborne illness are quantitative real-time polymerase chain reaction, gas chromatography olfactometry, enzyme-linked immunosorbent assay, pulse field gel electrophoresis, and flow cytometry. The presentation will focus on two techniques, that is gas chromatography olfactometry and ELISA. Now, the first technique is gas chromatography olfactometry. It is basically a technique that integrates the separation of volatile compounds using a gas chromatograph with the detection of odor using an olfactometer. In this olfactometric detection, a human assessor may qualitatively determine whether a compound has odor activity or describe the odor perceived. Or qualitatively evaluate the intensity of the odor or the duration of the odor activity. It allows the assessment of the relationship between a quantified substance and the human perception of its odor. Basically, it is a method to combine the information supplied by chemical characterization and by odor perception. In this presentation, the four major methods for gas chromatography are described and their potentials and limitations discussed. The methods include dilution analysis, detection frequency method, posterior intensity method, and time intensity method. Advantages of gas chromatography olfactometry. Gas chromatography in combination with olfactometric techniques can help to detect potent odorants without knowing their chemical structures, even at very low concentration. It is widely used in industries for quality control in the manufacture of many products for research purposes, from the analysis of meteoroids to natural products, and for safety from environmental to food to forensics. These are some of the examples of food and their functions, food products and their function in which GCO technique, that is gas chromatography olfactometry technique, is used for analysis or detection, such as coffee, milk and cheese, meat, fruits, and their function is flavor investigation and odor activity. Next technique is ELISA. ELISA is basically stands for enzyme-linked immunosorbent assay, which is commonly used analytical biochemistry assay. It was first described by Engel and Perman in 1971. It is a technique to detect the presence of antigen, antibodies, proteins, and glycoproteins in biological samples. Next is sandwich ELISA. It is basically one of the type of ELISA technique. It is most commonly used format. This format requires two antibodies specific for different epitopes of the antigen. These two antibodies are normally referred to as matched antibody pairs. One of the antibodies coated on the surface of multi-well plate and used as a capture antibody to facilitate the immobilization of the antigen. The other antibody is conjugated and facilitates the detection of the antigen. A capture antibody on a multi-well plate will immobilize the antigen of interest, or we can say the target antigen. This antigen will recognize and bound by a detection antibody conjugated to enzyme, biotin, and streptavidin, HRP. HRP is basically the horseradish peroxidase. ELISA technique exhibits following advantages. First is high efficiency, as simultaneous analysis can be performed without complicated sample pre-treatment. It is safe and eco-friendly. It is cost-effective assay. It is simple procedure, high specificity and sensitivity because of an antigen-antibody reaction. These are some of the examples and function of ELISA technique in food products that involve meat, fish, milk, fruit juice, etc. And their functions are species identification, protein detection, species discrimination, and labeling authentication. So the conclusion of my presentation is that now we understood that the food are substances which provide nutritional support for the body. These analytical procedures are used to provide information about a wide variety of different characteristics of foods, including their composition, structure, physicochemical properties, phytochemical properties, and sensory attributes. So food analysis and quality control process are compulsory for all type of food industry. These are the references of my presentation, and this is all about my presentation. Thank you.
Thank you, Dr. Ritu Tangwar, for your presentation, for your nice review and presentation. If anyone has any question, please. I think Rita Tangwar, we don't have any questions, so we can move further to the next and last presenter, Physiochemical.
Yeah, sure.
Nice to meet you. Thank you for your work.
Thank you so much.
So next presenter, it will be physiochemical and nutritional characterization of gluten-free snacks based on flax seeds. Please welcome to Dr. Wong Moore. Dr. Wong Moore.
Yeah, Dr. Wagmare.
Roger Wogmore. Sorry if my pronunciation was not right.
It's okay, sir. I'll be sharing my screen.
Yes, we can see your screen. Yes, we can see the full presentation, the introduction on page 2. Maybe you can move to page 1.
Yes, sir, sure.
Perfectly. Yes, it moves perfectly.
Yes, sir.
Yes.
Good evening, everyone.
Present yourself a little bit. Thank you.
Yeah, I'm Dr. Rosie Wagmare, and I'll be presenting a topic, physicochemical nutritional characterization of gluten-free snacks, which is based on the flax seeds. So basically, the consumer interest and attention towards healthy and nutritious food products has been increased in the recent decade. And according to the experts at the All India Institute of Medical Sciences, celiac disease affects around close to 6 to 8 million people in India. And you can see gluten-free market has recently become one of the most recognized market in the food industry, which is showing increase in the annual sale growth and new products in both food and beverage category we can see in the market. And awareness of the gluten-related illness has also been increased. Thus, increasing trend in research is focusing on the application of alternative grains to elaborate gluten-free products which are healthy as well as inexpensive. So basically in this product development, we have used flax seeds because flax seeds has been used as a precious nutritional food grain and traditional medicine in human diet for thousands of years. And more recently, it has been used as a source of nutraceutical and identified as a functional food. Where you can see there are a lot of health benefits which are generally attributed to high concentration of linolenic acids and lignans, as well as significant quantities of dietary fibers are also present in the flax seeds. So flax seeds has a unique, also have a unique fatty acid profile. It is high in polyunsaturated fatty acids and moderate in monounsaturated fatty acids. And it is also low in saturated fatty acids. So basically in this project, we have used Indian snacks, that is a matri, which is a traditional snack product consumed in the India. And actually matri is a popular deep fat fried Indian snack. And which is traditionally, if we see its preparation, it is traditionally prepared from the refined wheat flour, water and spices. So if traditional preparations like matri when modified with different combinations of flour could serve as a means of enhancing nutritive value of the food. So actually we have used, instead of refined wheat flour, we have used chickpea flour. And because a variety of fried traditional convenience and confectionery snacks are prepared from the chickpea flour in India. So actually this chickpea is a pulse legume that has a important source of gluten-free proteins. So therefore we have added chickpea as a main ingredient to substitute gluten in gluten-free matri formulations. And the carbohydrates of legumes such as like chickpea are high in resistance starch. That is actually associated with a slower digestion rate, therefore slower glucose release into the bloodstream. So this is actually the preparation of the flaxseed powder. So here we have taken the flax seeds. Those flax seeds are carefully cleaned, and all the extra impurities, anything has been removed. Then that was dried at 60 degrees Celsius for around 1 hour. Then it is grounded to get the powder, and then it is packed in the pouches for further application. And this is actually the formulation. And in this, matri was actually prepared by using different blends of chickpea flour and flaxseed powder. So control was 100% chickpea flour, and remaining we have substituted flaxseed with 10, 20, 30%. And all these the spices, red chili powder, turmeric, mango powder, black pepper, we used. This is the flow chart for the preparation of the traditional product, where we have taken as a formulation. Earlier I have mentioned that formulation we used. Then we prepared the dough, and it was conditioned for 10 minutes, and then it was like rolled into a dough was rolled to about like 3 mm thickness, and then it was molded to 4.5 centimeter diameter, and it was then deep fried for 2 to 3 minutes at 120 degrees Celsius. And stored in, it was then stored in pouches for further analysis. These are the actual pictures of the product, where we can see in control the color is like appealing, but as we have increased the percentage of matri, you can see little brownish color has been came in the product. So nutritional composition also we have observed, where we have seen like as the concentration of this flaxseed has been increased, the protein contain significant little percentage of protein has been increased, fiber also almost like you can see little some variation with the increase in the flaxseed we have observed. Then we have also seen the mineral composition. In that we have seen iron, phosphorus, and calcium. So with the calcium, we can see with all these minerals, as flaxseed is a pack of nutrients, so all this percentage also increased. And we have also seen the sensory. In sensory, actually the initially highest sensory score for test was reported for control, while the lowest score was reported for this flaxseed, which was prepared with 40% flaxseed, because people don't like that much taste of flaxseed. So the score for test like reduced with the increase in the concentration of the flaxseed. And we have also done the storage studies for around 60 days. And you can see like even 40% of product which contains flaxseed was acceptable, like consumer accepted it, they didn't reject, but the score was lower. And overall acceptability was slight highest. Overall acceptability was slight highest for 10% flaxseed, then 20% was much lower than 10%, then 30, and was less for least for the 40%. So basically the prospects of the functional food as well as the gluten-free product in the market is enormous. And in this product we have seen various analysis, also the sensory analysis, and we can say like 30% can be the acceptable. Even 40% is also there, but 30% consumer can easily consume it. And present study strongly support flaxseed is a treasure of nutrients which can be successfully incorporated in other food, traditional or as well as other food products successfully. Thank you.
Thank you, Rogi, Dr. Rogi Wogmore, for your nice presentation. If anyone has any question, we are open for listening.
Yes, hi, I would like to ask a question to Lovely and Sarma. I would like to know if you have added this floor to another product or do you have plans to do it?
No, ma'am. Actually, we have traditionally we use this refined flour only. So instead of this, we have used this besan, which is chickpea flour, and flaxseed also. So at laboratory scale only, we have prepared the product.
Okay, thank you.
Thank you, Dr. Rogi, one more for the nice presentation. I think this is if no one has any other question, this is the last presentation from this technical session. I think I give the word to Dr. Alok Saxena.
Yeah, thank you, Dr. Miguel, for nicely or excellently chairing the session. So I think there is no question in chat box also. So we can sum up the technical session. So I thank Dr. Miguel for chairing the session and other speakers, Professor Zingyu and Dr. Brandon, for explaining their thoughts and work on the protection of pesticides, then synthetic and natural antioxidant systems, and cold plasma as a sanitizing system for the food items. So I request Dr. Lovely to propose a vote of thanks for this session, technical session 2.
Yes, sir. Thank you, sir. I would like to express my gratitude to all esteemed delegates of the session for their presence and contribution, especially Dr. Medjul. Thank you so much, sir, for chairing the session. And thank you, Dr. Alok. And thank you, dear all students and participants. Thank you so much for joining this session. Thank you, all of you. So next session is technical section 4, and it will be started at 5:30. So we'll join the session at 5:30. Thank you so much.
So Monica, we can close this session?
Sir, we can't close the session. This will go on. Good
Yes,
evening,
we'll go on
Dr. Egan.
We'll move to the fifth, fourth
Yes, sir.
session, technical session 3.
Yes, sir. So good evening, Dr. Florence. Good evening.
Good evening to you.
Okay. So we have to start now at 5:30. So we'll wait for some time and then we can start the session.
Please excuse me, what time is it at your time?
We have to start at 5:30.
Yeah, what time is it in India now?
It's 5:10.
5:10. So in
Yeah,
20
5:10.
minutes.
Yeah, in 20
Okay.
minutes we're going to start the session.
Perfect. Thank you.
Thank you. For all the participants, we can take a short break of 10 to 15 minutes and then we'll be back with the technical session 3.
Yes,
Thank
Professor
you,
Michael
Doctor.
is there.
Yes, thank you, Professor Michael. Thank you so much. Thank you.
We'll meet again at 5:30.
Okay, perfect. Thank you. Thank you to you all.
Thank you so much. Thank you. Good evening, Dr. Karuna. Good evening.
Good evening. Good evening, sir. Good evening, Dr. Monica.
How are you,
Mandeep,
ma'am?
good evening.
Yes.
Good evening, ma'am.
Dr. Sunayan.
Okay, Monica, we can start.
We can start. Yes, Dr. Mandeep, we can start the session now.
Okay, sure, ma'am.
Yes, please.
Okay, a very good evening to everyone present here. I welcome you all to this session, that is technical session number 3. The theme for this session is responsible consumption and sustainable diets. I extend my warm welcome to chairperson of this session, Dr. Karuna Singh, distinguished invited speakers, Dr. Florence Siegel, Professor Joy Regenstein, and all the dignitaries, delegates, and participants. The theme, as I've already told you, is responsible consumption and sustainable diets. For several years, we have become increasingly aware of the impact of our diet consumption. It affects not only society and economy, but also our planet and our health. As a result, we are talking about responsible consumption and sustainable diets, but how we can consume responsibly. In the theme of this session, we have the two keywords, which are responsible consumption and sustainable diet. Responsible consumption means how we can be responsible consumers and make more consumption choices that are good for the planet and for our society. Whereas sustainable consumption means to consume in a way that allows us to preserve our resources and the environment as much as possible. So based on this theme, we are going to have different lectures by our distinguished invited speakers, followed by the oral presentations. Now, it gives me immense pleasure to introduce the session chair, Dr. Karuna Singh. Professor Dr. Karuna Singh is at present a professor, nutrition and dietetics in the School of Allied Health Sciences, Sharda University. She has 17 years of experience. Dr. Karuna has done her BSc honors in food and nutrition in 1995 and her MSc in foods and nutrition in 1997 from Vanasthali Vidyapeeth, Rajasthan. She has done her PhD from CCS University in 2008, and she has guided 40 MSc foods and nutrition, MTech food tech students and BTech food tech students, 1 MPhil and 4 PhD students. Dr. Karuna has authored more than 35 research papers in journals of national and international repute, 9 chapters in books, and presented more than 40 papers in conferences. She also handled projects awarded by government agencies like DST and by industry. She has also worked actively in organizing FDPs, conferences, webinars, etc. And she's the life member of various reputed professional bodies like NIN, Mushroom Society of India, Science Congress, etc. And she has many awards to her credit also. So with this introduction, now I invite Dr. Karuna Singh to chair the session. The topic of presentation by Dr. Karuna Singh is Nutri-Cereals, that is millets, the 21st century smart food. Over to you, ma'am.
Thank you, Mandeep, for the nice introduction. And it's my pleasure to be here back to my place where I left. So in this theme thought, technical session 3, the theme is, as Mandeep has just discussed, responsible consumption and sustainable diet. As per my knowledge, sustainable or ideal diet is one that is healthy, which has sufficient quantity and quality of all the affordable nutrient which is required by a person, and which is affordable, which is culturally acceptable, and which fulfill all the nutrient and health demand. Basically, these days, if I am not wrong, we are producing a lot, but even then various countries globally, if I talk, we are facing various type of various levels of, in fact, we can say malnourishment, and which is an alarming sign, and which need to be tackled in this particular scenario. So in this regard, I would like to invite our first speaker of this session to just highlight some important point on this particular theme and on this topic. Dr. Florence Siegel is a medical doctor with a master's in community health for developing countries. After 10 years in the field of consultancy work in both humanitarian and developed context in Africa, Latin America, and in the Middle East, she joined the Food and Nutrition Division in the UN Food and Agriculture Organization, where she worked for more than 20 years on household food security and sustainable livelihood and urban food system, as well as interinstitutional participant, small-scale food processing to improve the diet and strengthening livelihood in both emergency and developed context in Asia and Africa. Upon retirement, she pursued work in urban food system and then in urban-rural linkage for integrated territorial development. She joined in 2015 in the multi-stakeholder group on humanitarian food science and technology, and was actively involved in participation in the UN Food System Summit. She will speak today on the topic, the message is think global, eat local. I would also request all the participants to drop their questions in the chat box, which we will take after her presentation. Over to you, ma'am. Over to you, Dr. Eagle.
Can you hear me?
Yes, madam.
Yes, yes. You are audible.
Okay.
Yes, you are audible.
Thank you very much. Well, first, I would like to thank the organizers for inviting me. By listening to the very interesting presentations in the past hour or so, I was very impressed by the scientific quality of these presentations. I'm afraid I will not be able to raise to that challenge. I'm not a researcher, I'm not a scientist, and but I come from maybe a different planet of the food world. So I'm going to talk of responsible consumption and sustainable diets, but in particular try and see how I perceive the role of food science and technology in the necessary food system transformation we're facing. I'm not very good at computers. Okay, it still to work. No, it doesn't. Oh, shoot. Sorry. I'll get there. Okay, so basically where are we at now? The existing food systems are not delivering healthy diets. This is not exactly surprising because that wasn't the idea. The diets we eat are the outcomes on a variety of food chains, each of which has been designed in order to generate profit and supply specific foods, but the way these foods are prepared and combined does not necessarily mean we are the best possible diet. So it is has been agreed internationally. You will see I have put a lot of links that you can perhaps look at afterwards, which explains where I get my information from. So it is urgent to transform food systems if we want to ensure food security and if we want to address hunger and malnutrition without compromising the health of our planet, because right now we are failing on this. And consumers have a key role to play. So instead of thinking of food policy only as a supply-driven process from farm to fork, maybe we need to start looking from a different perspective, starting from the demand of what is it consumer need to eat or want to eat, and how is that going to be modifying the system. So as consumers, we all play a major role. We all buy food every day, prepare food every day, and what we buy actually is the reason this food is produced in the first place. So we often do not realize that if we change our food habits, it is going to oblige people to change the way this food is produced and maybe have more sustainable systems. So the first thing is consumers should adopt healthier food habits. And there has been a lot of work done by the UN, but a variety of academia and other institutions on food-based dietary guidelines. Because obviously you're not going to eat the same thing in your part of India and in the Andeans part in Latin America. The other thing that consumers are encouraged to do, and we are all trying to do to a certain level, is to limit the food waste. We are buying food that we don't always eat. We are throwing that food away. We are preparing too much food and throw it in the garbage also. Another important issue that is coming back, we need to connect consumers back to nature. Very often people just think food from the supermarkets, and a lot of kids don't really realize where that food comes, how it's grown, etc. And we usually do not know where it comes from. So we, as consumers, we have a responsibility to reorient food production. And there is a lot of work done all over the world on trying to encourage short food chains, eat local foods and eat foods in season. Eating raspberries, strawberries now in Europe is way too early, and eating tomatoes in winter makes very limited sense. So we need to start to see how we're going to eat also more organic food, because it will be more healthy and taste better usually. It has to be equitable food. We need to make sure that by buying super cheap food, we are not responsible for farmers getting really very low prices for what they are producing, or landless laborers being actually working as slaves. There is more and more push to have limited processing. So that obviously is a challenge if I am talking in the kind of audience we are here. There is a lot of work now happening on revisiting norms and procedures, restricting marketing of unhealthy foods. Cosmetic norms are increasingly questioned because trying to insist that this fruit should be this shape and this appearance means that we're throwing out a lot of products that do not fit with those norms. And very often, if they nicely irrigated fruits usually don't taste really that good. I didn't want to talk of patents and all this and intellectual property, because that becomes an even more difficult area to move into. One of the things to do for policymakers is to make the good choice the easy choice, and try and make sure that consumers have access to the food they need for a healthy and sustainable diet. And another issue we're facing now is packaging, because we get more and more plastic all over the place, and we know what this has led to. The link I have provided at the bottom of that page has been the call for action that has been presented by Consumers International at the United Nations Food System Summit that took place at the end of September this year. What was that UN Food System Summit? Well, it was decided by Mr. Guterres, who is the Secretary General of the UN, and it was held during the UN General Assembly of New York, which is held every year in September, and it was done on September 23. The aim is to set the stage for global food system transformation to achieve the Sustainable Development Goals by 2030. 2030 is just down the corner. And if we do not change food systems, it's very unlikely even if we did, I'm not sure we're going to reach it, but at least these aspirational goals are really important to focus on which directions we're trying to go and are we doing the right thing. There was a pre-summit organized from 26 to 28 July. I've put the links. There was a scientific groups. Here again, you will see who were the scientists. I regret that there's very few food science and technology, and that is going to be very much part of my message today. We need you guys, because it is a very important issue, and you're not always around the table in these interdisciplinary groups. There has been a series of coalitions, you can look at that too, and in each country there has been a series of national dialogues and also independent dialogues, and each country has come up with national pathways on how they saw the way forward to transform food system. So I've added in the link the report that was provided for India. And you can find, in all countries, you can find that in the food systems dialogue on the UN Food System Summit. What are sustainable healthy diets? I will complement what Dr. Sharma has been saying. I'm sorry for the repetition. So it's dietary patterns that promote all dimensions of individuals' health and well-being, that have low environmental pressure and impact, are accessible, affordable, safe, and equitable, and are culturally acceptable. The aim of that is to have optimal growth and development of all individuals, and to support functioning and physical, mental, and social well-being at all life stages for present and future generations. And can show you there is the link to that document is here. And in this site, you see the guiding principles that FAO and WHO published after a technical consultation held in July 2019. Now, there are in these guiding principles, several of them are very relevant to food science and technology. The first one is looking in particular at how to get appropriate complementary feeding. The second one is to restrict highly processed foods and drink products. The idea is that these foods should include whole grains, legumes, nuts, and an abundance and variety of fruits and vegetables, and moderate amounts of eggs, dairy, poultry, and fish, and small amounts of red meat. It has to contain minimal levels or none, if possible, of pathogens, toxin, and other agents. And we've heard the presentation on foodborne disease before. There's big importance given to the importance of safe and clean drinking water as a fluid of choice, I suppose, rather than sweet drinks. The importance is to maintain greenhouse gas emission, water and land use, make sure the production of these foods is not going to have too much environmental or health damage. The importance of preserving biodiversity, limiting the use of antibiotics and hormones, limiting the use of plastic and derivatives in food packaging, and reduce food loss and waste, etc. So I think it's an important document. You may all be familiar with it. So my plea would be, we need you to join the sustainability movement. So I've already talked of the UN Food Systems Summit. And indeed, there has been a group of food science and technology experts that have been starting to look at what to do in humanitarian situations, and you will be able to find the links there. And COVID-19 has really shown much more specifically the importance of food science and technology. The call for the action of food science to have more importance given to food science technology, we need to develop practical, affordable, and sustainable processing technologies to improve and diversify local practices, to produce value-added foods in local communities, facilitating year-round access to convenience and healthy foods, including complementary foods for improved diets, contribute to saving time and labor in particular and not only of women, reduce food losses by preserving foods and supporting local distribution and trade. Local markets are very important. Generate income and create employment in particular for youth, women, landless laborers and migrants, and therefore contribute to the revival of local economies and strengthen the cultural heritage and sovereignty of communities. And I've seen that tomorrow you're going to have a whole session on that. So food science and technology is, in our view, a key dimension of integrated territorial governance. And talking of humanitarian context, you may think, well, this is very specific, but if we look at where you have most malnutrition and most hunger and most micronutrient deficiency, well, it's usually in crisis situations. So what happens in those crises? Well, emergency programs and projects focus on immediate and short-term assistance to save lives. So it's food aid. And what they inadvertently do, they increase dependency of people on aid. It is very important in humanitarian situations to manage food safety, quality, and ensure convenience of food. And that's where you come in too. But humanitarian activities must be human-centered and not technology-driven. It is essential to promote the resilience of local food systems to respond to current and future threats, stresses, and shocks. We've seen that with COVID-19. We see that regularly with conflicts. We could actually see humanitarian situation as an opportunity for reorienting food science and technology. Or I'm not saying it has to be a combination of different approaches, but we need to catch up on this territorial approach. For the moment, we have a global system which is very much economically driven, and we should start looking at a territorial food system that should be looking more at sustainability and resilience. The system we have now is mostly externally sourced. It ignores local assets. It depends on globalization. Locally sourced food science and technology would allow to make better use, more sustainable use of local biodiversity and better use of water and energy. The global food system often distorts local economies and undermines local livelihoods, in particular traditional livelihoods. If we had a territorial food system, it could combine to local economy and jobs. We are always talking of standardization and norms. If you want to go local, it's probably going to be more important to build upon indigenous knowledge and culture and bring in the science we know that will make these healthy and safe for people. And we often distort food habits and generate nutrition transition and diet-related diseases, whereas we should be contributing to sustainable diets. In the situation in COVID-19, we have seen that the present pandemic and its impact have reflected to a significant degree the distortion and vulnerability of present food systems. We know that food-related policies in the last decades, and I'm thinking of intensive factory farming, large-scale slaughterhouses, standardization, etc., have in fact actually enforced a shift from more human-sized structure to socially and environmentally unsustainable business models. And we also have seen with COVID that local food processing and storage, and I'm thinking in particular in India, would have been desperately needed to ensure territorial resilience and avoid waste. As a conclusion, dysfunctional policies and an excessive bias towards globalization and privatization in the last decades have privileged productivity gains to the detriment of inclusiveness and resilience. Food science and technology has de facto contributed involuntarily to diet-related diseases. The food science and technology community must engage in and be acknowledged as key actors of the sustainable food system debate at territorial, national, and global levels. Food science and technology can facilitate local access to sustainable, healthy diets, and in particular, complementary foods. It is essential to promote the resilience of local food systems to anticipate and respond to current and future stresses and shocks. And last but not least, people engaged in food processing and other post-harvest activities must have decent working conditions and wages, which is absolutely not the case right now. I thank you for your attention and we'll be happy to answer to any questions.
Thank you, Dr. Eagle. Thank you for the very wonderful and very informative presentation. Various important points which were discussed in this presentation, I will take up a few points out of it. She discussed on how to tackle hunger and what is the need of, there is a need of a supply-driven to demand-driven perspective. Second important factor which she discussed is she discussed about the sustainable health guidelines which is given by WHO and FAO, and how humanitarian crisis and food science and technology role in just overcoming those problems. He shared various documents links which all participants can visit after and for more information they can visit all those links. Thank you very much for such an informative discussion, Dr. Eagle. So next we move to our second presentation. Is it okay, Mandeep? Can we move to next presentation?
Yes, ma'am, please.
Okay, so second presentation is my presentation. So I'm just, give me a second. So screen is visible?
Yes, ma'am, screen is visible.
Okay. So the topic which I am going to present today in this international conference is Nutri-Cereal, basically millet, which is a 21st century smart food. So if millet crop of past is now being considered as a crop of future, and the role in attaining the sustainable development goal is the thing which make this millet or which make this Nutri-Cereal very, very popular these days. In mid-20th century, we all, means globally, we are tackling with population growth, where major concern was food-related issues, and especially developing countries where food security is alarming and dwelling. And our traditional practices and indigenous food system, which have potential to have sustainability in providing all the nutrients, they are sustainable, means they have a power to enhance dietary diversity. We keep on leaving all those behind, and we just move forward for and keep attaining certain new and innovative perspective in our dietary pattern. Millets are one of the oldest food grains which is known to human, and earlier evidences were found in Indus civilization, that is around 3000 BC. And in India, if I talk of millet, have been mentioned in some of the Yajurved texts, and thus indicating the consumption in the Indian Bronze Age around 4500 BC. So not going in BC, even up to 50 years ago, millet were the major food grains which are grown and which used to be consumed by Indian population, and they are considered as coarse grain and poor men food, and we used to consume it as a part of our diet, and it make our way of eating, and it is a part of our diet. So millet are the neglected ancient grain cereal which are reintroduced as 21st century smart food to build the global health society. So millets, which is reintroducing again to our society, the reason is we Indians, or what we call as globally, we are following the Western mode of development, and developing countries lost their gene pool for millet, particularly which was consumed by poor population due to various reasons like colonization, deviation from the cultural ecosystem, services, coupled by government policies which was taken up to just fulfill the nutrient food demand. Basically, we what we call as green revolution, rice and wheat production to provide the food security, which lead to the reduction of the cultivation of millet by around 40% and hence consumption. In 1970, millet contribute to around 20% of the food grain of Indians. Now we are reducing, means we are leaving it, and it reaches to around 4 to 6% of our diet. And if we check this slogan which I have given, let eat smart, grow smart, and stay smart. These type of slogans are important, and once if I remember, and if I'm not wrong, in 2000, early 2000, in Madhya Pradesh, a slogan was given in India, kudo kudki hatao and soyabeen lagao. And it was a popular slogan which was just given to encourage the farmers to produce oilseed crops. More than a decade after that, central and state government has come back to adopt the mission mode approach for promotion of this technical coarse grain again, and then introducing millet again as important aspect of your food. So basically in 2018, Ministry of Agriculture and Farmers Welfare, they took up this part as to be promoted for nutrition security. And in mid 2018, the Department of Agriculture along with Niti Aayog took several steps to bring millet within main fold, or what we call as a part of national security scheme. Again, to just make this millet as a very important aspect, 2023 is announced as International Year of Millet. That shows that how this particular nutri cereal is important for sustainability. Child nutrition is basically considered as a very sensitive indicator of overall level of food security and hunger. And undernutrition manifested itself usually in many ways: stunting, wasting, underweight. And as depicted in the slide, India ranked 102 in among 117 developing nations in the Global Hunger Index. 2019, which is a very much alarming sign. Again, various surveys which have been conducted as depicted here have shown very clearly that the level of anemia is increasing. There is bit reduction in percentage of stunting and wasting, but overweight, one type of malnutrition, it is increasing in our population, children 5 years of age. Along with these, 80% of the women in the reproductive age is also suffering from iron deficiency anemia. So we are facing two forms of malnutrition: micronutrient deficiencies and other macronutrient deficiencies, because of the various reasons, which is one of the way which needs solution. Our food culture today is very much defined by various factors, and some of the important factors, as we have discussed before also: urbanization, globalization, industrialization, westernization. They have altered the diet by reducing the consciousness about the nutrition both in the urban and the rural areas. And access to non-traditional food has increased, which basically basically due to the change in the processing. And as my previous speaker also quoted, this is because of changes in the technology and food techniques and food science development and all. So nutrition transition literally shift the dietary habit to convenience and more easily available food stuff, leading to changes in the dietary pattern, moving more to the high calorie food and leaving traditional practice which need attention. So the concern point are more from less or for more, starch to protein to healthy food, stress agriculture, secondary agriculture and specialty agriculture, food, nutrition, health, environment and empowerment is to be tackled together. One health means health of soil, plant, animal and human being together. And skill and youth in agriculture is also important aspect when we have to deal with the problem or sustainability or responsible diet. So I will skip this particular slide because we have gone through this definition before also. So a sustainable diet and why millet is considered as sustainable diet is important, because it is these small grains are full of nutrition. They are rich in all the nutrient. They provide food security. They have power to provide economic security, and along with this, they have a power to provide safety for various diseases. So millet is an approach for a sustainable agriculture and healthy world, and we can use these what do you call a pseudo millet or what do you call as coarse grain as a part of sustainable development goal fulfillment and achieving sustainable goal. So millet, if I talk in terms of sustainable goal development, millet is fulfilling various sustainable goal, that is SDG 2, 3, 12 and 13. So basic objective of this goal is to end up hunger, achieve food security, improve nutrition, to have sustainable agriculture, to increase the production, and millet is fulfilling, is an exception, and we can use it for fulfilling all these goals, and we can make it a part of our diet to fulfill all these things. And they have a power to provide us food within, means they can be harvested in 70 to 80 days, and they demand less maintenance in the when we talk of production. They provide climate resistant features, and millet can help in providing climate action. So basically these all these benefits or all these points make millet very important, and which can be used as a part of sustainable development goal and sustainable diet, or we can make it a part of our diet, because millet are very important and superior to major cereals when we are talking in terms of nutrition. So in terms of nutrition diversity, millet contain a very good amount of all the macro and micronutrient as compared to rice and wheat, as depicted in this particular slide. Average protein content of the millet is around 10 to 11%, except that of finger millet, which is reported to contain within the range of 4.76 to 11.7 gram per 100 gram. And it is also means the protein is of which we get from millet have a good amount of essential amino acids like methionine, valine, lysine, and if I talk of finger millet, the total amino acid is around 44.7%, and essential amino acid out of it is around 33, means out of 44.7, 33.9% of it is essential amino acid. And generally, I'm sorry, it is providing around 44.7% of the essential amino acid, and which is at par of 33.9% essential amino acid reference data, which is provided by FAO. Millet is also very much rich in fiber, both dietary and crude fiber, and the highest dietary fiber content, that is 38%, is in little millet, and around 37% is in kudo millet. Likewise, finger millet are a richer source of calcium, and the seeds have a good amount of calcium, around 300 milligram per 100 gram seed has a calcium content, and that is important. Likewise, various other millet are rich in iron, and they have a good amount of iron in them. So my point here is that millets are considered as very important source of nutrient, not only of the macros but micros. There are certain limiting amino acids which are present among millets also, but we can use them to substitute the amino acid with cereal and with other nutrients, so as good quality proteins can be incorporated. So I'm not asking to just reduce other cereals, but by adding millets to the diet, we can diversify our diet, and we can achieve a sustainable dietary pattern, and we can improve the nutritional demands of the population by incorporating them in our food. The problem with millet, if I talk, is bioavailability. High fiber content and presence of some anti-nutritional factors like phytate, tannins in millet, they generally affect the bioavailability of the mineral. And there are few studies which suggested that absorption of iron basically many times reduced because of these anti-nutritional factors, which is a concern factor when we are dealing with millet. But there are many ways and there are many studies which have been conducted in food sector by which we can reduce these anti-nutritional factor and make these important micronutrient available to the population. But there are many new venues which need more attention, and on which lot of research are required, like more studies are required to just test how to examine the bioavailability of nutrient, including your B vitamin and others, and how they are working in vivo models. In vitro, we have just find out various bioavailability aspects. Bioaccessibility has been tested, but how it is working in vivo is important aspect which need attention, which need research. Effect of physiological state like pregnancy, lactation, age, where nutritional status increases. It is also a point how it need investigation. This point is important. And absorption of vitamin and mineral trends to respond to body demand is also important when we are dealing with millets. So these are some of the, I have just tried to just point out various important health effect as quoted by various literature in various in vitro and animal studies which have been conducted. They have a power of losing weight, bone health, diabetes, lowering the blood cholesterol, hemoglobin status, anemia reduction, means they work for as anti-depressant and anti-insomnia, has a capacity as anti-anxiety, antimicrobial activities. They help in fighting against malnutrition, and many researchers have quoted that they have chemoprotective agents for basically many type of cancers. So this all benefit make these millet very important bundle which can be used for tackling various problems. Like cardiovascular, if I talk of, it elevates the level of endinopactin, which protect the cardiovascular tissues by inhibiting the pro-inflammatory agents and help in reduction. Likewise, there are many researches and many data available online, which and various studies are going on, which is working on millet and how, at what mode and mechanism of action against a particular disease is working. So that is important when we are talking. So to reduce the bioavailability problem of millet, we can use technology, we can use the processes to just in various ways, like these are the various processes like soaking, malting, milling, grinding, fermenting, cooking, puffing, roasting. These all processes help us in reducing various anti-nutritional factors, and they help in improving the bioavailability of various minerals. They also improve the digestibility and nutritive values, and generally provide sensory attributes to the product. And by using all these processes, we can convert millet to various food products. And there are many food products which are traditionally being used and prepared in our household, but the need is to convert it into in a form which is convenient, easily available, so as it can reach the from rural household to market, and to our markets, and to means we need a demand for all those convenient foodstuffs. So this is the gist of what we have discussed. Millet, which is very much rich in nutrient, which is low cost, which is good, sustainable factors are there. This is good for environment. This is good for production also. So we need to use certain processing techniques which help us to improve the functional changes by increasing the bioavailability of minerals, by increasing the antioxidant potential. It increases the nutritional digestibility, and it helps in reducing anti-nutritional factors. It makes changes in various nutritional content, and these are the various products which are worked upon and on which are prepared in our laboratory by incorporating or by value addition. We have prepared all these products in our lab, but the point is we need to take it to population awareness, and extension of the same is very much required. So these are the points which can bring back the glory of this millet or nutri-cereal by enhancement of production or productivity, by promotion of nutrition and health benefits, value addition, processing and recipe development, entrepreneurship, startups, collective development, awareness creation, branding, labeling, promotion, policy involvement for the mainstreaming, and international outreach that is very much demanded for these millets. And India can be a part, or India can play a very important role in making this possible. So strategies can be used to increase the demand. Various strategies are the production and consumption of millet must be a part of policy making, like wheat and rice. Once we make it as a policy decision, likewise millet, and government is working on it. Research constant means a particular focused research is very much essential for validating advantage of millet as functional food. Traditional and non-traditional, ready-to-use, convenient, easily available food option must be provided, and they must be made a part of complementary feeding programs, which are the part which tackle nutrition of low-income group in our country. Those programs should adopt these food stuff for generally, like ICDS means it can be a part of ICDS feeding program. So millet-based complementary foods, khichdi, upma, roti, etc., can be a part of ICDS programs. Then R&D on millet as food security may be strengthened along with food security, nutritional security aspect, and functionality of it should be studied. Commercialization and promoting millet-based processing, ready-to-eat snacks, convenient food should be made as a public-private partnership, and it should be taken up further to create the demand and to reach the masses. And awareness regarding nutrition, health, and environment and advantage may be created as a strategy for communication and for advertisement. All these points will help us to make or to bring back the cereal or millet which was very much used in the past and which become a 21st century smart food, and we can make it a part which will help us to leverage our traditional ecosystem or traditional ecological knowledge, and we can use these indigenous food to achieve SDGs and beyond. The way is we have to incentivize the millet cultivation. We have to make a farm gate processing, value chain, value addition, marketing linkage, and all these things can be bring upon at one place, focused, and all these things help us to bring back millet to our diet, and which make us responsible consumer, which make us to eat this important millet, which we are just not doing. The point is we have to go local, we have to go for indigenous traditional food habits if we want to have nutritional security and sustainability in our diet. Thank you.
Thank you, ma'am, for such an enlightening and overwhelming session. Now I would like to call our next speaker, Dr. Joe M. Registon. Dr. Registon is a professor emeritus of food science and head of the Cornell University Kosher and Halal Food Initiative College of Agriculture and Life Sciences, CALS, and an adjunct professor in veterinary medicine and Jewish studies. He is an adjunct professor of food science of both Kansas State University and Shanghai Mai University in Thailand. He is a distinguished foreign expert and Xiangnan University in Wuxi, China. His BA and MS are from Cornell, and his PhD in biophysics is from Brandeis University. Dr. Registon has received 2 awards from Cornell University, efforts to promote multicultural diversity and outstanding accomplishment in science and public policy, and has received IFT's Elizabeth Steer Humanitarian, Burla International, and Carl Feller's Career Service Awards. He co-founded and edited IFT's Religious and Ethnic Food Division newsletters. An IFT fellow, Dr. Registon served on IFT's executive committee and was IFT's first congressional science fellow. He is a guest fellow of New Zealand's IFSTs and is co-editor emeritus of Food Biosciences and founding co-editor, the first English peer-reviewed Chinese food science journal. I welcome you, sir, for the session.
Thank you, Sunarayan. I hope people can see my slides.
Yes, sir.
I thank
It's visible, sir.
Thank you. I appreciate the invitation. I'm speaking from America, and I must admit my talk is a little bit more on sustainability and less on diet because it was rescheduled because otherwise I'd be speaking in the middle of the night. And my area of interest is total utilization of fishery byproducts as an example of why it is so important that we think holistically about all of our foods. We just heard about millet. What do we do with the other parts of plants and animals that are not what we're traditionally eating? And that's been my focus. So my mantra, as I call it, is to think about the pork industry, the beef industry, but the pork industry in particular is very proud of the fact that they use everything from the pig but the oink. Because fish are quiet, we have to do better. And that has been the thrust of a large part of my program, is to see how do we use all of these materials so that wild catch fisheries and aquaculture are optimized as, again, an example for those of you not in fisheries of what needs to be done. So the simple answer in the past has been to simply dump at sea or in the river or to landfill. But this is not an acceptable environmental solution at this point. So what are some of the solutions? One needs to start with whatever commodity one's dealing with a waste analysis. What is there? What do you have to work with that you're not selling? Can you do the right thing? You cannot do the right thing if you do not know what you have. And so you need to think about seasonality, daily fluctuation. And one of the things that makes fish plants a little different from other plants is the mix of species changes as the wild catch changes because of the movement of the fish in the oceans and the question of what the boats are harvesting and the techniques used. So fisheries are particularly complex, but also generate a large quantity of waste. So what is the total quantity daily, weekly, yearly? What are the different species? What is the quality of the waste? And that's another consideration for all of these products. Can you get a high quality, or are you getting materials near spoilage? And of course, fish are tremendously nutritional, but they do spoil very easily. And The other question is when you have something that has parts, animals are made up of many different parts, can you easily obtain the separate parts? The point being that in fisheries, the waste can be up to 70% when you fillet fish for Western style markets. On the other hand, many people work with fish, all they remove are the intestines before they sell and eat it. And some of that waste then is generated at the home, and that is more difficult to capture. So my emphasis has been on working with plants, fish plant processing plants, to optimize total utilization. We've put in efforts, we've put in energy to harvest, or we've fed these animals in aquaculture. We need to recover this. Now, the traditional products are rendering. The animal agricultural industry, generally speaking, optimizes its waste by rendering. It creates something called fish meal with whole fish, and that's a whole directed fishery. And there are people who argue whether this is good or bad, and I'll leave that for another day. There also is a lot of whitefish meal, which in many cases is mostly bone meal, very high in bone, but that means it is, as we heard, the importance of calcium and iron in the previous talk. These tend to be very high in calcium and iron. And there is fish oil, which has various uses. It is a very highly unsaturated acid oil, so it has handling problems, but it's also a very healthy oil with the DHAs and EPAs. Just as a comparison, fish do have a much higher potential waste on average going to rendering. But again, all of those animals where things are going to rendering, can some of that be intercepted for both higher value and better value for consumers? So what are the parts of the fish that I want to look at and give some ideas? And this again is to think holistically about the whole animal and what we're doing. And this could be a whole plant. Can we use things from the plant just as easily? Guts and viscera, head, skin, scale, bones, and something I will give a picture of, the frames, which is the backbone after you fillet a fish, which actually contains a great deal of meat. And then as we process for aesthetics, which one can argue pro and con, we trim fish, and we often have some very high quality meat that is just small pieces for that purpose. If we can only get the total byproduct, we can make something called silage, which is an acid hydrolysis that includes dissolving the bones. Formic acid seems to be the acid of choice if we want to feed it to animals. Interestingly enough, if one wants to use an organic fertilizer, those are often can be animal hydrolysates, and one therefore has to pay attention to the nitrogen, phosphorus, and potassium. One of the interesting uses as animal and plants interact, and I think as somebody who favors a balance, but it recognizes the importance of animal agriculture, is the use of things like foliar applications, household plants, and even commercial plants, using these animal hydrolysates as part of the nutrition for plants. For low value usage, but often where you don't have access to a fish meal plant and you don't want to dump at sea or landfill, compost at least retains some of the organic value. It uses both high nitrogen and high carbon materials, so it can also help with plant issues. And we actually have used it in an unusual situation where we had highly contaminated salmon from one of our Great Lakes in America. And we composted it and used that compost to help with growing what for Christians is the holiday of Christmas, where there is a habit, a tradition of using trees. And again, because those are non-edibles, we could use compost that is contaminated. And again, the idea being thinking through holistically our agricultural picture. The Korean market, we learned, will eat the stomachs of certain fish, and that's a potentially high value product. So you also need to be culturally sensitive, and I would argue at times need to find the global opportunities that may not exist locally. There are many enzymes in all of these systems. Some may have value in food production and other industries. One of the differences with marine enzymes is they tend to be less heat tolerant than mammalian. They denature at lower temperatures. That means you don't have to heat things as high, but it also means that in some cases at room temperature, enzymes are reacting where the mammalian enzyme actually needs heat before it starts reacting. So again, we have to look at what are our opportunities, what do we know? And again, just to remind people again, animals are complicated and we see many internal parts that need to be dealt with. And each of them may or may not have opportunities to recover them in a cost-efficient manner. And that again goes back to an issue that I tend to have a different view of, which is the economies of scale. Economics are important. People have to be able to afford food, and part of what we have to balance is that sustainable diet with an economically viable system. Heads can be used to obtain cheek meat. They can be used whole to make soup. And obviously, in some cultures, they are considered a delicacy. I come from a culture, the United States, where nobody wants to touch them except a few of our ethnic minorities. And so we have different challenges. The skin can be used to make leather. It's a beautiful leather, but so far has had limited usage. We need possibly, there are opportunities there. It is a source of collagen, which on heating becomes gelatin, which is a major food ingredient. And I've worked recently in the last part of my career, worked on fish gelatins, partly to serve cultural needs. People who are Jewish and keep kosher, people who are Muslim and keep halal, and many of the Eastern religions are or encourage pesca-vegetarianism. So those are opportunities for fish gelatin and collagen. There are people, we learned in some countries, worried about mammalian diseases in foods like gelatin. There's no evidence of it, but consumers sometimes get interesting ideas, and we have found that there are markets there. Again, because it's coming from fish, it has a lower melting temperature, which is good or bad depending on your food product and what you're trying to do. Interestingly enough, from a flavor profile point of view, we have shown that in fact it has a better flavor profile. Just to again highlight what is an area of specialization for me is in the United States, 40% of packaged products are kosher, even though the Jewish population of this country is less than 2%. On the other hand, globally, a quarter of the world's population is Muslim and does not eat pork. Many of the Eastern religions, including as I understand it, the Hindu religion, which is the major religion of India, have issues with particularly things derived from cattle. And there are those who wish to avoid beef. So again, the marine world offers some alternatives. Scales can also be used for collagen or gelatin preparation. We are seeing in Asia an increase in interest in these products. Not unfortunately, but somewhat discouraging in the sense that I'm not always convinced it's the most important thing we should be doing, is it's being used heavily in the cosmetics area, although there does seem to be some research suggesting that collagen gelatin actually does good things from the skin in terms of health, in addition to cosmetics. Scale gelatin is very different from skin gelatin, and one needs to remove the calcium from the scales as part of the preparation of gelatin from scales. Now, again, that means we have produced a calcium stream. Can we use that stream for nutritional purposes? Another product of scales is pearl essence, which is used to make natural women's nail polish, the cosmetic issue. It's been replaced by synthetic compounds, but the question is, could we return? If there's this move towards natural, which we're seeing in many markets, is there a new opportunity to use fish scales as a higher value total utilization product? This is a machine called a mechanical deboner. It is used throughout animal agriculture to remove flesh from bones and skin. Again, works differently with the different commodities. It has led to a great deal of the processed products that folks are using with these meat byproducts, and we can argue the pluses and minuses, but to the extent that they represent recovery of material that would have been wasted, they are increasing the food supply, which is part of sustainability, is to have enough food. And that gives you a fish, what I call a fish hamburger. Because it is running the bones through it, you get blood. Blood is good for iron and also has meat-like flavor. And so that is both good and bad. Surimi, which is used to make the imitation crab, shrimp, and lobster, they spend a lot of time and effort washing the mince. I am a proponent of using the mince directly. The iron needs to be kept in the product. The washing is a waste of resources, and the flavor of the product allows you to make more meat-like products. And we have done a lot of work in that area. And this is the typical surimi type of product that you will see in the marketplace, generally as a salad. The bones can be used in the canned products as a high level of total absorbable calcium. A can of sardines, 3.5 ounce can, has as much calcium and is absorbable as a glass of milk, an 8-ounce glass of cow's milk. The bone itself can be used as a source of gelatin. The yields and process are harsher than for skin, and so at this point, that is not a viable economic thing to do. This is the frames that I talked about. When you fillet a fish, you get the bone rack, and between all those bones is a great deal of flesh. About 50% of a bone rack with the head off, that's why the horizontal line, will be meat. So the recovery from what is this material that normally goes to whitefish meal and has a very low value, 50% of it is human edible and high iron product. So why aren't we using this? And one of the goals that I never got to was to look at the waste stream from the deboner. Could we use that for gelatin, collagen, or other materials, given that it has, again, a lot of bone? Trim can be used to make reformed fish fillets. Now that's, again, high technology, but it essentially, using an enzyme called transglutaminase, can increase our availability of fish. And for some of the institutional users, these reformed fish have the additional benefit of being uniform size for portion control, which is also important in Western diets where overweight is ironically the major disease of modern Western society. So those are some of the ways I look at using fish as a total utilization and maximizing the sustainability of our food supply is by emphasizing byproducts. I appreciate the importance of studying the main products, but where the real breakthroughs and increase in food supply is going to coming from is making sure we have less spoilage and more total usage. And again, if anybody is interested, you have my email. I'm happy to send you this PowerPoint. I have a longer version of this that I'm happy to send. And we actually have a cookbook that we wrote some years ago using minced and canned fish. So thank you for your attention. Thank you for letting me be part of this exciting program. And if anybody has any quick questions, I would be happy to try to answer them.
Thank you, Dr. Reginston, for excellent presentation. The major points which are discussed in his presentation is about fish waste and what is the need of waste audit. Various questions need to be answered during auditing. And he talked about fish rendering products, major component of fish waste. Thank you, sir, for this wonderful session, for this wonderful talk. Any queries? No, I don't receive any question yet. If any presenter, if any participants has any query, please write in the chat box. We can ask a concerned person and get your answers. Thank you. Yes, Sunan, over to you.
Yes, ma'am. I can see that there is no questions. Thank you, speakers, for such an enlightening session on the responsible consumptions and sustainable diets. Now we move towards the second part of the session, that is the oral presentation. All the participants are given 8 to 10 minutes for their presentation. And after that, then we'll take up the question answer round after each presentation. I request all the participants to kindly adhere to the timelines. And the first presenter of the evening for this session is Abhay Tiwari, Satyavati Sharma, Vasudha Sharma, Rupesh Kumar Srivastav from Indian Institute of Technology, New Delhi.
Can you see the presentation?
Yes, your PPT is visible. Can you put it on slideshow mode?
Yeah, sure. Can I start, ma'am?
Yes, Abhay, you can go ahead.
So a very good evening to one and all the dignitaries present here. First of all, I thank all the organizing committee of the AMIFOST 2020 for giving me this opportunity to present my research topic, which is on combinatorial benefits of probiotic and nutraceutical enriched mushroom for improving health. So I'll start my presentation with the stating the sustainable development goal, which are given by the UN nations, and for among the 17 SDs, which are given by the United Nations, one is the good health and well-being, and how you can focus on the good health, and I will be summing up in the coming presentation. So mushroom cultivations offers a highly efficient method of the biodegradation of the lignocellulosic rich material, let's say paddy straw and wheat straw. And in the cultural practice also, the waste utilization has been done by using the mushroom cultivation. So here I am just presenting the lots of demand, and in the past year has been there for the protein source, or especially for the vegans who avoid the animal source of the protein or the other nutrients or the nutraceutical which have the animal origin. So mushroom can fit well there, and it can also supply or bridge the demands of all the protein requirement and the other as the other vitamin source, particularly the vitamin D, because vitamin D source is majorly from the animal based source. Now, coming on to the main highlights of the presentation, why I have selected the mushrooms and the probiotic. As we know, mushrooms are generally regarded as safe, and they are having enormous potential towards the health benefit, like say it having an immunity enhancer, it has in digestion, and numbers of characteristics are there for the probiotics. Now, coming on to the mushrooms, mushroom also have enormous potential in terms of the bioactive which they are having. So particularly to talk about mushroom, they have the dietary fibers, they have the lipids, they have the polysaccharides, the beta glucans, which is acting as a prebiotic for the probiotic. That's why I am stating here that the combinatorial effect of both can have a win-win situation in gaining their nutraceutical advantages, or to develop a nutraceutical based product by combining both the products. So coming on the next, as we know, the nutraceutical is defined as a food part or that which provides the body with a medical or a health benefit, including the prevention of the treatment of disease. And probiotics are the live microorganism which, upon ingestion in sufficient quantity, which exert a special benefit beyond the basic nutrition. And prebiotics are the food for the probiotics. Now, the disease which I am targeting is the osteoporosis. As we know, the osteoporosis is a second global health problem after the cardiovascular disease. And after, it's like a very general, after the menopause cycle, like after the age of 40 or the 45 plus, every woman suffers from the estrogen deficiency, which leads ultimately leads to the porosis in the bone. So it's a very deadly combination. And women above 50 years of the age and more prone towards the osteoarthritis than the men. And according to the stat, 1 out of 8 men and 1 out of 3 women are affected by the osteoporosis. So it's a huge data. So I have taken just a one literature just to show the quantum of the, so the importance of the osteoporosis is that you can see even in the Asia is around 71.4% are the people who are having vitamin D deficiency, which is ultimately leading to the these kinds of disease. Now, I'm not going to the detail of the how the vitamin D take place, absorption take place in the body. First, we ingest from the food source, and or either it will get converted from probiotic to the from pro vitamin to the pre-vitamin D, and in this skin on the exposure of the UVB. So we have two enzymes present in the liver and the kidney, which ultimately convert the bioactive form of the this vitamin D, and then various it is goes to the different part of the body, and then the metabolic functions where it required, it get utilized. Now, coming on to the next thing which is important is that the role of the calcium and the vitamin. We will know that the calcium is also responsible, apart from calcium, magnesium and potassium are also necessary for the absorption of the vitamin D and all the metabolic cycle which occur after ingestion of the vitamin D enriched food. So here I want to highlight that the mushrooms are rich source of all the minerals which are present here. So combining this, we can achieve the mineral consumption as well as the vitamin D in that. Now, as I stated, the vitamin D deficiency can lead to the consequence of calcium absorption. Parathyroid hormone will be imbalanced, and then bone mineral density will decrease, and ultimately it will lead to the risk factor there. Then now, first of all, I want to mention that the mushroom is a highly perishable commodity. So because of the polyphenol oxidase which are present in the outer periphery, because the browning of the mushroom take place. So how to preserve that thing? So we can utilize the post-harvest technique. I am utilizing the UV radiation so that the perishability of the compound can be reduced, or the augmentation of the nutraceutical can be done in the mushrooms utilizing UV radiation. So here I am just depicting a photographical representation that how the photobiological reaction of the vitamin D occurs once the UVB irradiation incident on the surface of the mushroom. So here particularly different type of mushroom species is there. We have the provitamin, the initial molecule, let's say ergosterol, 7-dihydrocholesterol, or 22-23 dihydroergosterol, which is also known as provitamin D, which is ultimately getting converted into the pre-vitamin component once it is exposed to the UV light, and then it is ultimately converting by the thermal rearrangement into the vitamin D, the ergocalciferol, the dihydroco, and the various other photobiological byproducts are also formed. And then these are the the inactive form which we take in the diet. Now, in our technology, we have developed a UV chamber which is made of an acrylic chamber, which consists of two tubes, UV tubes, and which having a narrow band of 280 to 315 nanometers. And we have exposed the mushroom. Here I want to mention that the conversion of mushroom vitamin D in pre-vitamin to provitamin is very well known, and people have done it using in the fresh mushroom. But here I have I have established the fact that even the conversion of vitamin D can take place in the dried fruiting body of the mushroom. So here I have taken the lentimicetodes, and then the UV radiation I have done for the 10 minutes. And SPLC studies depicted that the after UV radiation, the amount was around 24.97 microgram per gram in the control sample, and then ultimately it is getting converted. Almost twofold increase is there in the irradiated sample, that is 49.31. Apart from the UV radiation, this vitamin D enhancement, several other parameters are also getting enhanced. That's a total phenol and the total phenolic acid are also getting increased, because it having some initial moisture level in that, and then because of the activity of the chalcone synthase and various other enzymes which are involved in the flavonoid pathway, the stress is created, and because of that the increase of these content is there. And apart from that, and the multimeric protein, phenolic compound are getting degraded into the monomeric compound, and the dissolvability of the compound and the extractability of the compound is getting increased after exposure to the UVB. Now, I have done the different radical scavenging assay, that is ABTS and DBPS. Similar trend was observed for the irradiated sample. Now coming on to the FRAP and the beta glucan, and here I am emphasizing again because beta glucan is a known prebiotic component for the probiotics.
So Abhay, sorry for interruption. Please try to wind up
I'm just winding up. So beta glucan can act as a modulator for proliferation of the growth of probiotics. So ergothioneine and the amino acid content I have just explored. And then the various other GCMS metabolite I have identified using the GCMS. So they are also getting enhanced. Now coming on to the probiotic, the initial screening of the lactobacillus has been done on the MRS, and then plate studies by using only this powder, vitamin D enriched powder, have been used to proliferate the growth. Then different percentage of the mushroom powder have been taken to proliferate the LA, and then you can see the significant enhances. Even in the same studies, you can see there is a presence of these LA bacteria is there. Now coming under the comparative study, yes, that UV radiated prebiotic produced better growth than the control one. Now coming on to the in vivo studies, first we developed the in vivo model, that is osteoporotic mice model with the help of AIMS. And we have administrated the group combinatorial powder in the osteoporotic mice. And then after 6 weeks, we have sacrificed it for the bone analysis. So bone analysis depicted that the bone histomorphic analysis clearly depicted that OVX and the SAM, osteoporosis have been induced. And then after treatment, you can see the LA group, and as well compared with the OVX group, the bone is getting improved. And when the combination is given, even that you can see the improvement in the bone. Even for the bone mineral density, the combination has proven that bone mineral density has been increased as compared to the control. Now, coming on to the next slide. So, ultimately, the technology you have developed is not delivered to the farmers or the processors. So, for that, I have just visited the Dholana area of the UP, where the mushroom global and the self-help group have been treated for the UVB utilization and to augment the vitamin D in that, and how can they use the traditional probiotic product, and they can use the this powder in their probiotic formulation and can achieve a good bone health. Now, I'm concluding that this present studies provide the sustainable strategy to improve the nutraceutical potential of the dried mushroom, let's say in terms of vitamin D and the TPC and the TFC. And also this methodology which I have proposed is also now approved by all the FDA and the EFSA or the World Organization. And also our study also provide that the symbiotic administration could improve the osteoporotic bone in the mice model. So the future prospecting that the strain which I have taken is the Lactobacillus acidophilus. So other probiotic strain like say bifidobacterium or the yeast can also be explored with the mushroom, so that that can be used to fortify the food and can formulation can be developed. So to sum up the entire presentation, I have just given the graphical abstract. So first I have just irradiated the mushroom, then I have proliferated the mushroom using the proliferated the probiotic using the mushroom powder, and then I have developed a formulation which having the active component of the nutraceuticals along with the probiotics. So I am the review part, the basic which the idea is which we have proposed is published in some in the IFST journal, and these are some publication from our group. And I wholeheartedly thank my three supervisors, Professor Rupesh from EAMS, Professor Vasudha from Jamia Hamdard, and my guide, Professor Satyavati Sharma from IIT Delhi, who has helped me to conduct this experimentation. Thank you.
Any questions? We'll take up the questions in another 2 minutes. Any questions? Okay, thank you, Abe. There has been no questions. Okay.
Thanks a lot.
Now we move towards to the second presentation from A. Silva, L. Cassani, Lorenco Lopez, C. Grosso, J. Simil Gandhara, and M. A. Prieto from University of Vigo, Spain. Your slides are visible. You can go ahead with the presentation.
Voice is not there.
Now you can hear me?
Yes, yes, we can hear you. Can you please switch on your camera as well?
I'm sorry. Okay.
It's okay.
Hello. Thank you all for the opportunity of presenting a part of the ongoing research work entitled Fucospiralis as a source of nutraceuticals. Macroalgae have been over the centuries an important part of the human diet, especially in Asian countries. However, consumers' appetite for natural, safer, and more diverse food sources are led to the increased interest in algae as food or food ingredients in Western countries. Furthermore, it's well known to the scientific community that marine organisms, especially algae, have a potential source of compounds with very important properties such as antioxidants, antibacterial, anti-inflammatory, anti-cancer, anti-diabetic, neuroprotective, among others. The scientific research on the theme is leading to the development of functional foods, pharmaceutical formulations, and cosmetics. Some of the most important bioactive molecules present in brown algae are sulfonated polysaccharides, pigments, and phenolic compounds. Fucus spiralis was selected among the group of brown algae collected from the Iberian Peninsula coastline, due to have shown promising characteristics in previous work. This algae is low fat, high fiber content, an edible macroalga that grows in the temperate climates in the eastern coast of Europe and in the North Atlantic coast of America. Some experimental data. The extraction solvent was selected out of several options as shown in the table. The best performer solvents were ethanol, acetone, and ethyl acetate. The choice fell on the ethanol because it gives the best yield, but mostly because it's non-toxic and suitable for food and pharmaceutical industries. Among the multiplicity of extraction techniques, microwave-assisted extraction was selected because it's time saver, has a low solvent consumption, and gives good extraction yields. The extraction parameters were optimized in a related work by response surface methodology, where the target functions were the yield, total phenolic compounds, and the antioxidant capacity. Some experimental remarks. Nitro oxide is an important intracellular and extracellular signal substance. The ability of Fucus spiralis extract as a nitric oxide scavenger was evaluated by deazotation using grease reagent. Concerning the reactive oxygen species, superoxide was determined by the NAD-PMS system, hydroxyl radical by the salicylic method, and hydrogen peroxide was measured by measuring molecule decay at 230 nanometers. The acetylcholinesterase and butyrylcholinesterase inhibition activity was assessed by the methodology described by Elman and co-workers, and monoamine oxidase A and B scavenger abilities was determined by measuring the formation for hydroquinone at 343 nanometers. Antimicrobial activity was performed by plate diffusion method. Oxidative stress is defined as an imbalance between the production of reactive oxygen species that causes damage to lipids, proteins, and DNA. Oxidative stress has been linked to several pathologies. Our results show that Fucus spiralis extract was capable to scavenge the reactive oxygen and nitrogen species in a dose-dependent way. Moreover, it proves to be more efficient than the reference substance for all tested species except for the hydroxyl radical. Alzheimer's disease is a progressive neurodegenerative disorder of the older population. The only symptomatic treatment proven effect to date is the use of cholinesterase inhibitors. The Fucus spiralis extract tested showed to have a moderate effect in the inhibition of the two cholinesterase enzymes. It is important to highlight the performance of the crude extract inhibition of MAO-B. The concentration of 1 milligram by milliliter led to 70% inhibition rate. This enzyme is crucial in the management of the Parkinson's disease, and both MAO-A and MAO-B also has an effect in chronic depression. The extract under study was also assessed for its antimicrobial activity against 6 different microorganisms. Fucus spiralis was capable of interfering with the growth of 3 of them: Bacillus cereus, Staphylococcus aureus, and Salmonella enteritidis. Fucus spiralis extract has a notable antioxidant capacity and could be useful in the management of the oxidant stress The extract show the capacity to inhibit the growth of some foodborne pathogens, and focus spirals extract will be helpful in the management of elderly associated brain disorders. In future work, we intend to pinpoint the molecules responsible for the bioeffects observed and to proceed with cell line studies in the effectivity of focus spiralis on the oxidant stress. This is the founding of our program. I would like to invite you all to come see us in Porto, and thank you for your attention.
Thank you, Ms. Arora, for the presentation. Now the session is open for questions. Does anyone have any question? So we can take up.
Yeah. I have one question here.
Yes, sir.
Can this algae, the extract of this algae, can we use it for food preservation? Will it extend the shelf life with respect to the microbial growth? If the extract is used in food processing or as a one treatment, if it is given to some food product, will it extend the shelf life?
I will, if I understand correctly the question, you are asking if this extract could be useful as preventing spoilage of food by the microorganisms. I believe so, but it's not very effective. The presence of this extract will decrease the velocity of the spoilage, but I don't believe that will resolve the problem.
No, as a biopreservative. So I want to use it as a biopreservative to inhibit the growth of some targeted organism in a kind of any type of the food. Will it work?
I don't know. Really depends. This in my experience, the effectivity of these extracts are highly dependent on the extraction technique.
Okay.
So it needs to be optimized for each function. Okay, but I believe that he has the potential to work, but further studies should be made.
Okay. Okay. How it is in the taste profile? Is it acceptable? Taste the extract. If I was, will it affect the sensory profile of the product?
I don't understand the question. I'm sorry.
Sensory, that is the acceptability, human acceptability. Acceptability as a taste. Suppose if I add to certain extent of this algae as such or as a extract in my food, will it affect the acceptability as a food?
Okay, if I understand the question correctly, you are asking if this algae will be accepted as a common ingredient in food. Maybe in Europe is not so common, but I believe that as the consumers are always asking for more natural products, different products and safer products, he has very good chances to be a future important ingredient in the Eastern cuisine. Yes.
Okay. Thank you. Thank you very much.
Okay. Thank you.
Thank you, Ms. Maria Arora, for answering to all the questions to our HOI, sir. Now we move on with the next presentation for the session from M.A. Adamu, J.K. Ikaya, Camila Rodrigues, Jane S.R. Combria, Cesar A.S. De Silva from Kano University of Science and Technology, Nigeria. Can we have your presentations?
Presenter is there in our session.
I can't, sir. I can't see their names. So shall we move towards the next presenter? And if they have joined, then we'll make an announcement afterwards.
Yes, yeah. I think we can start with the next one.
Yes.
Okay, ma'am. Okay. So we move on to the last presentation for the session from Ms. Jyoti and Dr. Shakun Tyagi from Mewar University, India.
Yes, good evening, everyone.
Ms. Jyoti, can you switch on your camera as well?
Oh, madam, actually, I'm on the way.
Okay, ma'am. Okay, then you can go ahead with the presentation.
Yeah, sure. Good evening, everyone. Here I'm present my topic, assessment of nutritional status and relevant. Next, I'm moving to the background of the topic. The importance of measuring body composition has increased due to the need to evaluate changes in the nutritional status, which can affect body reserve differentially. Subjects can gain body fat, fat-free mass, cellular mass component as a result of overeating and are due to sedentary lifestyles. These changes can only be detected by using valid body composition technique. And one of the most important aspects of health of individual is their nutritional status, which can be defined by as a result of interaction of body composition, energy balance, and as well as body functionality. In the present study, it is mainly focused on the role of dietary factors and physical activity level to alter the body composition of working women. Next, I'm moving to the methodology of the topic. In the present study, I have selected number of respondents were 100, and the women I have selected which were non-pregnant, non-hypertensive, non-diabetic, and the age group is 25 to 45 years old. The location of study was district Ghaziabad, and questionnaire interview method was used as a tool for data collection. Various questions have been asked regarding the general profile of respondent, in which I have asked the personal questions and family profile related questions and their health profile related questions. Next section I have in my questionnaire is anthropometry, in which I have asked their weight, their height, and then have computed their BMI. And next section I have asked their diet intake pattern, in which I have asked a question related to 24 hours dietary recall method and their food consumption frequency pattern. And next section I have asked about the physical activity pattern, in which I have computed their activity by factorial method, and then I have computed their PL and BMR rate. Then data have tabulated and various statistical analysis I have used for my. Next, I'm moving to the result and discussions, in which table 1 shows about the height, weight, and BMI of the selected subjects. The table shows that the majority of the respondents, that is 67%, were ranges normal, and 13% of respondents were underweight, 18% of respondents were obese class 1, and 2% of respondents were obese class 2. Next table shows that the mean and standard deviation of BMR, physical activity level, and total energy expenditure of respondents, in which BMR shows that 1302.3 with the variance of 88.11, PL is 384.8 with the variance of 3.078, and total energy expenditure is 2101 and 3.6 with the variance of 110.40. Thank you.
So Nan, yes, ma'am. Yes, ma'am. The session is open for questions. Does have any question? Please write in the chat box. No questions, ma'am. Okay. So we are done with for this session's presentation. Now,
so Nan, we can invite again that person who missed.
Yes, sir. Yes, sir. Okay. So A.M.A. Adamu, J.K. Ikaya, Camilla Rodriguez, James S.R. Coimbra, Cesar A.S. De Silva from Kano University of Science and Technology, Nigeria. Have you joined the session? Then you can start presenting.
Joined the session in the beginning, but I think there is some technical glitch, so they're not right now in the session.
Yes,
so I have just sent the email to, yes, I have sent the email urgent message to them. I think we can wait for 2 minutes, then we can
okay,
take the decision. Yes.
So all the presentations over? for the oral presentation?
Yes, sir. Yes, sir. They are the only one left.
Till the time they join the session. So I have one question from Abhay Tiwari. Mr. Tiwari, are you there in the session?
Yes, ma'am. Yes, ma'am.
Yes. Your work is very beautiful. You have worked upon the major deficiencies of vitamin D. I think every Indian nowadays is suffering from the same deficiency.
Yes,
So,
ma'am.
but my question is, have you tried with the wild edible mushrooms also?
Yes, ma'am. Actually, in the group.
Abhay, you are muted.
Yes, ma'am. In the research group, we have taken some wild species of subspecies of the Calocybia indica, which grows under the Odisha state, and some of the boletus mushroom also. And we have successful in developing the strategies for the fruiting body production as well as the mycelia production in the large scale production. So since mushrooms are highly season dependent, so just to overcome that fact, we are also producing the mycelia of that. So one project is that we have granted from the MFPI, under which we have just optimized the process of the production of mushroom mycelia. And in that, we are augmenting vitamin D as well as the vitamin B12. Both are nowadays having a very big problem, non-animal based, particularly for the vegan and for the vegetarians who avoid the use of vitamin B12 and the vitamin D from non-animals, from animal source. So we have developed that technique also, and we have given the technology to one of the company in the Noida. Ma'am, you are muted.
Ma'am, you are muted.
Try for other varieties as well. Yes. Thank you, because I think now everyone has to tackle the deficiency of vitamin B12 and vitamin D. I think
Yes,
Dr. Karna can she's the right person who can comment
yes,
on
ma'am.
these two things.
Yes, ma'am. Yes, ma'am. It's my opportunity to present my work in front of Karna, ma'am. I've listened in some of the occasions of the other various platforms also. So yes, it's my pleasure. And thank you for inviting me and accepting my abstract for the oral presentation.
So vitamin D deficiency and osteoporosis is a problem which needs such type of solution. Very good work, Abhay.
Thank you, ma'am. Thanks a lot.
Okay, ma'am. We have been joined by Mohammad Alhaji Adamu. Mr. Adamu, I think you can start with your presentation.
Mr. Adamu, if there is any problem, then please ask the IT team. They'll be helping you. Or you can share the presentation in the conference ID, so we can share the your presentation.
Yes, you can hear. I think I sent presentation yesterday. I think you can hear.
Just give me 1 minute then. Just give me 1 minute then. So Dr. Sunan, I'll be sharing his presentation. Just give me 1 minute.
Okay, ma'am. Okay. Okay, Mr. Adamu, I think you can start with your presentation. And on your behalf, ma'am will keep on changing the slides.
Okay, thank you very much. Good morning, everyone. I am Muhammad Alhaji Adamu from Kano University of Science and Technology, Ujul, Department of Food Science and Technology. I'm here and I was being opportune to present my abstract on the topic proximate composition and body weight of the albino rat profile feed with gurasa bread as affected by the addition of soya beans. Okay, gurasa is an indigenous bread that is generally produced in the northern part of Nigeria, particularly Kano, and in Arabian, India, they call it gurasa. As I said initially, I turned my name from Nigeria, Kano University of Science and Technology, Udur. I am here to present my abstract on gurasa, which is traditional food produced locally from wheat. As gurasa is an indigenous bread that is generally produced in northern part of Nigeria and is normally made from wheat flour. And the present study is seek to determine the effect of soybeans flour addition on the quality of this gurasa. As gurasa consumption cross all the age and it serves as a vehicle for nutritional well-being of the people. But I come up with the idea of incorporating a low-cost legumes, which is very available in Nigeria with a very cheap price compared to the wheat, which has a very high price. And in Nigeria, we grow a wheat in a very minor amount. And due to the high importation of the wheat, I come up with the idea of producing this gurasa with a locally produced crop, and it's good in Nigeria to grow at a very low price to substitute the level of flour by the addition of soya beans. And also, the improvement of this nutritional content of the gurasa cannot be overemphasized, which is high problem of malnutrition and high cost of importation of the wheat into the country. Next slide. On the findings, the increased level of soya beans increase the proximate content of the sample in terms of the protein, fat, crude fiber, and also consequently the nutritional value of the gurasa bread. Also, I come up with the idea of using the animals, which I use the albino rats, to feed them with the gurasa for at least 3 weeks to see the effect of their changes in terms of the body weight gains and also as well as other biochemical analysis. And finally, I found out that there is no negative effect on their health. Here is the table 1 containing the proximate compositions of the gurasa by the substitution of the soya beans flour. I measure the moisture content as the water activities, the ash, protein, the fat, the fiber, carbohydrate, and the energy in ton per 100 grams. Here is the formulation of the sample, which is 100 to 0 control, which is the normally gurasa is produced from a wheat flour, and I produce the gurasa with 100% control. And also I use the substitutions here, the 90% wheat, 5% soya beans, up to 50-50. That's the 50% of the wheat and also 50% of the soya beans. And I found out that there is increase in all the proximal composition based on the increasing of the substitution of the soya beans to show that protein is more higher in the 50, where there is more addition of soya beans, and also it is acceptable by the sensory panel. And also in terms of the body weight gain of the trial albino rats I use to feed them with the product. Based on the formulation from A to G, based on the substitution levels of the soya beans, I subjected the animals the 50 grams of the sample, and I measured the essential weight, the initial weight of the animals before, and also the final weight, and also the body weight change of the animals after the substitution, which show that the more substitutions, it increase the weight of the animals. And also I find out that there is no negative effect on the growth of the animals. And also finally, in conclusion, here is the picture of gurasa, which in Arabian call it flat bread, and I think in India they also call it flat bread, and in Nigeria we call it gurasa, which is common food for all ages in Nigeria, and it is affordable to all age in Nigeria. And even last time, as a result of high importation of the wheat in Nigeria, these people that produce gurasa, they go on strike. And finally, the result is establishing the addition of soya beans in the gurasa improves the nutritional profile and also making it potential good source of nutrient. And also it serves as a vehicle to prevent malnutrition across the children, because this gurasa is being used across the age. And also finally, there is no negative effect on the clinical nutrition indicator based on the animal feed. Here are some of the references. Thank you for listening.
Okay. The session is open for any questions. Does anyone have any questions? They can write in the chat box, please.
Yeah, one question, please. Yeah. See,
Yes, sir.
purasa is a flour, a blend of wheat and soybean. In your finding, you found that when it was fed to the rats, the intake is reducing. The percentage of soy flour increase when you increase in the blend, the intake of food is, then how the nutritional requirement will be met by the requirement for nutritional requirement. When food intake is reducing by the incorporation of 10% or 20%, then nutritional requirement will be affected.
Ma'am, should we wait or?
No, we can wind up now.
We can wind up.
Wind up and we'll send the
Okay,
question
fine.
to him.
Okay, ma'am.
Question
Okay.
to him. Yes.
Okay. So we came towards the end of the session, and I asked the coordinator of the session to kindly sum up the theme of today's session.
Thank you, ma'am. So we had wonderful presentations in this technical session 3. I thank all the esteemed speakers for this session. Dr. Florence Eagle talked about the role of food science and technology in food system transformation. Sorry. She also conveyed that we need to transform food to ensure that the food security addresses hunger and malnutrition. Some of the points she enlisted are limit the food wastage, connect to nature, make good choices, eat seasonal fruits. And she also discussed beautifully about humanitarian food science and technology. After that, there was very beautiful presentation by Dr. Karuna. She discussed about millets as a smart food and a sustainable diet, which is a neglected ancient cereal grain. The key highlights of her presentation were hidden hunger and malnutrition. She discussed about food culture today, nutritional diversity and nutritional composition and bioavailability of millets, health beneficial effects of millets, and she also talked about how the glory of millets can be brought back. Then after that, we had very informative session by Professor Joy Regenstein on total utilization of fish byproducts. He discussed about the waste audits, major components of fish waste, the fish rendering products. Very nicely, he has explained that the components which are normally discarded as waste can be utilized very efficiently. So with this, I thank all the invited speakers. And after that, in the second part of this session, we had 4 oral presentations, which were also very informative. Thank you. And now I request Dr. Sunayan to propose a vote of thanks.
Thank you, ma'am. Thank you all the respected speakers and also been accepting our invitation to join us for the sessions. Every session has been very informative. And I hope that every participant who has been there and present in this session have gained something. And we look forward for the day 2 of AMIFOST, 5th AMIFOST 2022. Thank you, everyone. Have a good evening. Thank you, sir. Now we can close the session, ma'am.
Yes.
Thanks once again to Dr. Florence and Dr. Rengston and Dr. Karun also.
Thank you.
Yes, sir. Sir.
Yeah, it's okay. Thank you very much. Thank you.
Thank you, everyone.
Yes, Dr. Karuna.
Thank you, sir.
Dr. Karuna, we miss you.
So I too.
Only one. Thank you.
Thank you, sir. Thank you, sir.
I
miss you too. I miss you all, my team. Thank you, sir.
Yes, very much.